a senior person, G. Murali Krishna Garu, former head of the department and chief scientist, CFTRI Mysore. So these are the three important lectures which we are going to have in the morning session. At the outset, to present the biodata of the first speaker, Professor Roman Reddy Ganta Garu, I am glad to invite one of the chairpersons of today's session, Dr. P. Suryanarayana, presently working as a scientist at ICMR National Institute of Nutrition, Hyderabad. I invite him onto the online platform, and just to introduce him, Suryanarayana is a very humble and very good person, gentleman. He is an immediate junior to me, and he did his MSc in our department during the year 1992 and completed PhD in 2004 with Professor Ramana as his guide. After completing his PhD, Suryanarayana joined at National Institute of Nutrition as a research officer, and then he is elevated into scientist F position by in different stages. Suryanarayana received two awards. One is the Memorial Donation Award. Apart from that, we have few fellowships, and in addition to that, Suryanarayana published a good number of papers, and he is actively involved in nutritional biochemistry research, mostly secondary complications related to diabetes, cataract, retinopathy, and so on and so forth. So he is also members, and I am happy to invite Professor Suryanarayana to read the biodata and present the Professor Roman Ganta to the August Gallery online. Thank you. Please, Suryanarayana. Suryanarayana, you are there?
Yeah, he's there.
Yeah. Please introduce the speaker.
He's on mute.
Yeah, you're on mute. You're on mute, Suryanarayana. Unmute.
Am I audible, sir, now?
Very good.
Yes. Thank you, sir. Good morning and welcome to the day 2 morning session. So it's my pleasure to introduce our first speaker of the morning session, Dr. Roman Reddy Ganta. Sir is the professor and director, Center of Excellence for the Western-born Disease, Department of Diagnostic Medicine and Pathology, College of Veterinary Medicine, Kansas State University, Manhattan, Kansas State, USA. Dr. Ganta has over 3 decades of research experience focused on important human and animal vector contributions. He has internationally well-recognized research program focused on several emerging tick-borne infectious diseases of global public health as well as animal public health importance. His major area of research is primarily on tick-borne rickettsiosis diseases caused by Escherichia species, Anaplasma species, and Rickettsiosis rickettis. Dr. Ganta generated over 20 million US dollar research support from various competitive funding agents since 1998. boards of several peer-reviewed scientific journals. With this brief introduction, I invite Dr. Roman Reddy Ganta to deliver his talk on tick-borne rickettsial disease research program, building the puzzle from the research to application. Uh, Over to you, sir.
Thank you, sir. Let me see. Can you see?
Yes, sir.
Well, thank you very much. And first of all, before I go into my research, as you can see from this title, there's there's a lot of information probably new to a lot of you, many of you. But before I go, I would like to talk about our department. Just a minute, okay? Can you see this department entrance? Most of you familiar. We all walked on these steps, just 3 steps, but the 3 decades of lifetime accomplishments could only be possible because of all the basic training that I got, and many of us here and many more who are not here got at biochemistry department. Um, so this, basically, this is the foundation stone for where I am today. For that, I am definitely thankful to the biochemistry department and the faculty who valued the education and giving the best in terms of training. And the biochemistry is a very unique field, which gives opportunity for those who get trained to be in various different disciplines: molecular biology, immunology, vaccine studies, biotechnology, you name it, anything that is possible with basic training. I think that my research presentation hopefully will illustrate that. Okay, if you go into the title of my presentation, it says Tick-borne rickets cell disease research program, building the puzzle from research to application. So I know some of you may not be knowing what is tick-borne, what are ricket cell diseases. And so since this is 20 plus years of research, so I don't call this as a research presentation, rather research program presentation. How we evolved or how we took advantage of the basic research in developing ways to translational research, so applied knowledge to control the diseases. All this story I will try to tell through this presentation. So in brief, the outline of today's talk will have the brief description of the importance of vector-borne diseases, in particular tick-borne diseases. Some of you know, some of you it's a new subject. And our research goals over the last 2.5 decades. And then progress from basic to applied research and how we developed the basic research knowledge into prevention methods. Specifically, I will discuss today mutagenesis. So many of you might know what is mutagenesis. It's in a way by which you can manipulate a certain regions of the genome to alter so that you can really investigate the importance of a gene or genomic segment. That's basically mutagenesis. So how we develop that application for the pathogen that we work is what I'm going to discuss. And more importantly, some of the molecular biologists, mutagenesis seems very straightforward. What is it so challenging? But indeed, it's very challenging if you work with obligate pathogens, such as obligate intracellular bacteria, which are the primary research area that we work on. And I will tell you how we overcome those challenges and how we use that knowledge in applying, in defining various genes and genomic regions for the bacterial in vivo growth, and how we are now translating that research into other very basic research questions that we are asking, such as the protein-protein interactions, how the DNA regulation is occurring, I mean, like the gene regulation is accomplished, and so various means by which how the pathogens escape. All this information we could take advantage by simply using one major molecular method, which is mutagenesis. So ultimate goal for any research to me, whether it's by our own research program or by others, it has to translate into some good outcomes. So good outcomes for a disease is the way of treating a disease or coming up with the good drugs that we can be controlled or have a vaccine that can prevent the disease. So this research definitely gets us there. That's why we have been quite successful in getting federal funding to continue our research, and I will describe that. So in this presentation, there will be a lot of new terms that I will be introducing, and a lot of research. It's literally like a roller coaster. You go from one area to another area. So wherever possible, I will try to simply explain in a simple way, and some areas that maybe you may have to go back and read if there is, and at the end, if you're interested for me to explain, I'll be very happy to explain. So let's talk from the vector-borne diseases. Most of you know vector-borne diseases account for more human disease and death over the centuries. This is the Centers for Disease Control and Prevention statement saying that they cause more disease than all other causes combined. But if you go back to ancient times to even probably in the future, vector-borne diseases continue to play an important role. When we talk about vector-borne diseases, there are a lot of different factors which can cause diseases in people as well as various animals. So most notably, everyone in India, I'm sure, knows about mosquito-borne illnesses like malaria, filariasis, chikungunya, dengue fever, all kinds of things. But that's not the only vector that we are talking about, and there are many other vectors. One of them is referred as tick. So the ticks are more challenging, and they are second only to the mosquitoes in terms of their impact, and their impact is even more and more in recent times, which you will understand when I present this. Then I will also be discussing about rickettsial diseases. So rickettsial diseases are caused by a bacteria. It's a gram-negative bacteria called Rickettsiaceae family bacteria. So what are rickettsial diseases? Basically are caused by this obligate intracellular bacteria. If you see this, what I'm moving around, these are the bacteria inside the phagosomes within the host cell. You may not have heard, or maybe some of you may know in the microbiology class, but everyone knows about another bacteria which pretty much is the reason why we breathe today and all the eukaryotes, which is mitochondria. So mitochondria is not new, right? So here is the mitochondria. If you do the phylogenetic relationship, basically all the rickettsial pathogens, here is the Rickettsia prowazekii and Oriensis etsugamishi, Ehrlichia schaeffiensis, Caudiria ruminantium. All these pathogens are pretty much close cousin to mitochondria. So that means these are mitochondria are endosymbiotic bacteria, whereas Rickettsia are pathogenic bacteria. If you look at the life cycle of a bacteria for a microbiologist, they may be free living or obligate, intracellular or extracellular pathogens, or endosymbiotic. So it depends on what is the host and pathogen relationship leads to. So in this case, the pathogens cause serious disease, so they are pathogenic bacteria. Now let's move into tick-borne diseases. So there are a lot of tick-borne diseases. If you look at, there are 4 columns I put, names of the diseases on the left. The next one is the etiological agents. The third one is the tick vectors, means what kind of tick, like malaria, mosquitoes like amblyomma, not amblyomma, Anopheles mosquitoes, Culex mosquitoes, like that. There are different tick species that can cause the pathogens to be transmitted. If you look at the far right, these are the hosts that are involved in getting the sickness. So let's go from left to right again. All the blue ones are the disease areas that we work on. These are all rickettsial diseases. And so there are up to here, these are all bacterial pathogens. And so this Babesia refers to something like a malaria parasite, epicomplex and protozoan. And the rest of the bottom here are viral pathogens. So there is a long list of bacterial, parasitic, and viral diseases or viral pathogens are transmitted by ticks. So if you look at the ticks, I just the highlighted like orange color here, Amblyomma americanum, what's one of the names of the ectoparasite tick. So if you look here, the same tick is the host for several of these pathogens, Ehrlichia scheffiensis, Anaplasma, Ehrlichia ewingi, and Rickettsia, Francisella tularensis, like that. So now you ask a question, how come only one tick can carry so many different pathogens? So that requires the understanding of how the life cycle of the ticks take place. So let's hold on to that thought for a moment. Then now move to the right. If you see here, hosts which are affected: humans, dogs, goats, and coyotes by this particular pathogen. Similarly, Oxodis species vectors transmit Anaplasma phagocytophilum. So there are 4 different hosts. So what that is telling, so these pathogens do not really have a specific host requirement. Technically they can be causing disease in any other host. So what that means is that the pathogens are obviously transferred from these ticks. So that definitely connects you. That means the ticks can feed on not just in these hosts, they can also feed on any hosts where they can get the blood mail, because these are ectoparasites. These are blood sucking parasites, just like mosquitoes, ectoparasites. So that means the multiple pathogens are transmitted by same vector ticks and also into multiple hosts. So therefore which requires the understanding of the life cycle of ticks. If you look here, here is the one of the ticks called Amblyomma americanum. The life cycle starts with the eggs are dropped by the female, which molt to once they hatched, small tick becomes larva. That larval stage feeds on number of different hosts for complete blood meal. These are not like mosquitoes. These ticks can be on a host for several hours to several days. So larva might feed for several hours, and then they molt to the next stage called nymph. The nymph again needs a blood meal. They feed on a different hosts, any one of these hosts that they can get, and then they progress again to adult stages, male and female. So they can once again feed on these different hosts where mating can take place, and the female lays large number of eggs, like up to 5,000 to 10,000 depending on the tick species. If you look here, this is the adult tick, very small, and put that compared to this larva and nymph are much smaller. So the pathogens, now you ask the question and everything adds up very well. When a tick can feed on number of different hosts, when pathogen is circulating in any of these hosts, the pathogens can be easily acquired by this larval stage tick, for example, and when they progress to the nymphal stage, the pathogen still progresses to that, and then they pass the pathogen to the next stage. So in this case, if humans acquire that pathogen from that tick, then humans can get the disease. Similarly, at the adult stages, humans, dogs, or deer, or cattle, whichever. So pathogens have a lot of opportunity to go through a number of different hosts, and likewise, this ectoparasite tick has a lot of opportunity to seek the blood meal. So this is a really complex phenomena where evolutionary hotbed, lot is going on, and a lot of pathogens can be acquired and passed on. So which makes it very complicated and very. One of the major funding agencies came up with a strategic plan to control tick-borne diseases. It's in about a couple of years ago. It's very straightforward. The plan is to improve the fundamental knowledge about tick-borne diseases, then to advance the research in developing better diagnostic methods, and accelerate the research to prevention, in this case prevention meaning developing vaccines, and support research in advanced treatment, so new drugs, because not many drug options are there, and also to come up with new tools and resources to advance tick-borne disease research program. So all these ideas were collectively thought as the great way to solve the tick-borne disease research program. And this is not just the NIH, Department of Defense, US Department of Agriculture, and various funding agencies feel the same. Why? Because military personnel travel to all parts of the world where they get the risk of getting the tick-borne diseases. Similarly, animals like food animals like cattle, sheep, goat, they get the tick-borne diseases, and which causes the economic losses, major economic losses. and companion animals like dogs and cats. So tick-borne diseases have a major problem, which needs to be tackled because, like I was explaining to you, it's a very complex story to deal with. It's not very straightforward because these organisms have a different way of surviving in these dual hosts, invertebrate hosts, vertebrate hosts, escaping the host immunity. So one must know how these organisms are sensing the host immune responses, how they are escaping, what are the unique strategies that they employ in evading the host response. If we don't know that kind of a fundamental knowledge, there is no way that we can solve this problem. Well, this is what NIH felt 2 years ago, but that's exactly the vision we had 20, 30 years ago. That's why I was able to convince our university that we need to start a center of excellence for vector-borne diseases with a primary focus on tick-borne diseases. And we have been carrying out very similar research, and we have been obviously working on fundamental knowledge, diagnostic tests, vaccines, and treatment-related research, as well as in advancing new tools. In this case, I mentioned about mutagenesis experiments. So we had the exact same vision that NIH had for the recent announcement. So now, if we come to rickettsial diseases, these are, like I said, is the gram-negative alpha proteobacteria. They can be subclassified into two families, Anaplasmataceae and Rickettsiaceae. Very simple way to put it, the Rickettsiaceae are the intracellular, but they escape from the phagosomes into the cytoplasm, whereas Anaplasmataceae pathogens, they reside inside the phagosomes. We have been working 20 plus years on Ehrlichia with a basic research because it was discovered in 1987 as the human pathogen. And if you look here, it infects macrophages. Similarly, other pathogens infect phagocytic cells like macrophages and neutrophils. And so there are other exceptions where, like Bun Hl, which is only cattle pathogen, infects erythrocytes, whereas ruminantium infects endothelium. Similarly, Rickettsia infects vascular endothelium. So you have to ask a question. The purpose of a macrophage, the purpose of a granulocyte, neutrophils, is to fight the infection and clear the bacteria. That doesn't happen. So in other words, it is like somebody is going right in the middle of a battlefield and ready to fight, but they know exactly where the bombs are, where to defuse, things like that. So they can easily, comfortably live there. That's exactly what these organisms do. So that means there are unique ways that they not only impact the innate immune system, but also acquired immunity. So even biologically, it's very unique to understand what happens about these organisms. So our research has been actively ongoing in all these organisms except this relatively new pathogen. So we have current funding and basic research on these organisms, and then to develop vaccines for 1, 2, 3, 4, 5 different pathogens. So before you go more into before I progress more, I want to explain you how these bacteria replicate in a host. Here is this has two different forms. One is called infectious form, which is also called elementary body, which is small and goes inside and replicates inside the phagosomes. If you look around, these are all the mitochondria. Once these replicating forms reach to certain number of the organisms, they will be escaped out from the cells by exocytosis or complete cell lysis, while some bacteria are still replicating inside the phagosomes. So this is a complex life cycle. So you can very well imagine that these different forms require different set of gene expression strategies. So dense core forms versus reticulate forms. Our initial research, we identified that these bacteria in the two different hosts, like macrophages to represent the humans and other vertebrate hosts, and the tick cells representing the tick host. So the protein expression is substantially different. And similarly, this is a different experimental design, in this case, total protein fractionation, but here is the mass spec analysis. So the dense core cells If you compare with dense core cells to dense core, there is a significant overlap, saying that the proteins expressed in the dense core cells are similar to each other, similarly reticulate cells. But if you compare the dense core cell protein expression to the reticulate cell protein expression, there is no overlap. Just like you see the bacterial protein expression differences in vertebrate host cells versus the tick host cells. So that means the organisms know exactly uh, how to change its protein expression specific to different hosts, and also to alter its protein expression for their benefit in the times when they are replicating, in the times where when they are infecting. So all that information is very critical for us to build the puzzle from the basic research to application. So in other words, to study the understanding of any vector-borne disease or tick-borne disease, as many of you know, even the malaria, it's not very easy to develop a vaccine. Why is that? Because of the dual host and unique things that these organisms do. Same holds true. So in other words, if you want to study and come up with good solutions, it's not just sufficient to just take these organisms, grow them in the cultures, or take some animal like a mouse model or something, or guinea pig model, and study that. It's not, because it's not going to tell the whole story. So what is important? What happens this organism when they're replicating inside the its natural vector ticks, and also its natural vertebrate host. It's called reservoir hosts, where the pathogen constantly maintains in nature, versus the incidental hosts like humans and dogs. The disease causes, the pathogen causes severe disease. So that means investigations have to take into consideration what happens in terms of pathogenesis, immune evasion mechanisms, and the gene expression changes, and host sensing. All these are critically important. So to study the pathogenesis, you need to put all these together. Host pathogen interactions obviously require the hosts, and then define immune evasion mechanisms, and then define differential gene expression, and then how the bacteria regulates its gene expression, and then ultimately use the knowledge to vaccine development. So what I'm going to tell you is all these different directions can be much more accomplished by using one molecular biology technique, which is mutagenesis. So why? Because you can take one gene at a time. You can break them down if you want, or multiple genes, and then I assess the importance of those genes or any of these, and whether or not we can develop vaccines at the end. So I mentioned at the very early on, mutagenesis is a very challenging subject for intracellular bacteria. You might ask why. First of all, to disrupt and restore, it's called is a very fundamental, because it helps us understand which genes are critical for the bacteria, and again to define better ways of controlling or better targets for drugs, etc. So one of the major challenges for obligate intracellular bacteria, like you know that in mitochondria there are very few genes left, like that there is a reason we call it as an obligate intracellular or pathogenic bacteria, because they don't have the full set of genes. It lost some genes. It has to depend on a host for its survival, for some of its support. So what that means is it only keeps the essential genes. There are a lot of genes which are non-essential or literally gone from the pathogen. So if you put that to mutagenesis, if you try to randomly knock down or specifically target a gene to break it down, that bacteria will be dead. So that means the outcome is going to be lethal. So there is no way you can tell that this gene is essential or the bacteria died because of the manipulations. So that makes it very complicated. So other challenging is that because they live inside the host cell support, the moment you take them out of the host cell, bacteria rapidly die. So that means you have a very micro window, very small window where you can do the manipulations before they went back into the host cells for their replication. Other complication is that these bacteria, particularly the rickettsial bacteria, most of them that we investigate, they do not have their own natural plasmids. So what are plasmids? Plasmids are extracellular, I mean the extra chromosomal DNA which replicates inside the bacteria, and often they carry antibiotic resistance cassettes and various things. So in molecular genetic engineering, molecular biologists typically use those to manipulate certain genes and knock down and so on. Because they don't have natural plasmids, we don't have that luxury to use the bacterial plasmids to do our research. So with all those complex challenges, we have carefully designed research experiments, but successfully came up with a solution to develop targeted mutagenesis. So our initial efforts for targeted mutagenesis are that we thought logical approach. If you select a gene, genomic region where it's called intergenic spacer. So intergenic spacer means between gene 1 and between gene 2. It's less likely that that segment may contribute a role. So therefore, if you create a mutation in that region, we may be able to standardize the protocol, and then subsequently we can apply it to other experiments. Similarly, we selected some silent genes. Silent genes meaning a gene X may be expressed in vertebrate hosts but not in the tick host. So like the differentially expressed genes are the genes which then don't seem to be expressed in both places. So some we call them pseudo genes or silent genes. So there are a lot of ways. So the long story short, when we took all these approaches, we are able to demonstrate targeted mutagenesis, and yet they were only short-lived, like in this number 6 refers to 6 days. After 6-7 days, pathogens are dying with those mutations. So we thought there may be something more to it. What is the best way to come up with a good solution to such a problem? So then we reasoned, how about let's go and create a random mutations in the genome and see which genes are identified as non-essential or maybe less detrimental for the bacteria, and then use that targets to define the targeted mutagenesis methods and then apply them to the next. So that's exactly what we have done. So we came up with HIMAR1, which is one of the transposase system. We created that construct. This construct basically expresses mCherry, which is like a red fluorescence protein, and antibiotic resistance cassette. But HIMAR1 has one unique way. It recognizes left and right repeats, and this segment from this plasmid can be integrated anywhere within the genome of a bacteria where A and T are side by side. And the probability of A and T in any genome is 4 to the power 2, 4 times, so 16. So every 16 nucleotides you expect to see A and T randomly. So the chances are that we are bound to get some mutations in the genome of Ehrlichia. So that's exactly what we have done, and indeed we created about 60 different mutations. That's what is listed. Each spot represents, this is a Southern blot experiment, DNA blot analysis, to show every time there is a mutation, you see one spot. That's exactly what you're seeing here. So when we mapped them to the genome by genome walking experiments, we identified that it's not a challenge to create mutations in Ehrlichia species. So as it's illustrated here, we could create almost 60 mutations, and almost 60% of them are within the open reading frame, meaning within the gene coding sequences. And the remaining 44% are in the non-coding sequences. So what that means is that mutations can be anywhere in the genome. So these are all the non-essential targets, at least under the in vitro cultures. If they are non-essential for in vitro culture growth, what good such mutations for understanding the biology? So then we said, well, maybe they do when we take them to the in vivo situations. That's exactly what we have done. So how do we do the in vivo research? So we developed animal models. In this case, this is the deer, which is a natural host for this pathogen. So we created the mutations, and here are the mutant organisms. We put them back into the host cells, and then we inoculated them into the animals with this animal model. If the gene is essential, what you expect to see is the bacteria, you the mutant bacteria, you don't see them subsequently when we screen them. If the gene is non-essential, meaning if you create mutation, the bacteria should survive. So what in other words, we're looking for negative selection. If a bacterial gene is essential, they clear fairly rapidly. If it's non-essential, they stay on. So because naturally these organisms have the ability to survive and maintain infection constantly in a host. So to represent incidental host, like in humans, we developed an animal model with a dog. So essentially the same outcome. At the end, what we are doing here is we're putting the animal, allowing the ticks to feed on. What that happens is those are survived in the animal, maybe still picked up. So by the ticks, what that means is those genes are also non-essential for the ticks. On the contrary, you don't see them in the tick. That means those mutations may be essential for the tick, but that does not tell whether what is important for the vector. So in this case, tick. So for that, we came up with a new animal model where we obtain the ticks, we allow them to feed completely on an animal, and then they are technically, if you allow them for incubating for 3 weeks, they will molt to adult stages. But before they are molted, we need to inoculate various mutants in this tick to allow them to molt to the adult stages. If the mutant is, the gene is essential, then you don't see them molting into the adult. Gene is non-essential, you will see them progress. So that's how you screen for the presence of which genes are critical for the bacteria for their survival in the tick host, and which genes are critical for the bacteria for the vertebrate host. So with those approaches, we have identified a number of different genes as essential for the bacterial growth, some in the ticks, some in the vertebrate host. Here is the list of all the bacterial genes which are essential for the vertebrate hosts. If you look here, there are several membrane associated proteins, and some are protein synthesis assembly. One is a multi-drug resistance efflux pump. Some are biotin, heme biosynthesis, and like that. So it's quickly telling us which genes are important for the bacteria. So immediately we know that. So that means if you manipulate the bacterial protein synthesis genes, obviously you can imagine that they will not do good. They don't survive very well. And so same like biotin synthesis. So that means biotin is essential for them. So if you are manipulating the genes involved in biotin biosynthesis, bacteria will die. So what we have done, okay, this is a great basic knowledge. What do we do with it? So we selected 3 genes, like these 3 membrane associated, because okay, since they are on the membrane, meaning this outer surface of the bacterium, so if you create the mutation, so they are not surviving in a host, so that means these are essential for some good reason. So if you look here, the first one, we don't have any way of knowing what exactly that protein does, but the other 2 we do know. One is encoding for pH homeostasis. So what is pH homeostasis? So to help maintain the pH of the organism, bacteria inside the phagosome. Phagosome has a pH more close to acidic pH, as you know, more close to pH 5. So that means intracellular pH for the bacterium cannot be pH 5 unless it has ways of surviving. So what it does is it has to have a active pump to pump all the protons out of the bacteria so that it maintains its physiological pH more close to 7. So that's why we thought this particular gene or this particular protein is critical. The next thing we selected is membrane associated porin-like structure protein, which is called phase head to tail connector protein. So it is like some kind of protein that connects the bacteria into the outside. We'll discuss more. First question we asked, what kind of host response exerted with these 3 mutations? Because one of the goals is to develop a vaccine. So when we tested the immune response, we got 3 interesting outcomes. The first mutation caused a very weak host response. In other words, it did not really cause major host immune activation. The second one created a moderate host response. So which you can measure it by many different ways. In the third one, which gave the strong host response. So there is strong activity of B and T cell response. Then we ask the question, which one of these three mutations gave the best protection if we were to use it as a vaccine? First one, surprisingly not, it gave no protection. So we reason that this bacteria clears so fast from the host that host did not get chance even to induce its immune system to be active. The second one gave a moderate host response, but also the protection is moderate, meaning that the bacteria is still surviving, but at a very low level. And the third one gave a strong host response, also gave a complete protection. So we asked the question, what is the scientific basis for these three different outcomes? First, sodium proton antiporter protein, like I mentioned, it helps maintain the pH, intracellular bacterial pH. So we reason that probably there may be other supporting proteins which can complement the function of this particular protein. And then the next question is, what is this phase head to tail connector protein? So first, for that, you need to know what is phase. I will explain those. So one of the approaches, we wanted to see how the host response was very similar in these mutants, which gives the best protection. So this particular one, which is wild type, comparing with the wild type protein expression, they're very similar, which you expect. Same thing with the phase corrector protein mutation. The overlap is very similar, which is what we expect when we compare the replicates. Same is true this one. And then we compared the mutation from the phase protein with the phase the antiporter protein. There is no similarity, and similarly, there is no similarity for this with the wild type. That means this mutation caused so much change in the bacterial phenotype that whatever the mutated bacteria for which the host created an immune response, that's not really recognized when that's not similar to the wild type bacteria. On the contrary, when we use this phase protein mutation, if you look here, it's very similar. The protein phenotype is very similar to wild type. So what that is telling is that when a host sees this mutant bacteria, it induces the immune response. When the wild type bacteria comes, it's very similar to the mutant bacteria. In other words, immune system is fully to activate and deal with the active infection when it comes in wild type. That's only possible with this mutant, but not with this sodium proton antiporter protein. So what could be the reasons? There could be a number of reasons. So then we ask the scientific basis. First of all, the antiporter protein, there is a lot of redundancy. If you look here, all these different gene tags reflect different genes for the antiporter protein. So that means there is a huge redundancy in the Ehrlichia genome, or this bacterial rickettsial bacterial genome. So they help complement. In turns, bacteria also makes a lot of changes to itself, which makes it complicated. So first we tested how the pH homeostasis is maintained. So we came up with the E. coli surrogate system. In other words, we took E. coli. We knocked down E. coli genes because this is called functional complementation experiment. So in that E. coli, we put the Ehrlichia gene one by one with a plasmid. If it complements the E. coli, then E. coli should grow fine under stress conditions due to sodium, in the presence of sodium chloride. When we complement with all these genes one by one, we tested, can that be really complemented or not? And what happens with the mutations? First, the gene 379, the mutation that we discussed, it has a moderate complementation E. coli surrogate system. But on the contrary, at pH 5.5, various other genes inside the Ehrlichia genome, they can complement very well. Whereas the same gene, which did not complement very well at acidic pH, but complemented very well at the basic pH, at pH 8. Similarly, this is gene. So together, what this is telling, that the bacteria has so much of redundancy, some to support the bacterial growth during the infectious form, and the others inside the acidic phagosomes, there are various genes which are contributing to the bacterial survival. So then we asked, what is this phase protein we are talking about? Yes, phase is basically virus. Phase is a virus, bacterial virus. So this is a typical bacterial virus. If you look at this, there is a phase head. There is this tunnel-like shape. It's like this is what it connects to the bacteria. So if this phase infects the bacteria, it can send its own genome into the host cell to cause lysis or whatever damage that it can cause to the bacteria. That's what is basically phase. But not all phases are bad, or not all bacteria can die because of the phase infection. So with time, all these various phases were taken up by the bacteria for their own benefit, which we call it secretory systems. So here is like a type 1, type 2, type 3, type 4, type 6, and so on. So what they do is basically, in other words, the phase genome was integrated as part of the bacterial genome for the benefit of the bacteria in survival inside the host cells, such as people. So in other words, if this is a phase structure, the phase is integrated as part of the bacterial genome. It's like a reverse phase, and instead of getting killed, it can use this reverse phase for its own export and import of macronutrients, micronutrients, and also some kind of proteins called effector proteins. What are effector proteins? Obviously, pathogenic bacteria, to survive, they have to come up with unique ways to alter the host immunity. What they do, they send their certain proteins out from the bacteria into the host cell cytoplasm and also into the host nucleus. That will alter the host gene expression for the immune response. So then bacteria can survive. So there is a huge benefit to have this kind of a phases. That's exactly what bacteria do. So what we have observed is a mutation in a gene, and there are several genes in the same surrounding region. So these are all related to the phase proteins. So this is somewhat similar to the bacterial type 6 secretory system, which was initially reported in Vibrio cholera, but now in about 30 to 40% of all bacteria have those. And the protein that we created mutation, and it is somewhere here in this phase-like structures. It's like head to tail connector protein. So long story short, what we have done is we created a mutation in a gene which may have impacted the bacterial contact with the host cell, so possibly by impacting its exchange of nutrients, uptake of nutrients, or sensing its own effectors. So this, while this research is undergoing, what we have done is we looked at what exactly this particular protein that we are we created mutation has. So if you look at the structure, the secondary predictor structure of the bacterial protein, and compare to one of the typically known bacterial phase protein, and if you superimpose them, they form a ring-like structure. It's like a 13-mer ring structure. It forms a kind of pore through which something can go out and come inside. So exactly what we have defined, and this phase head to tail connector protein seems to be playing an essential role in metal ion homeostasis, zinc and iron. This work was published recently. So this gene promoter region has a number of recognizable sequences which contribute to the zinc and iron binding elements. Not only that, when we take the bacteria, when we grow them, in the absence of zinc or in the absence of iron or ferric chloride or ferrous chloride, so there is a higher activation of the promoter activity. Not only that, all the several genes that we have demonstrated, the wild type compared to the mutant, always the mutant started making more and more of the proteins from all the surrounding genes. So in other words, what is happening is when we create a mutation, it is putting the bacteria into high metabolic flux starts making so much proteins to compensate, but yet the bacteria do not survive very well. It may be because the metal ion acquisition is severely deprived in this mutant bacteria. So all else is same except that the bacteria fails to get these micronutrients, which leads to the bacterial rapid killing, but it also offers great protection against the host immunity and to fight, and so cause serves as a good tool for live attenuated vaccine. So what we have done is we wanted to develop a targeted mutagenesis. We failed to do that. Then we went to random mutagenesis and we identified number of genes. Then we demonstrated that helped us understand at a basic cell biology and biochemistry and molecular biology of the pathogen to define the pathogenesis and helped in our vaccine studies. And then since we know that we can create specific gene mutations, for example, this phase structural protein gene mutation, if we can use the same target and we can develop easily targeted mutagenesis methods, then why you care? Why we have to go to that great lengths in doing that? Then we can simply translate that research to other related pathogens like various Ehrlichia species and Anaplasma species. So that will ultimately give us a way of developing vaccines studies and also pathogens. So in other words, we can come to full circles by taking this basic knowledge and developing targeted mutagenesis and then leading to defining the immunity and vaccine development. That's exactly what we have been doing and what we have done. So how we did that? We selected the same 3 genes for defining the targeted mutagenesis. So we created antibiotic markers with codon optimized, meaning that you can use the codons that are best used by the bacterium, so that even when we manipulate these foreign proteins like gentamicin resistance cassette or other antibiotic cassettes and mCherry and other various proteins that we express can be easily expressed at high levels. And also we carefully selected several promoter segments from the bacteria which are constitutively expressed, like transcription elongation factor or ribosomal proteins or differentially expressed proteins. So we selected a lot of all these promoter segments to drive the expression of all these cassettes, such as these reporter proteins as well as antibiotic cassettes. When we do that, we can easily develop targeted mutagenesis disruption methods. Indeed, that's what we have done. This is a complex molecular structure. If this is a Ehrlichia genome, we are showing here how we created this mutagenesis by introducing this foreign DNA and disrupting a gene of interest. So these are all the various pathways that we go into come up with generating this mutation. And so that is called allelic exchange, because you are taking 2 segments of similar sequences and in between you are putting the foreign DNA to disrupt a gene of interest. And to restore the gene function, so we created another allelic exchange method where we restore the complete gene. So this is called, when you create a mutation, you want to make sure that the mutation is not having off targets. In other words, it's not someplace else creating mutations as well, or polar effects. To prove that, you basically put the mutation back, restore the function, then to demonstrate that mutation that we create is indeed the mutation that has the impactful outcome. So we have successfully developed by careful construct designing and then performing electroporation experiments, screening for the antibiotic resistance, and then verifying by molecular methods such as polymerase chain reaction, DNA sequencing analysis, southern blot analysis, and so on. So here is one of the example constructs how we have created and tested, and here is another restoration construct. So once we have done it, we demonstrated that this mutation is indeed visible and functionally disrupted. We used E. coli system, in this case antiporter protein gene disruption. So the wild type, you expect the protein to be expressed, so you see the transcript here. Mutant we don't expect, so there is no transcript. And the restored mutation, we expect it back again, so we do see the protein being RNA being expressed. And the same we have demonstrated using the protein method. Here we are looking at the protein expression or the culture growth variation. If there is a mutation, the bacterial growth is impacted. Here is the negative control where the mutation caused the E. coli to be deprived. If you put the E. coli gene back, the growth is restored. So when we have done something very similar, the E. coli with the bacterial gene incorporated, functionally complemented, and when we took that, created in this like mutation, again the even though the gene is there, but because it's a mutated version, the protein expression is down. So that's what you are seeing, the culture is activity is down, and we functionally complemented it, so it reverts back to the functionally restoration of the antiporter protein function in E. coli. So with all these protocols, where do we go? What we have done is we extended this research to define targeted disruptions in various genes. For example, stress response regulation. It's one of the ways when the bacteria undergo stress, a lot of proteins get aggregated. That aggregation leads to inactivation, which leads to the bacterial death. So we are investigating that. So what benefit there is that now we can come up with a good drugs to target for this stress regulatory system of the bacteria. And the promoter mapping studies, in other words, to define how a specific gene is expressed. And similarly, DNA protein interactions, like I'm mentioning, how the differential gene expression is accomplished. So if we create the mutation with the DNA binding proteins with the tag, then we can see where the tagged protein is interacting in the genome of the bacteria. And likewise, we have created mutations for the effector proteins which are translocated into the host cells. So again, there are genes that are essential. We cannot really create any mutations to inactivate because bacteria die. In that case, we can create a knockdown mutation effect. It's like knocking down the expression of protein, not completely inactivating. So then vaccine studies to extend. So all this research is currently ongoing. So I will briefly talk about one of these same essential gene, how we extended the research and how we have developed a vaccine for one of the pathogens. So here is the knockdown effect. There is a multi-gene locus that we investigate. There are differentially expressed in vertebrate host cells versus the ticks. So if this protein, if we create a mutation, the bacteria is going to die. So we investigated without inactivating the whole bacteria, can we really reduce the bacterial protein expression? So how we have done that is use this targeted mutagenesis approach to generate antisense RNA targeted to that specific protein. When it is made, this RNA and this antisense RNA collude with each other, and which causes the reduction of the protein expression. This approach was successfully accomplished. If you look here, the wild type bacteria grow fine. This mutant antisense bacteria growth is less. It's quantifiable. And if the total proteins look same, but specific protein expression is, you look here, there is a significant decline. That's the same for a period of several hours of investigation. What is more is when we have created that antisense bacteria, it is the wild type, there's a lot of bacterial growth. When we have this antisense bacteria, there is growth is significantly impacted. But what is innovative is that that gene knockdown caused a complementation of the expression of the related gene from another protein. It's like this gene called OMP19. When we knocked down, OMP14 started expressing high. Similarly, OMP20 started expressing high. So the bacteria is still complementing, but at least these biological changes, then we can now define with the help of this targeted mutagenesis approach. So how we took the advantage of this mutagenesis approach in vaccine development is, so we have been investigating 4 different pathogens. This particular pathogen causes infections primarily in dogs and also in people. And this particular pathogen infects, it's an important foreign animal disease pathogen, infects cattle and goats. And the third one, which is Anaplasma phagocytophilum, which infects humans and horses, cattle, and various different hosts and the dogs. And Anaplasma marginal, which is an important food animal pathogen, meaning that infects cattle throughout the world, including in India. So what we have done first, we identified whether the gene homologs are present for this phase head to tail connector protein. The answer is yes. This whole cassette of the genes, which are phase related proteins, and the genes are extensively conserved. And the next thing we did is create mutations on these similar genes by using the targeted mutagenesis approach. Let us talk about one of these. It's called bovine anaplasmosis pathogen, which causes infections in cattle. So here is the mutated version of the bacteria, which we confirm. For now, I will ignore the right panel because we do not discuss that. And what is bovine anaplasmosis? Bovine anaplasmosis is caused by the pathogen called Anaplasma marginal. This is another pathogen belong to the rickettsial pathogen. It's a major economically important disease in livestock throughout the world, including India, US, and various countries in South America, various countries in Africa and Europe, etc. So it can cause high mortalities in the animals and causes significant drop in milk and meat production, and causes abortions and fertility problems and so on. With this, what we have done is we created a targeted mutation in that the gene that we identified as providing the best protection in the canine host experiments in previous studies in the phase protein. And then we tested that mutation, how can that be helpful in vaccine development. Here is the clinical disease when we infect the animals with the pathogen. So there are several ways to measure. One of the measures is that since it infects erythrocytes, it's the packed cell volume, which refers to the number of red blood cells present. With time, the red blood cells drops quite dramatically, more than 50% decline, and which causes the animal to be severely anemic and die within a short span of time. And we tested the inactivated antigen as an example. The bacteria still replicated fine and the disease still progressed. On the contrary, when we created the mutant bacteria, which has the mutation, which gave the bacteria still replicate a little bit, but then they disappeared fairly quickly, but host induced a strong immune protection, which prevented the red blood cell breakdown. So basically within the healthy range that we have observed. And similarly, when we looked at the bacterial circulation, the severe disease, there is a high level of bacteremia, which we can see in the systemic infection in the blood. And the same is true when we use a different version of the vaccine. But on the contrary, if we create the live attenuated vaccine, the mutation basically caused the bacteria to clear and it gave the complete protection against the infection challenge. So I have taken you through the Hughes scientific ride, explaining you about ticks and tick-borne diseases and how we have developed various molecular tools and how we have used those molecular tools in understanding the basic biochemistry and biology of the organism and pathogenesis and genes essential for the bacteria, and how we are using those mutagenesis approaches to define and develop new vaccines. So this research obviously cannot be accomplished by one person. We have several funded grants from National Institutes of Health, and we have been continuously funded from 2002, and research is still ongoing with 3 active projects. And we also have our center foundation funds that we generated, which also support, and some funds also coming from the industry or endowments. And there are a lot of collaborators, but what is more are all these people worked in the past, and these are the long list of young and active investigators in my program. Typically in a given time we have anywhere from 12 to 14 people working in our research program. So without all their hard work, there is no way that what I discussed today could have been easily accomplished. So with that, I'll be very happy to talk to you if you have any more questions.
So now this session is open for discussion. If you have any questions from audience.
Suryanarayana, can I ask a question? This is Shivakumar from ISA Tirupati.
Sure, Shiva. Just you can proceed.
Professor Ganta, it was fabulous work. I really enjoyed listening to your work.
Yes.
A lot of molecular biology.
Yes, sir.
One naive question. I joined late. So, if I understood you correctly, you are trying to bypass the need for new antibiotics for the antibiotic resistance strains and develop these mutant strains as vaccines. Is that right?
Yes, it's live attenuated vaccine.
Right, yeah. So, I mean, the mutant genes, are they conserved across different strains?
The genes are conserved. That's exactly what I showed you in the slides.
So the vaccine developed so it would be helpful to kind of protect against across the strains, or is it strain specific?
Oh, that is a very good question. Yeah, you're asking if there is a strain of this bacterium, what vaccine that we created, can it protect against other strains?
If the gene mutated is similar to
the gene is mutated similar, but strains may be different. So the genes which are here, these are very much similar, but the strains may be different. But here is the question.
Yeah.
For example, the bacterium that I just talked about, which is the bovine anaplasmosis. Previously, in southern Africa, there are vaccines in market. That vaccine is basically Anaplasma central. It's a different species altogether. Live infection, they put it into the animals. They give a protection against this different species. So that means even though the species is different, it still gives the protection. So my scientific reason is, even because they created the mutation is from the same species, not even a different species, but it gives complete set of exposure for the bacteria to the host. So host induces the strong immune response. So it is likely that it gives the complete protection against widely different strains. The scientific basis is because the heterologous species can protect, obviously homologous species, but a virulent form can easily give the best protection. So my hypothesis is that it protects, but the research has to be done.
Yeah, one more related question is, now that you identified some new essential genes by your mutagenesis screen, would you be also interested in developing drugs that would specifically block those genes?
Yes, that's
come up with new antibiotics. Yeah,
exactly. Yeah, that's the idea. That's why I briefly touched base. ClipB is one of the important genes. We have colleagues in the biochemistry department here we are working with, and what we are trying to do is, because ClipB is one of the proteins involved in the reactivating the aggregated proteins. So the stress leads to protein deaggregation. You're familiar with that, right? So when you to relieve the stress, what you do, this ClipB and DNA, K, and all those proteins make a form. Basically, the proteins are again reaggregated. So from the aggregated form are separated out like spindles. They go through some kind of a channels and take them out as fully functional proteins. But if we target that protein to develop a drugs, because ClipB like proteins are also present in host cells, but the proteins aggregation completely different. So we can develop a good drugs against these bacteria. That's one of the directions. And similarly, as I mentioned, there is a biotin biosynthesis seems to be essential for the bacteria. So we are also looking at that. And so likewise, there is multi-drug resistance protein gene seems to be playing a role. So we can easily come up with the different ways of coming up good drugs. And you might ask, why don't we have a drugs? We only have one antibiotics currently in use for all these rickettsial pathogens, which is a doxycycline, which is a tetracycline derivatives. So we
Right.
definitely need one of the ways is to come up with good drugs. That's one of the goals of the NIH. Yeah, the answer is yes.
Fantastic. Thank you so much.
Thank you.
Is there any questions from the other participants? So if there are no other questions, I have a couple of questions from my side, sir.
Yeah, sure.
Since this is involved in the immune system, is there is any difference the effect of pathogens based on the age based, sir?
These pathogens? Yes.
Different age people, will it be effective the same like what we see in the elderly or in the younger people? So will it be same? The effect will be same?
Yeah, let me take you to one of the slides. That's a very good question. The best I can answer is by showing this slide. And that requires me to share again. Share the screen. Okay, if you look here,
Yes.
these pathogens, like Ehrlichia schiefensis, for example, that infects children and people above 50 or above 60 or more aged groups, the disease can be fatal. Same is true with rickettsia, Rocky Mountain spotted fever, for which we are right now working on the vaccine development. It's really deadly in children, like 6 to 10, up to 10-year-old or younger.
Okay.
But on the contrary, if you go here, Anaplasma marginal, which is another rickettsial pathogen in cattle. But if the cattle 1 year or 6 months or younger, nothing happens to them. 6
Okay.
months to 2 years, there is a moderate disease. 2 years and longer, there is a severe disease. So there is always a pathogen specific variation that
Okay.
hits differently in the different hosts, and different pathogens do different things. So not all else is the same, but that's basically what we know.
Okay. Okay. I have one more question. Is there any role of comorbidities of the particular person? So will affect the disease severity, sir? If the pathogen is affected to diabetic person, so the severity of the pathogen will be the same like as healthy man, or it will be any variation?
No, that's an excellent question, sir. Um, Here is another challenge with these pathogens. These are all bloodborne pathogens, as you can imagine. These are basically their systemic infection, monocytes in the blood, and these are all the neutrophils in the blood, right? And these are like erythrocytes. So it's basically bloodborne pathogen. And
Okay.
people with compromised immunity, like AIDS patients,
Okay.
they die with these diseases more severely. And same people undergoing chemotherapy or people going through the cancer treatments, these are deadly. And you know what is other challenge with these pathogens? People often don't recognize and realize it. Blood transfusions.
Okay.
When people get a blood from a person, if they're not tested, this pathogen is circulating at low level, because often any pathogens, this is like a balance. Initially, the infection raises high, then they come down at very low level. It's virtually not detectable by most of the molecular methods. Even the most sensitive molecular methods, they test negative. Only way you can test positive, it is possible in animals. You take large sum of blood, for example, in cattle, you take 500 ml of blood, you put it in splenectomized cow, steers, then you can see the infection. So that means infection is such a low level, and because these are also intracellular bacteria, they're very difficult to even approached by drugs and antibiotics. But the bacteria is still persisting, and if someone receives a blood transfusion, for whatever is the reason, and the bacteria there and their immune system is down, they get this disease, then they can die.
Okay. Okay.
So there are other problems associated with it. Yeah.
Sure. As the last question, I don't want to bother you further.
No, go ahead.
If a person having hemoglobin like normal, like 13 grams or 12 grams per DL, if the another person having hemoglobin low like 8 or 7, will the infection will be same, like the mode of infection will be same, or there will be any difference in that?
No, the mode of infection is basically tick transmitted or blood transfusion or needle inoculation. It's the same, but
Okay.
people with weakened immunity, this is called, bouncing back. It's opportunity bacteria to grow
Right.
very well. So, like you're saying, if someone has low hemoglobin, the chances are that they're having difficulties breathing, difficulties, jaundice, and other complications because of this severe anemia. And obviously, they don't fight the infection very well. The chances are that this disease can bounce back with a more severe outcomes.
Okay. Okay.
And again, like I said, there are different pathogens with different
Okay.
outcomes, but they all do the same thing. Like this particular disease, like for which we recently got funding, this
Okay.
kills people in 10 days.
Okay.
Yeah, within 2 weeks' time, a person can die with it. It's a
Okay.
very severe disease, and it can kill up to 30 to 40% of the people. And what is challenge, because these are tick transmitted pathogens, often people don't recognize that there is a tick on them, started feeding on them, because they have exposed by simply walking, or maybe they have a dog, they picked up infection and the ticks from someplace sources, and ticks happen to crawl on the people, and then they get the sickness. And by the time they recognize, it may be too late, and often they don't even tell, or doctors even fail to diagnose that, oh, it may be tick-borne disease, and that
Okay.
can lead to serious outcomes,
Okay.
because
Okay.
the death rate can be anywhere from 20% to 80%, very severe.
Okay. Okay. Is there any questions from the other participants?
I have a simple question, Raman sir, Raghav.
చెప్పండి.
Do they have, I mean, do they adopt any methods or approaches to escape immune system like other pathogens, these ticks?
The ticks escaping immune system, you're asking?
Yes.
Oh,
Any-
yes, that's a different subject altogether. Yes, the answer is yes. The
Right.
ticks,
Hmm.
the ticks need to feed on a host, like depending, there are three different stages I mentioned to you, right? Larval stage, nymphal stage, and adult stage.
Right.
All three stages, like larva is too small, it doesn't take much time for it to complete its blood meal, but still stays on a host for several hours.
Right.
Same thing with the nymph. It takes 1 week, and adult it takes 3 weeks. So, for an animal to stay for 3 weeks, obviously they have to do something, right? Because we start sensing and start sensing that there is an ectoparasite feeding on them. So
Yes.
what they do is the ticks release the saliva. With the saliva, there are analgesics, and they change the immune activity, the immune modulators. They change the immune system completely different, and that causes the ectoparasite to feed on the host, and also it gives the benefit to the bacteria to get in. So it's a double-edged sword. It gives the opportunity for the ticks to feed, and at the same time, it gives the opportunity for the pathogens also to escape the immune system.
Right.
So that's another area of research we are not actively pursuing, but other researchers do, and it's one of the areas that NIH is interested in funding that research, and they have been funding also.
Thank you.
Is there any questions from the other participants? If there are no further questions, so I thank Professor Raman Reddy for his excellent presentation. That is very complex, sir, to understand, but you presented systematically in such a way so the new area people can able to understand how your research went from the basics to till the gene mutation until the vaccine development by targeting the genes. This is very excellent presentation. We thank on behalf of the organizing committee on my own behalf. I thank you, sir, very for your excellent presentation, sir. Thank you very much.
Well,
Over
yeah,
to
it is
you,
not
Raghav.
just a couple of years research. This is like a 20 years research. That's
Yes,
why I call
this
this
is
a program.
program.
It's
Yes,
program
yes.
overview.
It's a long way of running. So long from it's began some 30 years back to it's still running. It's a long way again
Thank
to it's just to run. Over to you, Raghav sir.
Yeah. Thank you, Professor Ganta, Raman Reddy sir, for the wonderful presentation on tick recognition diseases. It's very informative and interesting. As usual, you present in your own style. Thank you for that. And I invite Organizing Secretary Subhashini Devi to speak on this occasion. Subhashini.
Sir, good morning, sir. Thank you very much
you.
and for your excellent presentation. There is a message from Kandimalla Ekambar garu, your classmate. Nice
okay.
work. Yeah, you can see that in chat box.
Oh, okay. Okay.
Sir is saying, nice work, Roman Reddy. I am recollecting the days, SKD days at AU, when we are studying MSc and subsequent meeting in AIMS, when we are doing PhD. Last time we invited Ekambar sir. He gave a message. Maybe he is your classmate.
Yeah, he is. He is. Yeah, look forward to catching up with him. And we are planning to have I don't know, Shivakumar is still there. Shivakumar, we would like to have all of our classmates to meet somewhere in Zoom meeting one of these days. And actually, next week I'll be meeting two of my classmates, Nalini and Vijay Kishore. But what is more,
Sir, actually, if you are free,
say that again.
please
Yeah, I'll be
continue.
visiting them I'll be meeting them in Chicago. But what I want to tell you that our group, our batch of 13 students, I think, as I know, at least 12 of them, I think they have reached good heights. Everybody has been quite successful, and either they have become a faculty members or they're contributing to the industry. I know Shivakumar is in India is doing extremely well, and so like that. All that is only possible because of the outstanding training we got at the biochemistry department, for which we are always proud and we are always thankful. And I believe that the same tradition is still continued. The good teaching is the pillar of the Andhra University contribution to young generations. I think that is something the new generation should take it, besides all the active things that you are all doing. For this, I'm definitely thankful to all of you for that. And thanks for calling me every time there is a chance for me to come and associate with you. Of course, I will always come anytime there is an opportunity.
Yeah, I just want to say a couple of words about Roman Ganta. Not only your science is excellent, your words are also excellent. Thank you.
Thank you.
Come on, that's right. Yeah, I think I see Shivaprasad sir on the line. Sir, Shivaprasad sir, you are there? Shivaprasad sir?
Is that D. Shivaprasad or A. Shivaprasad?
Yeah, D. Shivaprasad sir. Okay,
So-
he's on mute.
Oh, easy. Okay. From UK, Leeds.
No, D. Shivaprasad, our teacher.
Okay. Yes, yes.
Shivaprasad. Wait a minute.
Hello. Hello.
Hello, sir.
Yes.
Sir, could you please?
Yeah, I enjoyed listening to Ganta,
Yeah.
Roman Reddy.
Yes.
Wonderful talk. Yeah. Keep on doing this work, Reddy.
Sir.
Thanks a lot. It's very good work. Learning something every time you speak, we learn something new. Good. Very fine. Very good. Great talk. Good.
Thank you very
Thank
much,
you.
sir.
Thank
Thank
you.
you very much. Thank you.
Thank you.
Thank you, sir. Thank you. Let us move on to the next presentation. Once again, I welcome the participants and invitees to the second lecture
Sir.
program. Yeah,
Just
please.
1 minute.
Yeah, please.
Sir, when you visit Andhra University, please collect a Golden Jubilee memento, what we made especially for Golden Jubilee.
I'll be happy to take it, Randy.
Yes. Yes, sir. When you collect and when you come to university, you please collect it, sir.
Definitely.
Thank you very much, sir. Sir, over to Raghavra, sir. Please continue, sir.
Thank you. Definitely the presentation will be an added one to your collection, sir.
Thank you.
Thank you. Once again, I welcome the participants and invitees to the second lecture program of this morning session. To present the biodata of Professor B. Murthy, our next speaker, I invite chairperson of the session, Dr. Sivakumar, onto the online platform. Sivakumar, you are there?
I'm here. Yeah.
Yeah, please. Right.
I'm here. Yeah.
Yeah. Just few words about Sivakumar, chairperson. Dr. Sivakumar is also one of the distinguished alumni of our department. He completed his MSc during the year 1993 and 94. Very active man, very friendly person. His research areas include NF-κB, MAP, signaling, inflammation, immunity, and cancer. And he has vast experience of postdoctoral fellowship. He was postdoctoral fellow 2004 and 2005, Fritz Lippmann Institute, Germany. In fact, he completed his PhD from University of Karlsruhe, Germany. And next, postdoctoral fellow, University of California, USA. And 2009 to 12, research associate, University of Cincinnati. And he was a track assistant professor, University of Cincinnati, 2010-2013. And 2014, he served as a faculty, Louisiana State University, USA. Presently, he is an associate professor, Indian Institute of Science, Education and Research, simply ISET, Tirupati. And he published very good papers in wonderful, reputed journals. A very nice person, a friendly one. And I invite Sivakumar to present biodata of Professor B. Murthy, one of our distinguished alumni. Please over to Sivakumar.
Yeah, thanks very much for the kind invitation, and I'm happy to be here. And thanks for inviting me. I would like to now invite Professor Bhagavathula Murthy. Professor Bhagavathula Murthy is a distinguished professor. He is currently a professor and director at Neonatology Research Program, Baylor College of Medicine. He is also a Kurt Renderath Endowed Chair, Texas Children's Hospital, and a director of BCM Rice Superfund Research Program. Dr. Murthy started his career into biochemistry by doing MSc in biochemistry at our Andhra University in 1981. And then he got selected as a PhD student at Indian Institute of Science, Bangalore. He completed his PhD in 1988 from IISc Bangalore. And those days, I have to say, emphasize on this, those days getting into IISc it means, it indicates his intellect level. No, we don't need to discuss on that. Yeah, then he, after completing his PhD from IISc Bangalore, he did a postdoctoral fellowship at Wisconsin, Medical College of Wisconsin. And then he joined the Department of Pharmacology at Baylor College of Medicine as a research associate, and was also a junior faculty member at Baylor College in the early 1990s. He then joined the section of neonatology at Department of Pediatrics and Baylor College of Medicine, 1995. And Dr. Murthy's laboratory has been investigating the mechanisms of regulation of cytochrome P450 enzymes by hyperoxia. And he relates his findings to hyperoxic mediated lung injury and inflammation. And he's also investigating the mechanisms of regulation of CYP1A, 1B enzymes by polycyclic aromatic hydrocarbons in relation to carcinogenesis. He has been working in the general area of cytochrome P450, drug metabolism and oxidative stress. and he has published enormous number of papers and obtained close to $45 million. That's fantastic research funding. So that clearly indicates how important is his research by simply by looking at his publications and the funding that he received for his program. With this, I would like to invite Professor Bhagawatula Murthy to present his excellent piece of work and a few encouraging words to the juniors. Please, Professor Bhagawatula Murthy. The stage is yours, sir.
Can you see me or?
I don't see you. Yeah, now we can see you. Yeah.
You can see me. Okay, good. All right. Let me share my screen. Can you see my screen?
Not yet. We see you, but not your screen.
Okay. Now, can you see?
Not really.
I put it in the screen mode.
Sir,
You can
the
click
bottom
on the
you have
share.
share option, sir.
I did.
Yeah,
I did.
share option.
So you should highlight that and should share it.
One second. Let me one minute. Okay. Let's see. Okay, got it now. Now can you see the screen now?
Yes, sir. Yes.
Sorry. All right.
It's visible now. Yeah.
Now can you see the screen?
Yes, it's visible.
Okay. Now let's go from the beginning. All right. Can you all see? All right. Thank you. First of all, I would like to thank. Is everybody able to hear me?
Yes.
Can you hear me, sir?
Yes, please.
Thank you all. It's midnight for me now, but it's okay. I usually go to bed at 3 in the morning every day, so it's fine. Now, I would like to thank the organizers, especially Subhashini Garu, for inviting me to give this presentation at the Golden Jubilee celebration of Andhra University Biochemistry Department. Thank you. And first of all, I would like to congratulate the organizers, all the faculty actually, and of course led by Subhashini Devi Garu, but I'm sure everybody else, Raghav Rao, everyone, is contributing, conveners, co-conveners, and whatnot. This takes a lot of effort to organize something of this. I attended the inaugural event yesterday. Part of it. I couldn't attend the whole thing. It was very late for me, but I did my best. But I saw the VC speech and everything. I was very, very happy and proud to see the enormous development of the Department of Biochemistry since I was a student when I joined in 1979 to 81. Like Roman Reddy said, he was my senior actually. Roman Reddy gave a fantastic talk, and I know him very well, quite well. And I'm really proud of his accomplishments and achievement. Like I said, quite a few of us have done quite well in US or other countries, India, and whether it's industry or teaching or research, academic people have done, and really happy and proud of that. And let me first of all thank my teachers, Professor Radhakrishnan Garu. He recruited me as a student in 1979, I remember. Then of course Govardhan Reddy Garu, and then Ramana Garu, no more now, but he was a great teacher. Of course, then Rama Rao Garu, Sri Ramul Garu, Ravi Garu, ASPR. I think all the faculty I've covered, and they would not only just teach, they would be personal friends. That's what I think we appreciated most when we were students, especially. They were like not just teaching courses and leaving, and any time we would be hanging around in the department, they would come and give advice what to do, how we should establish in our careers. That's one thing. And everybody was friendly, the staff, the scientific staff, the attenders, including everyone, for that matter. I do thank all of them. like Roman Reddy mentioned, this biochemistry department created a foundation for us, created a place to train, to learn. And like he said, in many fields you can develop. You can be a toxicologist, you can be a molecular biologist or a virologist, bacteriologist, whatever. But the biochemistry master's degree gives a fundamental knowledge of the advanced courses, we used to have. And that was, and actually ours was the first external pattern. I don't think many of you know this. 1979 batch was the first external pattern of exams, semester system. So all our exams were set by others and evaluated by outside. So that was later again it changed to non-semester or year-end pattern. But anyway, without much ado, I would like to now start my talk. I don't want to waste a lot of time on, but I would like to thank, like I said, very encouraging. I would like to encourage each of you students to. There's a lot of research opportunities still, whether it's US, UK, or India right now, and so it's for the biochemistry today. The future is still very bright. A lot of things are unknown. All right, so let's start about my research on molecular mechanisms of prevention of polycyclic aromatic hydrocarbons by omega-3 fatty acids implicated for human lung cancer. All right, so potential PAH sources. What are PAHs? PAHs are polycyclic aromatic hydrocarbons. They are present in environment like California wildfires, Superfund sites, cigarette smoke, air pollution, PM2. You name it, PAHs are there. Diesel exhaust. So all of us humans, we also consume that in the diet, especially in the Western diet, like charcoal broiled steaks. They have benzoypyrin, PAHs. Even in India, when we have fried stuff like pakoras and bhajis, the oil that we fry becomes really very black after a lot. That's all benzoypyrin, PAHs. So they're not good for you. So it's not good to use charred material, especially the oil, because of the heat. These compounds are formed. Now, so these are Superfund sites. We actually, I'd like to thank Shivakumar Garu for his very generous and kind introduction. And so these are the cigarette smoke. I talked about all this. So lung cancer. So what is lung cancer? So in the United States, it's the biggest killer, or even in the world, in males and in females. So lung cancer was the second largest cancer after prostate cancer, and there are about 121,000 cases every year in men and another 112,000 in women. It's a bad disease. Like I tell you, somebody has metastatic lung cancer, they give you a year to survive. So it's terrible. And so our goal is to figure out the mechanisms of how cancer is formed, actually, or cancer develops, and especially in environmental exposure. I'm not getting into the genetics of lung cancer. Most of the lung cancers are caused by environmental exposures, like cigarette smoke. All right, so I'm going to be focusing mainly on cytochrome P450s, like Dr. Shivakumar mentioned. I've been working on this P450 field ever since I was actually a graduate student at IASC, 1983. When I joined and Professor Madhyastha, we started working on cytochrome P450 enzymes. So I had a pretty solid PhD in toxicology in those days. Even now I continue. So it's almost like, I would say, close to 40 years of research in drug metabolism and cytochrome P450. And I've been very fortunate in a way to continue my PhD work in US when I was first recruited as a postdoc to look at vitamin D metabolism, and then I came to pharmacology at Baylor, where I continued P450s, and then the list goes on. So, and it's really, I'm in a way really fortunate. So humans are exposed, like I already mentioned. So cytochrome P4. So what are P450s? Okay, they are the main enzymes in liver and many other tissues that metabolize drugs, xenobiotics. So you have probably heard about xenobiotics. Xenobiotics are compounds that the body has not seen. So it could be like cigarette smoke, or it could be drugs, but I'm not including endogenous. So P450 since then are also involved in endogenous metabolism, like bile acids, steroid metabolism, and these are different isoforms of P450. So now, so they're, like I said, they're highly inducible. So there are many isoforms. There are 57 in humans, and there are total P450. There are over 5,000. Lots in plants, in fungi, and in the mammalian system. So, but in humans there are 57, highly inducible. So when you give an animal a compound like phenobarbital or 3-methylcholanthrine, you can see an induction of cytochrome P450, and that multiple forms we already talked about. There's overlapping substrate specificities. They're also involved in carcinogen activation. So that is what I'm going to focus on. So although these enzymes are usually good for you, they metabolize, detoxify drugs. They also can actually activate carcinogens to uh, very reactive ultimate carcinogens by P450 dependent metabolism. I'll go to that in a minute. So I'm going to be focusing on three isoforms. I wouldn't worry too much about the different. We'll another time we can talk more. So the 1A1, CYP1A1, I'll be saying 1A1, 1A2, 1B1 all the time. There's no need of keep repeating. CYP is inducible in liver and lung, constitutive in lung and other organs. And 1A2 is liver specific, constitutive and inducible. And the 1B1 is present in extrahepatic tissues and is inducible. So 1A2 again very important. It's liver specific, constitutive, but still is an important enzyme in the metabolism of carcinogens. So the one family, like I already told, 1A1, 1A2, I've explained this. So this is the bidirectional promoter. If you look at the gene, this is the 1A1, 1A2 are like head-to-head arrangement in the gene. So the same promoter elements, they actually control so on the left side and the right side. You can look at the gene structure and the regulation of this bidirectional promoter. So how are pHs induce cytochrome P450? So this is the aryl hydrocarbon receptor, which is in the cytosolic. So what happens is the pH is bind to the receptor in the cytosol, and then it goes to the nucleus, and then what happens is it binds to xenobiotic response elements and then activates the transcription of these genes. So this is the way the AHRRNT work, and these are the AHRR. This is the repressor. I wouldn't worry about it. So basically, by binding to the promoter, the AH receptor complex, so the PAHs, they activate CYP1A1 gene expression, 1A2, and also 1B1. So these are the multi-step processes of carcinogenesis. A lot of people are probably familiar with this, but this is talking about this, taking into consideration that some of them may not be familiar, especially the students. So I would like to go a little bit towards the very basic steps in carcinogenesis. So the first step is tumor initiation. So this is DNA damage by carcinogens or carcinogenic metabolites. It's irreversible. And when a compound binds to DNA, it can form a DNA adduct. That's the covalent binding of the benzoypyrin or methylcholanthrine metabolite, and it will bind to the many times to the G2 of guanosine or adenosine, N2 of guanosine. So what happens is when these adducts are formed, that they are not repaired, it can lead to mutations, and that mutation can lead to tumor promotion. Depending on where the mutation is, suppose the mutation is in a tumor suppressor gene, that inactivates this tumor suppressor gene, leading to carcinogenesis, right? And same thing if there is activation of the oncogene by this mutation, that can lead to oncogenesis, right? So we also have tumor promotion that leads to alteration of gene expression. That's the second phase by tumor promoters, and there's an increase in cell proliferation. And once that happens, no stopping to that. Then there is a continued cell division, and then they proliferate, and they start attacking the host, and they grow over the host, and then they're taking all the nutrients, and then leads to further tumor progression, further DNA damage, chromosomal abnormalities, clonal expansion of aberrant cells, leading to apoptosis sometimes, but sometimes the normal cells start dying. And so this is the bad stage. So our goal actually is to figure out what the mechanisms are and then see if we can prevent this. So this is the basic metabolic activation of benzoypyrin. Like I told you, this is a benzoypyrin molecule by P450 interaction. It can form a 7,8 epoxide and then a diol, and then a further metabolism leading to this 9,10 epoxide 7,8 diol, leading to the ultimate uh, uh, Bay region hypothesis, looking at DNA adducts formation. So our hypothesis, we have four hypotheses to be tested. One is the P450 would play an important role, like I said, in the sustained induction of CYP1A1 by 3-methylcholanthrin. That the 1A2 null mice would be more susceptible to MC-mediated adduct formation and tumorigenesis, and 1A1 and 1B1 would be less susceptible. The opposite. So they're very there's a lot of sequence homology between 1A1 and 1A2, but they are completely opposite in terms of carcinogen. That is the most interesting. We published this paper recently in Toxi. We also wrote a very very coveted review in Seminars in Cancer Biology, just published. It has a very high impact level, so review article. And because of my experience, they invited me, and I was proud to publish that paper. And the last hypothesis is that, so how do you prevent omega-3 fatty acid like fish oil? This fatty acid that represent fish oil that would prevent lung carcinogenesis by PAHs. So the design of the experiment is, so what we did is we took 8-week-old female mice or 1A2 null mice, treated them with a single dose of 3-methylcholanthrin, that's the PAH, like benzoypyrin, and then we looked at P450 gene expression, measuring the activities, Western blots, messenger RNA, very, very simple experiments. And then for lung tumorigenesis studies, we took their AJ mice or the 1A2, and then we treated with a single dose of either methylcholanthrin, and we evaluated 20 weeks. So the tumorigenesis studies are much different. Then we first do IP injections and look at a lower dose regimen to look at immediate induction of P450's DNA adducts. So the DNA adducts are formed very fast, within 1 or 2 days. But the tumors don't form until 20 weeks, 24 weeks, 30 weeks in mice. And in humans, it's 20 years. So if you could smoke for 20 years, usually nothing happens, but only after 20, 25 years is when cancer is formed. Now, so these are actually, I'm going to talk a little bit about our earlier work. So then the reason I'm talking is I'll impress upon you why I'm talking about this old work that we did initially when I came to pharmacology at Baylor. This is how we got into this field at all. So I'll explain to you. So we took adult rats, and then we treated them with either corn oil, which is a control, C, and then we also used methylcholanthrin, that is the dark triangles. Then we have light triangles, this is control. And if you can see, there is a sustained, we treated them for 4 days. Then we looked at 1 day, 8 day, 15 day, 20, there's a time course. As you can see, it plateaus beautifully. So there is a clear cut dose, with the time, there is sustained induction. Even at 45 days after drug withdrawal, you can see that there is quite a bit of P450 1A1 activity here. And whereas in the control, it's all same. But then the MROD activities is 1A2 is also sustained, but not as much as the 1A1. So this was an interesting finding. These are Western blots again showing the 1A1 and 1A2. These are two proteins we could separate very nicely by Western blot. You can see the molecular weight of 50.2. There's two nice bands, but even at 45 days, you can see sustained induction. And this is very important because why is it that this enzyme keeps hanging around even after withdrawal of the drug? That's another project that we are working, got funding for figuring out the mechanism. But the key is it has a physiological role. Because of the sustained induction is when the cancer actually ensures, So that's why it's an important experiment to be done. So these are basically, then what we did is we wanted to know if the 1A2 has a role in the sustained induction. So what we did is we took 4 different groups. We have corn oil wild type, this group, the first group. Then we have wild type methylcholanthrin, knockout 1A2, and then 1A2 with MC. You can see the wild type and then we can see there is a sustained induction, right? 1 day, 8 day, and 15 days in mice. The other experiment was in the rats. But in the 1A2 knockout, you can see very interestingly the sustained induction kind of goes away, suggesting in liver the 1A2 plays a role in persistent induction, right? Because in the wild type it's there, but in the knockout the induction falls, although the methylcholanthrin is around. This is lung. You can see lung is very different. We have sustained induction in the lung all through the 15 days. And the reason why this is important is this is organ specific or tissue specific, and the lung is the target organ for PAHs, not liver. So PAHs don't form liver cancer; they only form lung cancer. So the fact that in liver the P450 go down, the 1A2 knockouts actually show lower induction, lower sustained induction. It didn't really impact, but when it comes to lung, we have a sustained induction, suggesting that the sustained induction of P451A1 in the lung contributes to lung carcinogenesis. Very, very nice, interesting experiments. These were back in the day. These are old studies, but still I thought they're very impressive. Even today, they are all very much valid. So now you can see these are the 4 groups you can see, and this is you can see the Western blot in the lower panel. Wild type 1A2 null, you can see that in the you can see 8 and 15 days, you can see the 1A1 induction is there at 1 day, and you can the null mice we don't have the 1A2 anymore. So these are the Western blots in liver and lung, clearly showing. So we have wild type corn oil, wild type MC, corn oil knockout MC. You can see pretty good induction, but less than the wild type. But in 8 and 15 days, as you can see, there's sustained induction of 1A2, the 1A1, but the 1A1 disappears. There's no band at 1A1. And in the lung, you can see there is sustained induction even with 3-methylcholanthrin in the knockout 1A2 knockout mice, suggesting that in the liver and lung, the regulation of P450 is completely different. These are actually transgenic mice. Again, I thought I'll show you this nice picture to show. So what we did is we introduced the human lung 1A1 luciferase reporter gene into wild type mice, so that we can now look at lucifer is actually close, right? So when you have induction, even without killing the sacrificing the animals, euthanizing, we can see this. We do imaging of the luminescence. We have this, so we put the mice in this imager, and you can see in the wild type mice, you can see some induction here, 1 day, 8 day, 15 day, but in the 1A2 null. So what we did is we bred that against the 1A2 null background. It's beautiful. You can see 1 day in the 1A2 null mice, you can see it's very red. You can see the second panel here on the right is really red. That means very high induction. So clearly suggesting that in the absence of 1A2, the induction capability is very, 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 very high, suggesting that in the normal animal, the 1A2, what it does is it degrades the MC, metabolizes it, so you don't have much. But when you lose 1A2 or when you delete 1A2, you can see a lot of induction of 1A1. You can see the fluorescence here, the second panel here. The same thing, these are the image density, you can see is really nice. 4 groups. The first group, blue bar is 1A1 luciferase wild type corn oil. The red bar is 1A1 with methylcholanthrin. And then the 1A2, you can see the green bar, corn oil, we don't have much, but the blue bar is the 1A2 null mice. You can see very, very, very strong induction. And this correlates with what we see in the upper B panel. You can see the red very, very clear. I don't have to show you that. So this is the chip assay actually using the H receptor, suggesting again that what we do is for the chip assay, we immunoprecipitate and then we bind to the DNA. So what we do is we isolate nuclei actually, and we lyse them, and then we break the DNA into small fragments, and then we immunoprecipitate with the H receptor antibody. And then what we do is we do this PCR. So what happens is the HR complex that is not affected because it's bound to the promoter region. So you can see this band, the 1A2 is very strong, and in the 1A, this is the 1A2 null, as you can see. So all the studies clearly suggest with the Western blot, gene expression, RNA real time, and in the chip assay, these all beautifully correlate, suggesting that we have a nice story to tell, confirmed by different techniques, molecular techniques we use to confirm our findings, and uh, also delineate the molecular mechanisms by which these PAHs induce cytochrome P50, 1A1, 1A2 at different levels. So for the cancer initiation study, so what we did, we used, like I mentioned to you, so all the studies so far we did the small, lower dose and also shorter time points. For the cancer initiation, what we did, we took these three different groups of mice, wild type 1A1 and 1A2, and we gave corn oil, or we gave benzoypyrin 40 micromole per kilo, or methylcolanthrin one dose, and then we look at so this like, initial induction say 1 day, 8 day, 15 day, but then we kept these mice up to 20-25 weeks for our cancer studies. Sometimes longer, sometimes 36 weeks. So this is the mechanisms of how the sustained induction happens. This is our hypothesis. So what happens with methylcloranthrin, it binds to the H receptor, and then RN, and then it goes to the cytosol. So this is the binding of the MC with the H receptor. And then what happens, our hypothesis is that MC can form a metabolite called MCX, and that will in turn go, that will also bind to the H receptor. So this MC and the metabolite complex will bind to this H receptor and bind to this the promoter region, and then activate 1A1 gene. But in terms of this sustained induction, our hypothesis that the MCX binds and it competes here, and then it still continues, and it binds covalently because it has a hydroxyl group. We think it's 2-hydroxy MC. So once it's bound, it doesn't go away, so your induction continues. So that is our hypothesis, still to be proven, but I think it's pretty feasible or viable. So this is how the sustained induction can happen, and it can continue, and it will keep the gene on, which means more carcinogenic metabolites, more mutations. So I think this is what I think is the exciting part of our story. So I'll talk a little bit about switch gears now, talk about DNA adducts. So what are DNA adducts, right? So why are we looking at DNA adducts? I told you that PAHs can bind DNA and they can form adducts. So when people did correlation with, it's called a correlation coefficient, you can find a pretty strong correlation between DNA adducts and tumor formation. That's why looking at DNA adducts is a good biomarker, because that can predict what can be the stage of cancer. Because there's no need of looking at 24 weeks, 30 weeks, 40 weeks, when we can look at DNA adducts initially and then can at least predict it's a very strong carcinogen. They'll definitely form tumors later. So this is the benzoypyrin. So this is the P32 post-labeling assay. I learned this from the Andrea's lab in the early 90s when he developed my ex-mentor. So what we do, the advantage of this is, in the past, before this P32 post-labeling was discovered, of course it's not a new technique anymore, it's 40 years old, but before that they used to inject radioactive carcinogens into mice, and then like DMBA, dimethylbenzantacene, very strong radioactivity, and then keep washing the DNA many times and looking for binding, but they were all still not very foolproof because it could be tight binding. But with P32 post-labeling, it's certainly covalent. You take the DNA from the animal, you don't label it, you don't inject any mouse radioactive. Regular carcinogen is okay. Same thing with humans. Cigarettes most exposed, you can take WBC, extract DNA, and then digest the DNA, and then we label it with P32 in vitro. That's why it's called post-labeling. And then we found these adducts, the transpiring adducts, the thin-layer chromatography, and then you expose that to autoregressive. Now we have the phosphorimager. We don't do X-ray films anymore, but it's nice quantitative data. This is the DNA adduct I was talking about. So you see, this is the benzoypyrin molecule, clearly 2 hydroxyl groups. Here you can see, and then it can bind to N2 of G, guanosine. This is DNA. So you can see the benzoypyrene molecule down here and forming adduct. So these are the initial experiments we did with fish oil actually. So so what we did is we exposed mice to corn oil, which has no adducts. Then we have AJ mice, female, and then we have 182 mice, and we treated them with 3-methylcobalamin. As you can see, the 182 knockouts have much higher level of DNA adducts than the wild type mice. So now what we did is we looked at two different genotypes, the 1A1 knockout and the 1A2 knockout. I told you these are very, very close, lot of sequence homology. But why did we do this? Because we wanted to know if there's any difference between these two enzymes when you knock them out in terms of DNA adduct formation. As you can see, these are the three different genotypes. Wild type, we have 1A1 null, the green bars, and the red bars are 1A2 null. As you can see, the when you look at DNA knockouts, the DNA adducts are less in the 1A1 knockout, but higher in the 1A2. And same thing, male and female. So clearly suggesting that when you knock 1A1, you get less adducts. When you knock 1A2, you get more adducts, suggesting that in the normal animal, 1A1 plays a pro-carcinogenic role, whereas 1A2 plays an anti-carcinogenic role. So this is DNA adduct formation, again, same thing. In pH, I don't go into too much of details here. 1B1 again, very interesting. When you knock 1B1, which is again for a lung specific enzyme, which find less adducts, suggesting that 1A1 and 1B1 both are carcinogenic. So we did this experimental design. I think I showed this before. But we looked at 1A wild type, 1A1 and 1A2, and these are the tumor data. As you can see, very nice. The wild type, you can see a lot of tumors. Benzoypyrene and methylcobalamin, these are all tumors. And you can see the 1A2 knockout, you can see a lot more tumors here, and you can see they are really pretty big. So this clearly suggests that the 1A2 plays a protective role. So when you knock it, you get so many tumors. So you just compare these two. Now again, this is again 1A1, 1A2 wild type mice. Benzoypyrene, you can see tumors here. You can see tumors here. This is expected, and but as you can see here, the 1A1 knockout almost has no tumors, but the 1A2 has tons of tumors. You can see this one, a lot of tumors, and benzoypyrene we still higher than wild type mice. This is the histology of tumors. You can see corn oil, methylcobalamin, benzoypyrene. You can see clonal group clumping of these cells. These are all carcinogenic. With 1A2, there's a lot of tumors here. You can see where it's the naked eye, you can see all this. Of course, it's a microscope, but what I'm saying is you don't have to use a lot of technology. You can clearly see the. Then we quantitate, of course. So our overall conclusion is that the 1A2 show increased pulmonary carcinogenesis while 1A1 might display resistance. This is tumor multiplicity. You can see wild type mice, green bar, blue bar, sorry. The green bar is 1A1 knockout and 1A2 is the red bar. You can see really high tumor multiplicity. And this is male and female. And we have pulmonary tumorigenesis. This is again the same thing I showed again, there's no need to repeat. Again, these are additional experiments we are just showing. So our overall theory is that when methylcobalamin or PAHs are given to 1A2 null mice, there's an increased pulmonary induction, P450, more DNA adducts, more lung tumorigenesis, and then lung tumor. So our working model shows that the 1A1 show resistance to carcinogenesis, whereas this is basically the model I already showed you, and the 1A2 bile increased susceptibility because there is higher adduct formation and higher induction of P450, higher tumor formation. All right, so this is now I'm going to switch gears a little bit. So we wanted to look at tumor prevention. This is the last phase of my talk. So basically, what we try to do is we exposed mice to fish oil. We gave a fish oil diet actually, and then what we did is we maintained this for 1 month. When I say fish oil contains omega-3 fatty acid like eicosapentaenoic acid and di docosahexaenoic acid (DHA). These two are omega-3, very important polyunsaturated fatty acids. And we gave fish oil, and as you can see very clearly, benzoypyrin DNA DG adduct, and we also have total DNA adducts. The first group is corn oil. Corn oil means just benzoypyrin without the fish oil diet. And as you can see, in each group, uh, the next is fish oil. Second group, you can see less adducts, the red bar, and even in the black bar shows less BPD adducts. And but we same thing with when we give higher dose of EPA or lower dose, higher dose of docosahexaenoic acid was the lower dose you can see, and this is the last one is a mixture of both EPA and DHA. You can see clearly that the DNA adducts are much less in the DHA or fish oil treated mice compared to corn oil treated mice diet. So these are very, very interesting. So what happened is when we exposed these mice, we took these livers and lungs, extracted the microsomes, measured activities of P450 or gene expression actually. As you can see, the first group is corn oil mice, but in the all these this group second group onward, everything is much less P450, 1A1, 1A2, and 1B1, and the H receptor. Same thing in liver and lung. So what's very striking was these omega-3 fatty acids. Not only they're reducing the RX, they're also decreasing P450s. So does that make sense, right? Because you have less P450, you have less DNA RX. So this is an interesting observation. And these are DNA repair enzymes. They were all modulated because this could also happen at the DNA repair level, not just P450s. So this is our experiment showing very interesting. The black bar is the 182 null, corn oil mice, and then you have the blue bar, greenish, showing the lung tumor formation. After EPA DHA, you can see much less tumors here. And in the right side, same thing, EPA DHA, you can see benzpyrin and methylcholantrin, you can see much less tumors. And you can even see in the panel here very clearly that when you give methylcholantrin, you get quite a bit of tumors, but when you give the right bar, you can look at EPA DHA, you can see much less tumors. Very nice. So what we're trying to say is whether it's DNA adduct formation as well as lung tumorigenesis, both are very, very much reduced by the omega-3 fatty acids. So this is the histological section. The left bars are clonal selection of these tumor cells, corn oil, MC, benzpyrin, and the right panel is EPA, DHA. You can see very, very nice. So almost no tumors here. So this is our overall hypothesis. So we feel that the pH is the omega-3 fatty acids can act at either the 1A1, 1A2 level, or they could also act at initiation or the DNA repair level. We don't know really exactly where they are acting, but each of these mechanisms are very important for us, whether it's DNA adduct level or at the tumor progression level or at the metastasis level. So we are still doing experiments as we speak on the different aspects of omega-3 fatty acids that they can at different stages they're able to control. So actually right now what we're doing is we are giving the inhibitor for soluble epoxy hydrolase. So what soluble epoxy hydrolase inhibitor does is it takes these omega-3 fatty acids and then because actually the epoxides that are the active ones, it will convert them into diols and then the activity is gone. So we want to inhibit that step. So you have increased accumulation of these omega-3 fatty acids and then can lead to more protection. That is our hope. So the conclusions are, our results support the hypothesis that hepatic 1A2 enzyme plays an important role in the persistent induction of 1,1-by-methylcholine. And that the 1A2 plays an anti-carcinogenic role against methylcholine-mediated carcinogens, while 1A1 and 1B1 both play pro-carcinogenic roles. And the third conclusion is the 1A2 mice could be developed as novel animal models for new future chemoprevention studies in humans, natural products like omega-3 fatty acids. And the last conclusion is omega-3 fatty acids can prevent lung tumorigenesis very clearly. We are doing a lot more studies now. We have this 1A1, 1B1, and 1A2 different knockouts we are having. We are also having the combined knockout like 1A1, 1B1, and triple knockouts and double knockouts. So all of them we are doing. They are all very, very expensive experiments, doing 9-month, 8-month tumor studies, and we are also doing a lot of reverse phase protein array experiments, RNA-seq, global RNA-seq, single cell RNA-seq, for getting multi bioinformatic data on this mega complex pathways. I'm not showing you all that because we don't have the data yet, but all these experiments are absolutely ongoing and leading to some very, very nice publications. I'm pretty sure in 2-3 years down the road, we will see a lot of papers coming from our lab because this is a very, very unique model. Nobody is doing this kind of work anywhere in the country. I know that because they're not cheap, very expensive, and B is they're moved on to different aspects, but the basic foundation is not being pursued by a lot of investigators. So that's why we are trying to do very simple experiments, but again highly mechanistic and very impactful. Because we want to now go into clinical trials in humans in MD Anderson. I'm collaborating with folks in MD Anderson to look at see if we can give lung cancer patients who would like to give omega-3 fatty acid and see if this can reverse the tumors to some extent. So that's something we are trying to talk to our oncologists at the VA. And so our goal right now is not just do animal experiments, but actually take it to the next step. That's translational lung cancer. And so we can also get tissue banks from tumor banks which are available. We want to look at this fascinating study. The weakness of our studies are the limitation actually is that sometimes these omega-3 fatty acids also can be more toxic. So we're trying to also make analogs through our chemistry core to see if some of the side effects can be, because as you know, the biggest challenge in lung cancer, even today, there are so many drugs are coming, chemotherapeutic drugs. Every day we can see new drug coming into the market, but many times they fail because they cause side effects. But omega-3 fatty acids is good because it's fish oil, it's present in fish, and so you can take it, extract it, make supplements, and sell them so there are less side effects. And also it helps against many things, against inflammation, against arthritis, against many diseases. It acts in a beneficial way. But so we would like to now move on to the next phase. So that's going to be my focus on the next 2-3 years, and we try to renew this R01 grant. So these are my collaborators, Dr. John, like Dr. Reddy, Ganta mentioned. Without a big group, we don't have a very large group. We have 8 to 10 people right now, but Dr. John, I thank these investigators who are like in the lab a lot, doing the experiments, and graduate students, postdocs. But we also have other projects, so I don't put too much on one group. And these are all the funding from the NIH. And we have quite a few grants. The Super Fund is the latest one, P42. It's a $10 million grant that is actually talks about PAH exposure, Superfund sites, and looking at hyperoxic lung injury. And so that is actually, I'm proud to say, that is the very first Superfund awarded to Baylor College of Medicine, to the entire Houston. No, they never had one before. And I was proud to direct that and apply for it. And because I also collaborated with Rice University and UTMB in Galveston, and we have about 80 people in that Superfund project, and key personnel included. They all basically report to me, and I'm taking this project forward. The goal is to renew that in a couple of years, and this is a huge deal for Texas Children's, Baylor College of Medicine, and for the whole Houston, because they didn't have one before. There are 28 centers, Superfund centers in the US, and Superfund sites are actually toxic sites where a lot of waste, hazardous materials are dumped, and that causes a lot of problems, cancer, diabetes, and many diseases in humans. And so our Superfund project actually looking at preterm birth and lung injury. And next time I'll talk about that aspect. This time I wanted to focus on lung cancer, but again, we still always all these actually crosstalk with each other. These projects, whether it's lung injury or lung cancer or PH is super fun. And so this is really exciting times for us, and we are able to generate a lot of enthusiasm among trainees, a lot of graduate students, postdocs. I'm going to, in fact, one of our postdoc got a Casey Donnelly Grant Award recently, and we are getting diversity supplements. So my like to just give you encouragement. Now my phase is, apart from doing my own research, I'm mentoring a lot of junior faculty, a lot of fellows, and a lot of physicians getting into research. I'm seeing their careers through by helping them get their own grants. And because I always believe that the student success is the professor's success, our success. So I want to see many of my students, fellows, they are already in very high positions. Many of them are, some of them are division chairs elsewhere, and they're always grateful to my mentorship, because my one of the goal, and actually Shivakumar probably didn't mention that. Uh, of course, I probably forgot also to tell that I got two mentoring awards recently for mentoring. These are the department level, which means, and our department of pediatrics has 1,500 faculty, and among them I was the one given the award for teaching and research and mentoring. So I'm very proud of that. I also got awards for teaching and overall Baylor. But anyway, without much ado, I would like to thank all of you for your attention. And I would also now ready to take any questions. Thank you all. And this is Baylor College of Medicine, Texas Medical Center. This is the Texas Children. This is my office, the one you can see in the building here.
Sir,
Sir, I have a
outstanding. Hello.
Sir, I have a primitive question. I mean,
Okay.
for your fatty omega acids, and then as a food supplement after 50s, many doctors are prescribing omega-3 fatty acid as a supplement daily. And of course, you suggested it's a preventive for this carcinogenic effects. But if it is accumulated high concentration of omega-3 fatty acids, and it's forever, it's in the body, and so that it won't induce any carcinogenic cell in the liver as well as the lungs, or if high concentrations of this fatty acid accumulations, what will be the repercussions for on the body?
That's an excellent question, Dr. Nathaniel. Awesome question. I don't know the answer to that. But I don't think it's bad, because I know a lot of people are taking high. Actually, the doses that we give in animals are exactly what humans take. We are not seeing any bad effects so far with omega-3 fatty acids in humans, taking for other reasons, not cancer, for arthritis. So there's also the omega XL, which gives you very, very nice, pure compounds, supplements, nice supplements from New Zealand. They get from the mussels, they isolate, and they are not seeing any bad effects of omega-3 fatty acids. It also helps against cholesterol, blood pressure reduction, and so many great benefits it has. So I have not seen. Of course, anything too much is bad for you, of course, but one has to control that, and then then we'll see how. I still think it's quite positive.
Yeah, I'm aware of that. The doctors continuously prescribing fatty omega-3 fatty acids or omega-6 fatty acids.
Correct.
And then how long they can take, throughout the year or 1 or 2
Yeah,
months?
you can take it. Actually, you can take it. Absolutely, you can take it every day for years. Nothing will happen.
Okay. As a normal metabolite, it can be digested, but it is, synthetic. If it is from fish oil and all, it's a natural introduction. If synthetic also, no harm to take the continuously.
No
and
harm,
as a
no.
preventive measure, we can take it consideration. Whoever is
Correct.
taking omega-3 fatty acid cannot be induced by this carcinogenic.
Correct.
It's a,
So
and once it is
we go
effective,
for Yeah, go ahead, please. Okay.
Yeah, currently the COVID-19, it is inducing some sort of, black smuts in the lung. And for that, those people, if they take intermittently, it can be avoided. Or once it is affected and it is already on the disease site, also it acts upon.
Anti-inflammatory. Yes, you're right.
At intermittently they take, and it will be the effect will be lessened while
Yes.
the COVID enters into the lung.
Yes.
And if it is already entered and started the black smut, and that time also it acts upon and can
Yes.
increase the dose and
We have
can
not
give
done.
the benefit.
We really don't know about COVID yet because we have not studied that, but I believe it will be pretty good.
Right, sir. Right, sir. Thank you, sir.
Thank you.
Okay, before we go for further questions, let us first formally thank Professor Murthy for an outstanding talk. No wonder why you are getting so much of funding. It's really a fabulous piece of work, and I can see that the junior faculty working under your mentorship are so fortunate to have you as their mentor. Excellent work, and I hope the juniors at the Andhra University who are listening to this talk will get motivated to do science, to take up science as their career. And further questions, please, anybody, if you have any questions.
Thank you, Dr. Shivkumar.
Any further questions, please?
Sir, I have a question. So, will it N3 works on other type of cancers?
That's an excellent question. I don't know the answer. That's great. I don't know.
Oh, yes, that's only the question from my side. Thank you, sir.
So I have a couple of naive questions, Professor Murthy. This 1A2, in the absence of 1A2, if I understood correctly, the recruitment of AHR onto 1A1 promoter enhances.
Yeah.
How does that how does 1A2 prevent otherwise the
right? So our hypothesis is that the parent compound is metabolized by 1A2. So when you delete the 1A2, a lot of parent compound is around. So it has more of that. It can occupy the receptor and activate the gene. That is the simplest explanation. There may be other mechanisms, I don't know.
And the enhanced tumorigenesis in 1A2 null mice,
yeah,
is it 1A1 dependent?
Yes.
And you did the double.
So when we delete, that's a great question. So when we deleted both 1A1 and 1A2, we got a lot of tumors. And in the triple knockout also 1A1, 1B1 and 1A2, we got a lot of tumors, suggesting that 1A2 absence can by itself cause a lot of tumors.
So it is 1A1 independent.
Well, yeah. If you remove 1A1, but 1B1, one of them have to be there. 1A1 or 1B1, one of them have to be there.
Okay, right.
Otherwise, it cannot form adducts.
Right. And one last question is actually the NF-kappa family members such as RELB was shown to be a part of the AHR receptor, and it binds to the promoters of where AHR binds and contributes to the transcriptional regulation down the line. So in your tumor model, do you see any role for the NF-kB, particularly RELB?
That's a good question. I have not looked into that at all.
Right.
We have to look at it. Thank you.
Okay. If any other further questions are there, please. Otherwise, let us formally thank Professor Murthy for a fantastic talk. Yeah, so Vashni, you have a question, please. Yeah.
Please. Thank you, Murthy garu, for your excellent presentation.
Thank you.
Our students are benefited richly by your presentation. Thank you so much.
Thank you. Thank you for the opportunity and.
Sir, please stop the presentation. Murthy garu.
Yeah. Okay. Sorry.
Thank you. Bravo, please continue.
Okay. Gone, right?
Thank you, Murthy sir.
Okay.
Raghuram garu, please continue.
Raghavara sir, are you there? Sir, welcome Murali Krishna sir.
నమస్కారం అండి.
నమస్తే మాస్టర్. బాగున్నారా సార్?
బాగున్నా అండి.
బాగున్నారా?
బాగున్నా. మూర్తి గారు బాగున్నారా? చాలా రోజులైంది. ఎప్పుడు వస్తున్నారు మైసూరు?
వస్తాను నెక్స్ట్ టైం.
రండి. మీ బెంగళూరులో మీ బ్రదర్ ని సిస్టర్ అని చూడడానికి వస్తారు మీరు. ఐఎమ్ వెరీ హ్యాపీ. You have carried out an outstanding piece of work, and you are, I think, you are the true brother of your Dr. Ravindranath. He is such an eminent chemist, and your sister-in-law is also an outstanding scientist. I am very happy about the progress which you have made. Yeah, really happy to see you.
Thank you, Andy. Thank you. And for you, congratulations. You have done extremely well as well. Very nice.
Yeah, now I think we have to retire at 60 years in CSR. I have retired already years back. Unfortunately, fortunately or unfortunately, we can't continue up to 68 or 70.
So the retirement is 60 or 65?
No, for us 60 only.
Oh no, that's bad.
Yeah,
you're too young to retire.
DRDO, ISRO, Atomic Energy are all 60 only.
Okay.
HRD institutes are 65.
Yeah. But anyhow, we have to
actually, it was funny because we have a new division chief, and she just came from Cincinnati, and she started because I'm the main research person in the section, and she's my boss now. And she said, "Oh, I hope you're not going to retire." I said, "No." I said, "At least 20 years, I'm going to continue." I told that afterwards, let's see. Then she's happy. But I don't know. I'm just joking. But we don't know. It depends on our health and everything.
No, here in India, situation is different, Murthy.
Yes.
Because a lot of unemployment problem. So invariably they keep 60. Unlike in academic institutes where you can go up to 65 and plus another 3 years, 68.
Right.
Yeah.
All right. Okay. Thank you then.
Yeah, Murthy garu meeku chaala late ayinattundi kada. Already must be 2 o'clock.
So I will look at the recording. I hope Subhashini garu can send me some recordings.
Yeah.
I will listen to some part of your talk.
Sure, master. We'll send.
Okay.
We'll send once this events are finished. We'll send.
Okay.
We'll send soft copy of souvenir also to all.
Okay. Thank you so much. Thank you, Raghav.
Thank you, Murthy garu, for sharing your wonderful research on this platform, and congratulate you once again for receiving award recently. Say my good wishes to your kids.
Yeah.
Thank you very much.
Yeah, they're 21 years now.
Oh, very good. Thank you, Sivakumar, for chairing the session. Sivakumar, you're there?
Yeah, I'm here.
Yeah. Thank you for sharing the session and spending your valuable time with us. Thank you for that.
Thank you, Raghav.
Let us move on to the next talk. I am glad to present the biodata of our most distinguished alumni of the department, Dr. Murali Krishna garu. I'm very happy to present his biodata on this platform. Dr. Murali Krishna garu hails from Machilipatnam, Krishna district, Andhra Pradesh. He did his masters in our department in 1979 and then moved on to Mysore, CFTRI, for his PhD program, and got PhD award from the University of Mysore. Then he moved on to postdoctoral fellowship to Germany on DAD fellowship in the year 1988 and 90. Later, he was selected for the prestigious DBT overseas associateship and carried out work at University of Leipzig, Germany in the year 2008. and probiotics. He led CFTRI, DAE, DBT sponsored fellowships at CFTRI Institute, and he published more than 75 journal papers in peer-reviewed journals. In fact, for his credit, he guided about 14 PhD students and supervised 25 MSc or MTech dissertations in biochemistry, biotechnology, food science faculties of University of Mysore. I'm very happy to mention he received several awards. He received Glyco Science Award in the year 2002, Lalji G. Smarak Nidhi Award, and CFTRI Best Publication in Basic Science for the year 2005 and 6. Apart from that, he also received Best Scientist Award in 2010. And he is a recipient of Carbohydrate Excellence Award by the Association of Carbohydrate Chemists and Technologists in India in the year 2013. During the year 2018, he was awarded with most prestigious Lifetime Achievement Award of ACCT during 2019. He was bestowed with ACCT Fellowship India at the International Carbohydrate Symposium held at Lucknow, University, Lucknow. Presently, he is serving as a task force member of the national funding agencies and advisory board member for and several other international journals. It numbers more than 25. And for his wonderful contributions, his PhD students were placed in India, USA, and Europe and occupied many good positions. And he was a president of SBC Mysore local chapter, that is Society of Biological Chemists, and he was a convener for 85th annual conference of Society of Biological Chemists held at CFTRI Mysore during the year 2016. And I'm very happy to mention that he is an active person in our ABA group, Andhra University Biochemistry Alumni Association group, and he posts very interesting and informative postings. I sincerely thank him for that. We are very happy for those postings, sir. Especially I wanted to mention. And with these few introductory words, I invite Dr. G. Murali Garu to deliver talk on structure functionality of non-digestible carbohydrates.
Thank you, Dr. Rajesh. And there are so many familiar faces, and I'd like to thank, first of all, the Vice Chancellor, who was the Chief Patron, Dr. Prasad Reddy Garu, and the entire biochemistry staff, the conveners, Dr. Subhashini Devi, Dr. Raghav Rao, and Dr. Hemlata, who is retiring this month, and Dr. Professor Radhika, Dr. Aparanji, and distinguished audience, students and participants. I'm very happy that my classmate Dr. Elizabeth is there, and our 3-4 year junior Dr. Sharda Garu is also there, and so many people names are familiar, and I would like to welcome all these people for this presentation, and Dr. Ramachandra Rao, who is a very good student of plant cell biotechnology, and he has been doing extraordinarily well. And welcome Ramachandra Rao. And
good morning, sir.
Yeah. And without, I think already all of you are 40 minutes behind the schedule. I don't want to take more time, and I would like to go ahead with the presentation. And can I have the slides, madam? Or actually,
Yes, sir, we are presenting.
yeah, madam. Okay, madam. Yeah, all of you are aware that Biochemistry Department of Andhra University is celebrating its 50 years of establishing the department in 1971. Incidentally, I was there at the Silver Jubilee, and I am happy that I am there at the Golden Jubilee also. I attended offline in 1996. Now 2021, I am on online because of the various factors that are, I think, prevailing. The situations, everybody can't go and present the things personally. But anyhow, nevertheless, I am very happy for the invitation. And my talk is the structure functionality of non-digestible carbohydrates. And all of you are aware that CFTRI, Central Food Technological Research Institute, is a post-harvest technology institute. It is one of the leading laboratories of Council of Scientific and Industrial Research. 38 laboratories are there. We concentrate on food research and technological aspects of food research. Can I have the next slide, please? Hello? Next. This talk I am dedicated to the memory of my late parent G.S. Ramchandra Rao, Srimati Krishnakumari, my late brother Dr. Mangaraj Gudipadi, and my nephew Arun Kirti. Both of them unfortunately died in the accident in USA in 2005. My brother was so supportive to me with respect to this biochemistry program, and my uncle Dr. N.V. Nagabhushnam Garu, and Professor Subbarao Garu was the principal of AU Science and Technology in 1974 to 77, and Professor T.M. Radhakrishnan Garu, and Professor T.G. Reddy Garu, and Professor T. Ramana Garu, who are excellent teachers who are not there with us now. I dedicate the lectures to my parent, my brother, and Professor Subbarao Garu, and TMR, TGR, and T. Ramana Garu. And to briefly, as already informed, I have 77-79 batch. I would like to remember my teachers with gratitude: Dr. S. Sivaprasad Garu, Dr. Kottapalli Ravi Garu, Dr. A. Ramarao Garu, Dr. Sriramulu Garu. Our teachers, when we joined, Professor D. Sivaprasad Garu went to Australia on deputation to do PhD, but I could meet him 3 or 4 times in CFTRI, and my best regards to all of them for their excellent teaching and guidance. And I want to remember my MSc classmates, best wishes to them: Dr. Varaprasad, Bhaskar Reddy, M.V. Subramaniam, V.V. Subramaniam, M.D.M. Prasad, Venkata Krishna, Dr. Varalakshmi, Dr. Prabhakar Lakshmi, Dr. Elizabeth, Dr. M. Vijay Lakshmi, Srimati Lakshmi Kumari, Srimati Lakshmi Prasanna, and Srimati Baby. So we are 14 students. At the end, we are 13 students because one of them, Srimati Baby, left in between. Okay, next slide, please. Yeah, there is a saying from Hippocrates that let food be thy medicine and medicine be the food. So with this one, so much of research that has gone in for centuries together all over the globe with respect to food and their constituents and their chemistry, their biological effects on various disease conditions. And in our CFTRI, we concentrate on research on food ingredients, cereals and pulses, and other bioactive compounds. Next slide, please. Next slide. Yeah, this is, all of you are aware of these things. Since some students are there, I would like to mention monosaccharide, disaccharide. Everybody knows these oligosaccharides. Some of them students may not be knowing. This raffinose, stachyose, and verbascose. They are all sucrose, galactose linked sucrose. This is a trisaccharide, stachyose, tetrasaccharide, 2 galactose residues present in alpha-1,6 linkage, and verbascose, around 3 galactose residues present linked to sucrose. So the starch, everybody knows amylose and amylopectin, and non-starchy polysaccharide, you have pectins, polygalactonic acid esters, and hemicelluloses, you have mixed linked beta glucans, and arabinoxylans and arabinogalactans. These arabinoxylans and arabinogalactans are very, very important polymers, and their importance in several of the diseases is getting well documented. With respect to nutritional classification of carbohydrates, you have monosaccharides, glucose, fructose, and galactose, disaccharides, lactose, sucrose, and maltose, oligosaccharides, maltose, polysaccharides, starch. All these are available. So we have the enzyme machinery to digest all these things. So we get the energy. The unavailable non-digestible oligosaccharides is raffinose, stachyose, verbascose, azugose, because we don't have alpha galactosidase in the small intestine, so they don't get digested. Invariably, they will go into the large intestine and gets utilized by the microbes. And resistant starch is digestible, but resistant starch is the one which you get it after several heat processing treatments. You have a retrograded amylase which is called resistant starch. And fructo-oligosaccharides and xylo-oligosaccharides. Xylo-oligosaccharides are the degradation products of arabinoxylans. By the action of the endoxylenase, you get xylo-oligosaccharides. And dietary fiber is a common name which is used for plant non-starchy polysaccharides and lignocellulose complex. These are all, you don't get energy out of this one, so they are called as unavailable and non-digestible carbohydrates. Next slide, please. Next slide. Most of the non-starchy polysaccharides which are not digestible have the beta linked polymers, and we don't have the enzymes to digest, as I told. Next please. See, there are several health benefits which have been ascribed to this dietary fibers, be it with respect to diabetes, atherosclerosis, colon cancer, and mineral absorption. And here one more. Especially, it has a lot of effect in the immunomodulation with respect to the gut-associated lymph tissue. It is the largest immune organ situated in the gut, where continuous exposure to diverse antigens takes place, and it contains about 60% of the lymphocytes in the body. Especially the carbohydrates, undigestible carbohydrates, which are now called as prebiotics, has a role in the immunomodulation. Next slide, please. This is the schematic representation of serial arabinoxylans. You have beta 1,4 linked xylan backbone with arabinose substitution at 1,3, 1,5, and 1,2 linkages. And you have D-glucuronic acid present as a side chain substituents, and you have cinnamic acid derivatives, which is either ferulic acid or coumaric acid or caffeic acid, are linked to 5th hydroxyl group of arabinose. And it is ester linked. It has a lot of important functional attributes. Next slide, please. So, elevation of disease symptoms. It is with respect to diabetes, these polysaccharides are highly viscous. They slowly release the postprandial glucose. They will impair the glucose release, and reduction in insulin concentration, and high viscosity and soluble nature of arabinoxylans are responsible for slow gastric emptying and reduction in the small intestinal motility. Atherosclerosis is with respect to the prevention of cholesterol absorption by bile acids and lower the plasma lipid concentration. And colon cancer, most of it binds the mutagens by uronic acid and ferulic acid containing arabinoxylans. So they have a positive effect with respect to all the diseases such as diabetes, atherosclerosis, and colon cancer. And most of the people know about this constipation. From time immemorial, people used to consume the dietary fiber with respect to eliminating the constipation. It facilitates easy passage of undigestible material and their excretion. And binding of metal ions, it's a dual nature. Phytic acid, which binds the metal ions, will prevent the metal ion absorption, which is sometimes detrimental. That is the only negative fact about it. Serial non-cellulosic polysaccharides structure function relationship and overview was published in critical reviews in food science and nutrition, and it was cited 103 times. So Dr. Subbarao, who was my second PhD student, who was an alumni of our biochemistry, has been doing extraordinarily well. He is a professor in JSS Medical College, and he has already guided 8 PhD students, and he has published more than 100 papers. I think now time is ripe for us to say goodbye, and we have to call these youngsters for the presentations. And the impact factor of that journal is 9.98, and we could publish in the dietary fibers an invited article in dietary fiber, fruit and vegetable consumption and health. Editors Klein and Muller, Nava Publications. Dr. R. Shyam Prasad Rao is my third PhD student. Both papers both publications have received quite good citations. Next slide, please. Yeah, essentially, as I told you, we have isolated the cereal arabinoxylans. We have isolated from rice, ragi, wheat, and in this chromatogram, see, there's a lot of research that has taken place all over the world with respect to dietary fiber effects, but there are hardly any research with respect to the individual components of the polysaccharides of the dietary fiber. So what we did is, you see, the arabinoxylans can be isolated with water-soluble arabinoxylans are there, water-insoluble arabinoxylans are there, because water-insoluble arabinoxylans you can't extract with water because they are highly ferulated. Ferulated is a cinnamic acid derivative, 3-methoxy-4-hydroxy cinnamic acid derivative, or comaric acid or caffeic acid. They will not be soluble in water. So most of them, when they have diferulates, these polysaccharides cannot be extracted with water. But you have water-soluble polysaccharides, arabinoxylans, which with low amount of ferulic acid will get extracted. We have fractionated both rice and ragi arabinoxylans on the DAE cellulose column. The major fraction is 0.1 molar ammonium carbonate in both the cases, and you have water-related fraction. Other fractions are somewhat limited, and 0.2 molar alkali fraction is the other third major fraction. So next slide, please. So what we did is Dr. Shyam Prasad Rao has carried out the structural analysis of purified arabinoxylans. Actually, purification of a polysaccharide is a challenging thing. One has to employ 3-4 methods: ion exchange chromatography, gel filtration, and you have to reverse phase HPLC, and you have to get a pure polysaccharide before you contemplate of the structural analysis. Methylation, all of you know what is methylation. It gives you the structural analysis, but all the hydroxyl groups that are linked in other than linkage, they will get methylated, and you have to hydrolyze them, and you get the methylated monosaccharides, and you have to see by gas liquid chromatography and mass spectrometry. And pyridate oxidation speed degradation are supportive methods for methylation. Pyridate degrades vicinal hydroxyl groups and generate aldehydes, and these aldehydes can be further converted into alcohol by Smith degradation, and hexoses will give you erythritol, 3-tol, whereas pentoses give you glycerol. So with this one, it is a supporting evidence for the methylation thing. IR spectroscopy gives you functional groups thing, and optical rotation, alpha linked arabinoxylans will have high amount of positive value, whereas beta linked will give you negative value, and proton NMR, 13C NMR, proton NMR will give you unequivocal assignments with respect to the structure of the polysaccharide. We could arrive at beta 1,4 linked arabinose and xylose at the third position and second position. This publication has been published in Food Chemistry. What we did is that we have seen the structural changes during malting. Malting is a controlled germination. What are the structural changes that have taken place during malting? We could see, and this has been published in Food Chemistry. Food Chemistry is in a very important journal. Its impact factor now is around 6.3. Next slide, please. It was cited for 41 times. And I would like to say that nobody has attempted the antioxidant activity of the individual fractions of these arabinoxylans. What we did is we have taken all the fractions that have been obtained by DA cellulose and determined its antioxidant activity, and we could document that 0.1 molar ammonium carbonate with respect to rice is the most potent antioxidant polysaccharide as compared with other fractions. And even ragi also has a high amount of 0.1 molar carbonate, has high amount of antioxidant activity. These polymers can be taken as functional foods. And this publication was published in Phytochemistry, and it was cited 151 times, and it was rated as best publication, annual award from CHRA in 2006. Next slide, please. And we have seen the effect of these arabinoxylanes on the phagocytosis of yeast cell, and in the presence of this barium hydroxide also, you can, ragi polysaccharides not only isolated with sodium hydroxide, even with barium hydroxide and potassium hydroxide, we could isolate this polysaccharide. Barium hydroxide gives you very rich arabinoxylanes, and potassium hydroxide also gives arabinoxylanes with some contamination. We have seen the effect of these arabinoxylanes on the phagocytosis. Then we could see that with 25 micrograms, 50 micrograms concentration, it has a positive effect with respect to phagocytosis. This publication was published in Journal of Food Science and Technology. Next slide, please. This is comparable with inulin, which is known as a very effective polysaccharide with respect to phagocytosis. This is the just the figure with respect to the phagocytosis. Next slide, please. Yeah, XOS means xylo-oligosaccharides. When you take arabinoxylanes and treat with an endoenzyme, it randomly cleaves the polymer and liberates the xylo-oligosaccharide, which varies in the degree of polymerization between 2 to 8. So these oligosaccharides are not digestible in the small intestine. Invariably, they go into the large intestine and increase the beneficial bacterial population such as bifidobacteria, lactobacillus, and pediococcus. So we wanted to see the functional properties of xylo-oligosaccharides as well as the chemistry of it. Next slide, please. So what are the energy losses and energy gains from carbohydrates undergoing fermentation? When these oligosaccharides or carbohydrates goes into the large intestine, carbon dioxide, hydrogen, and methane will be liberated, and you have short chain fatty acids such as acetic acid, propionic acid, and butyric acid. Butyric acid, a lot of research has gone into it with respect to cancer prevention, and in turn, this butyric acid is converted into carbon dioxide H2O with concurrent liberation of ATP. Next slide, please. We have developed a procedure with respect to both water extracted polysaccharide, water extracted polysaccharide from cereal and millet brands. And in the acetate buffer, if you take around pH 4.8, xylanase is very effective at 50 degrees. And you treat it for 2 hours, and most of the oligosaccharides will be liberated at that time. And then you precipitate with ethanol to precipitate the undegraded polysaccharide, and you take the supernatant, concentrate, and you can load on Bioagel P2 and then separate the oligosaccharides. So we got the oligosaccharide, the yield is maximum in ragi, followed by wheat bran and maize, and green gram and Bengal gram. We have very less amount of soluble oligosaccharides from arabinoxylanes. Next slide, please. What we wanted to say this, I can tell you with confidence, this a comparative account of xylo-oligosaccharides was not attempted in the literature. What we did is that we have taken isolated these oligosaccharides and seen the in vitro antioxidant activity by beta-carotene emulsion method, and we could prove that xylo-oligosaccharides from ragi has the maximum antioxidant activity as compared with wheat, maize, and rice. So these oligosaccharides were isolated by insoluble arabinoxylanes, and this paper was published in Food Chemistry, and it is cited already 112 times. Dr. B.R. Venus Sri is working in Davanagere in college. Next slide, please. We have separated these oligosaccharide and Bioagel P2, as I told you. WOV5 means this is wheat oligosaccharide 5 and 4. Both are found to be major oligosaccharide, and these oligosaccharide structure has been deduced by doing ESIMS and NMR, and it was found to be a disaccharide and trisaccharide. Next slide, please. So for this you get 437 and arabinose xylose ratio is 1:2, and 2 xylose residues and arabinose is a trisaccharide, and fifth one is a disaccharide, that 305, and you get is 2 xylose residues. Essentially, what you have to do is that you have 150, 150, 2 pentose residues, then it will be 300 minus 18. You will get 282, and the sodium attack 23 add, and you get 305. Likewise, you should calculate for the trisaccharide also. You have to eliminate 2 water residues, 18 plus 18, 36, and you get what you get 150 into 3, 450, 450 minus 36. You get 414 and 23, 437. That is the way to calculate the mass, and you can once again do GC analysis and determine whether it is arabinose or xylose in the oligosaccharides. Next slide, please. So NMR unequivocally give you the structural analysis with respect to C1, C2, C3, C4, C5. The ppm values are already established. You can easily determine the structure by comparing with the standard things. And xylopyranose 1 means xylopyranose residue at reducing end position. Alpha arabinose Fa3H2 means arabinopyranose linked to O3 and xylopyranose 2. Major oligosaccharide is a linear disaccharide xylobiose and branched trisaccharide with a single substituent of arabinose in the side chain. Okay, next slide, please. What we did is we have taken the Bengal gram, BGO means Bengal gram oligosaccharide, WBO means wheat bran oligosaccharide, and we wanted to see how they promote the activity of the beneficial bacteria. We have taken Lactobacillus brevis and DRH and Lactobacillus plantarum and Pediococcus pentosis and Pediococcus pentosis, two strains we have taken, both Lactobacillus brevis 2 strains and Pediococcus pentosis 2 strains, and in this one we could document that wheat bran oligosaccharides promote the activity much better than the Bengal gram oligosaccharides. Next slide, please. Next slide, please. And we could document these oligosaccharides. We could document the antibacterial activity with respect to E. coli and Bacillus cereus and Staphylococcus aureus, and we could document that these oligosaccharides inhibit growth of E. coli and Bacillus cereus and Staphylococcus aureus. Next slide, please. We have seen the immunomodulated activity of purified xylo-oligosaccharides with respect to the Wistar rat splenocyte as well as thymocytes, but these are not that much immunogenic as compared to the standard carnia and arabinogalactone because of the not having the functional groups such as maybe ferulic acid and ester-linked acyl groups. This was published in Trends in Carbohydrate Research. Next slide, please. And effect of Bengal gram xylo-oligosaccharides in the production of nitric oxide by peritoneal macrophage in vitro. These xylo-oligosaccharides definitely promoted the nitric acid liberation as compared with arabinogalactone and lipopolysaccharide. The effect is less, but nevertheless it is definitely much more than the control, several fold much better than the control, both at 24 hours and at 48 hours. And 72 hours also you have the effectiveness of this Bengal gram xylo-oligosaccharides. Next slide, please. You see, I wanted to tell you a prebiotic definition wise is undigestible carbohydrate is called prebiotics. A probiotic is a beneficial bacteria which is maybe lactobacillus or bifidogenic bacteria. And symbiotic means the preparation of the combination of both symbiotic and both prebiotic and probiotics are called put together. It is symbiotic preparation. What essentially what we did is that we wanted to test the probiotic and symbiotic milk sample used for antioxidant activity experiments, we have taken different lactobacillus species, Acidophilus NCDC 011 and Lactobacillus fermentum NCDC 156 and Lactobacillus brevis NCDC 01 and Lactobacillus plantarum NCDC 020 and Lactobacillus NCD 017 and milk plus xyloalgosaccharides are controlled, and these SLA, SLF, SLB, SLP and SLC are this is symbiotic preparation. So that's why we added a symbiotic. These are all probiotic things. So essentially here you see that probiotic after 24 hours you can see the values as compared with the symbiotic sample. Symbiotic sample, the pH has gone quite low. So that means it has a lot of beneficial effects as compared with the probiotic cultures. Next slide, please. Among this one, NCDC 01 was the most effective compared to all these microorganisms. We could prove NCDC 01 is the most effective. Next slide, please. And we have determined the total phenolic acid content of probiotic and symbiotic samples at 0 hours and 24 hours. And we could determine that caffeic acid, syringic acid, comaric acid, and ferulic acid. Among all these things, ferulic acid is the most preponderant. And we could correlate with the total phenolic acid content with FRAP assay as well as DPPH assay. And it correlates very well with respect to the antioxidant activity, the total phenolic acid content. Next slide, please. Next slide. So we have determined just now it will antioxidant activity of probiotic and symbiotic samples. Definitely symbiotic samples are much more effective as compared with the probiotic samples. You can always see after 24 hours this one is quite effective. The particular microorganism which I have told, NCD01 is much more effective than the other symbiotic preparations. Next slide, please. Yeah, another property which we have seen is that evaluation of symbiotic preparation for cholesterol lowering property and growth versus cholesterol assimilation. Here also NCD01 is the winner among compared to other lactic acid bacteria, and with respect to glucose was taken as the control, and NCD01 is the most prominent candidate with respect to the cholesterol lowering property. Even the percentage was found to be around 5 to 6%, nevertheless, it is much better than the other lactobacillus microorganisms. Next slide, please. Wild tolerance and acid tolerance of lactobacillus strains are also seen. NCDC01 is found to be the most effective, 2.82 compared to other things like 156 and C20 and 17. So effect of pH2 and viability of lactobacillus species. This is, here also NCDC01 is found to be much better than the other microorganisms. Next slide, please. Previous slide, please. Previous. Yeah, cholesterol incorporation of the cellular membrane, lactobacillus exposure and glucose control. Here also you can see NCDC01 is much more effective than NCDC 156 and NCDC 11 and NCDC 20 and NCDC 17. So we have, this one was published in Journal of Food Science and Technology. Leonard Lacerado he is now working as a head of the department and in the biotechnology department in Mangalore. She is a gold medalist. She has done extraordinarily good piece of work, and now she is carrying out the prebiotic research. Next slide, please. So, understanding the utilization of xyloalgosaccharide by lactobacillus transcription analysis. Actually, there are two things. One is whether the oligosaccharides will be degraded by the enzymes first, and then it will be transported, or there are specific transporters like ABC transporters, transferase system and secondary carriers. This is the carbohydrate transporters in the gram-positive bacteria. Next slide, please. This is the expression analysis of equus-induced genes in Lactobacillus brevis, and we could document that ABC type maltose transport system and ABC type sugar transport system and ABC type sugar transport system, perisplasmic component, and D-xylose proton supporter. So you can say that ABC transporters are involved with respect to the oligosaccharide transport. Next slide, please. This is real-time polymerase chain reaction quantitative PCR expression analysis. ABC1, ABC2, and ABC3 are upregulated, and xylose this is downregulated. So relative expression levels of XOS-induced genes during mid-logarithmic growth of Lactobacillus brevis by real-time PCR data was normalized by geometric mean by of two reference genes, rare B and rec K, and the control condition was growth on xylose. Essentially, what it indicates that xylobios or xylotriose are not degraded outside, and xylose is not taken up. That means the oligosaccharides are taken up by these transporters, and they are taken into the cell, and then it gets degraded by xylosides. Next slide, please. Yeah, with respect to the conclusions, XO mixture obtained from ragi showed good antioxidant activity, which is higher than wheat, maize, and rice xylo-oligosaccharides. The prebiotic activity has increased with decrease in chain length. That means xylobiose is the most potent with respect to prebiotic activity as compared with xylotriose or xylotetrose or xylopentose. Ferric reducing power as well as DPPH radical scavenging activity was significantly higher in all the symbiotic preparation as compared to that of probiotic samples. Lack of mitogenic and co-mitogenic activities of these, sorry, previous, please, kindly, previous slide, please, previous slide. Hello? Yeah, lack of mitogenic and co-mitogenic activities of these xylo-oligosaccharides can be ascribed to perhaps the absence of ester-linked acetyl groups and low degree of polymerization. Lactobacillus brevis showed significantly higher cholesterol lowering and bile salt deconjugation ability compared to other strains, which I have mentioned in the previous slides. Analysis of gene expression by real-time qualitative PCR showed a significant upregulation of genes putatively coding for the components of ATP binding cassette, ABC transport system. Actually, I have prepared for one more thing, arabinogalactan. Dr. Kiranmai, who was my last PhD student, she was from the Andhra University only. Now she is working in Anakapalli as a research associate in the Acharya Ranga University campus, and she has done work on arabinogalactan from green gram. She could prove the importance of the arabinogalactan with respect to modulation, with respect to antioxidant and prebiotic and the oligosaccharides, and with respect to even with respect to effect on the cancer cells also. Next slide, please. Now I would like to thank Chief Patron and Vice Chancellor Professor PVGD Prasad Reddy Garu, Organizing Committee of the Symposium, especially Professor Suhasini Garu, Subhashini Devi Garu, Professor Ragarao Garu, Professor Hemalata Garu, Professor Radhika Garu, and Dr. Aparanji Garu, and all the advisory committee members for their invitation. Next slide, please. Yeah, in your life, in anybody's life for that matter, nobody should think that they came up on their own without the encouragement of people, the higher up bosses, and the students who are working with you are all equally responsible for the growth of any individual. In that case, in that way, I have to, because they told me, they mentioned that I have joined as a research fellow in CFTRI. No, it is not correct, because I joined as a scientific assistant. An opportunity has been given by Dr. M.R. Raghavendra Rao Garu for registration of PhD. Actually, I got ICMR fellowship to work for PhD in CDRI, but I did not proceed, and I worked in CFTRI itself as a junior scientific assistant, and I got 8 promotions and became chief scientist, that is cadre 2, next to the director cadre. I retired as number 4 in the institute. Dr. Tarnathan Garu is a very eminent organic chemist. I could do my PhD in 82 to 85. And my thesis was evaluated by Jirla's Harvard Medical School person, and Professor C.B.N. Rao, not C.N.R. Rao, C.B.N. Rao, carbohydrate chemist. And then I went to Germany. Dr. Rajgopal Rao garu is a former director, great scientist, who has encouraged me to apply for German fellowship, and he has recommended for that fellowship. I am truly grateful to him for my career development. And the former directors of CFTRA, they never came in my way with respect to my research. Late Dr. Bun Hl garu, Dr. S.R. Bhaumik garu, he is in Calcutta now. Padma Shri Dr. Prakash garu, who worked as for 16 years as the director, who was always encouraging me for doing PhD. And Professor Ramrashegan garu, during his term, I was the head of the department. He gave me a lot of administrative responsibilities as Research Council Secretary, as well as I was the SBC President. He has cooperated with me with respect to conducting of a national symposium, which was a great success, and the headquarters people are very happy about the organization of the symposium. Next slide, please. Yeah, essentially I wanted to tell you that without student, no researcher can say about their research. You cannot go on doing bench level work till you are 60 without good students, and they are very dedicated students. Dr. M. Nirmala worked in MLSS, and she is as assistant professor in Cedar Serial Medical Center, Los Angeles. Dr. Subbarao, who was alumni of CFTRA, is a very good biochemist, and he is presently working as a professor in Department of Biochemistry, JSS Medical College. Dr. R. Shyamprasad Rao is working in the Bioinformatics Division, Enopaya University, Mangalore. Dr. Chitra, she is working as assistant professor in Central University, Kasaragod, Kerala. Dr. Madhavi Lata, who has worked on esterases. My lab is not only in chemistry thing, we worked quite a bit on enzymes also. She is working as a senior research engineer in Umeå University in Sweden. Next slide, please. Next slide. Anita Gopal, she is application scientist in Thermo Fisher, Bangalore. She worked on the serial starch acid of porcine pancreatic alpha amylase isoforms. Dr. Shamla Hegde, she worked on purification and biochemical correction of ferulic acid esterase from Aspergillus niger. She is working in Supreme Pharmaceuticals, Mysore Private Limited, and then good Mysore. And Dr. Madhukumar, he is bioactive. He has worked quite extensively on bioactive oligosaccharides from Bengalgram. He is the head R&D department, NESO, opposite to CSRT and Mysore. Dr. Leonard is working as assistant professor and HOD at Department of Biotechnology, St. Aloysius College, Bangalore. And Dr. Vinasri is faculty visiting scientist in the University of Davangere. Next slide, please. Savita Prashant, she is working as assistant professor in PG Department of Biochemistry, JSS College, Mysore. Shruti Raju, recently she has been given scientist C post in CDRI. She worked on characterization of protein polysaccharide complexes from Azovan with reference to their biological activities. Maushami Shankar, she is from Visakhapatnam, and she is from Geetam University, and she is assistant professor, Department of Biochemistry, St. Joseph College for Women in Visakhapatnam. And Kiranmai Keza also is from West Godavari district, and she is working as research associate, regional agriculture research station, Anakapalli, Acharya NG Ranga Agriculture University. What I would like to say that I would like to thank all these bright research scholars who have worked under my guidance. Because of them, my sustained hard work, we could progress quite a bit with respect to my carbohydrate research. And I would like to say that CFTRA carbohydrate research has been internationally recognized. My guide, Dr. Taranathan, and my students and Dr. Salima's student, we have total together around 40 PhDs, more than 40 PhDs came, and we have could publish more than 300 papers, and the center is well recognized. Of course, we are all retired now, but there are youngsters who are there to take up forward the research. Next slide. Thank you very much. Because it is due to time constraint, I have taken only 45 minutes, and I have not presented part of the work of Dr. Kiran Mai, because it is quite a bit delay is there, so I don't want to rather step into your lunch time. Okay, thank you very much, Subhashini Devi Garu, and all the organizing committee members, Raghav Garu, and then Hemalat Garu, who is retiring this month, and I wish her a happy retired life, and Professor Radhika Garu, and Professor Dr. Aparanji Garu. And it's a real nice thing that I could be presenting my data both at Silver Jubilee and Golden Jubilee things. I don't know about in future what will happen, because I'm retired. I will not have any candidates to present any more work.
Please be ready for Platinum Jubilee also, sir.
I don't. But I cannot present the same thing, sir. Maybe you have to call my student Subbarao and others because I'm not working anymore now. So anyhow,
You have left out so much thing. Those things you can introduce that time.
Yeah, it's great. Andhra University is the time which we spent is really memorable. And what Professor Radhakrishnan Garu used to tell me, Murali Krishna, concentrate hard work because I'm not the topper anything. I'm not the most brilliant student, but I used to be first class student, but I used to go for movies. He told I went for 150 movies in the first year and 150 movies in the second year. So he was very serious about thing, and he used to tell, unless you work hard, you cannot come up. That same hard work I brought to CFTRI, and it was evident. Okay, sir. Thank you.
Thank you, Murali Krishna Garu, for sharing your research on this platform. Really, it is very interesting and informative. And really, we thank you for your valuable time sharing with us. We are happy to have you on this occasion.
Yes.
Yeah. Quick few questions, if anybody interested to ask. One or two questions.
Hi.
Yeah, please.
Am I audible?
Yeah, you're audible. Please ask.
Elizabeth, very nice to see you. You are always there for every talk. Tell me, Dr. Elizabeth.
Yeah, some internet connection problem here existed. But anyway, my compliments to GMK. He exactly remembered his colleagues, his fellow students, and his classmates. And I should compliment further because exactly he remembered the classmate with their initials and all he posted here. So it's a very and
Sorry, Madam, here, and Murali Krishna sir is known for that, and very minute things he remembers like encyclopedia. We used to say while during our PhD time in CFTRI.
But Ramchandra, I forget. Yeah, thank you.
I have one basic fundamental. So intermittently, I missed some presentation slides because of the internet. But otherwise, in the earlier talk, we have the polycyclic aromatic hydrocarbons repercussions in the lung cancer. Here, non-digestible carbohydrates again can be converted as polycyclic aromatic hydrocarbons. So, at the large intestine, one slide was showing it can be converted into alcohols or water and carbon dioxide. So, if it is as sustained as polycyclic aromatic hydrocarbons throughout the digestive system up to large intestine, what are the repercussions in terms of initiating the cancer cells, cancer disease?
Actually, these oligosaccharides invariably gets degraded into the large intestine as acetic acid, propionic acid, and butyric acid. Butyric acid is known to prevent the colon cancer. That is documented in the literature. So, if a high amount of butyric acid is there, definitely there will be reduction in cancer. I cannot comment on polycyclic compounds because I have not covered the literature with respect to that one. But as far as fermentation is concerned, the effectiveness of this oligosaccharide fermentation is with respect to bifidogenic bacteria as well as lactobacillus bacteria that are present in the large intestine. But essentially what we did is we have chosen different bacterial lactobacillus things, and we have proved that one of the particular lactobacillus NCDC01 is the most effective compared to other organisms. But essentially, this is you are dissecting the things. But at the end of the day, if you want to see the entire effect, one has to go for animal experiments and take the feces and then see what are the microorganisms that are present still there and how they have utilized these things. That type of research was done in Europe quite a bit with respect to the animal experiments. Even for that matter, they have done human experiments also. In our country, it is very difficult to do human experiments.
So you mean to say when we took that non-digestible hydrocarbons, if any person is taken the probiotic, it is no harm. Otherwise,
Yeah.
it will be accumulated as alcohols, as you showed one side methyl and ethyl and propyl alcohols.
Yeah. No, that is acetic acid, propionic acid, butyric acid as the short chain fatty acids which do get liberated once these carbohydrate, undigestible carbohydrates gets fermented. If you have a high molecular weight polymer, it will not, you take cellulose as such, which is beta-1,4 linked polymer, which is completely insoluble and crystalline, it will not get degraded in the large intestine. Whereas arabinoxylans, it has solubility and there is degradation quite a bit with respect to the polymer per se. But when you take the oligosaccharides, they are completely fermented in the large intestine because we have beta xylosidase is there, which cleaves the oligosaccharide, and we have arabinoperoxidase is also there. It will cleave the arabinose, and we have now sugar transporters are there, which will take up this oligosaccharide as such, and they will be degraded by these exoenzymes in the by bacteria. So you get the short chain fatty acids, and those short chain fatty acids are very important with respect to cancer, colon cancer.
Yes. Yeah, I'm GMK talking about. Shall I continue? Yeah, I'm talking about non-digestible hydrocarbons. Of course, all starchy materials are polycyclic aromatic hydrocarbons. Am I right?
No.
So,
talking about
non-digestible.
You are talking about starch derivatives or starch?
For starch, all carbohydrates are starch, you can say, can be converted into their metabolic pathway as polycyclic aromatic hydrocarbons. Okay, and these polycyclic aromatic hydrocarbons from the non-digestible starches accumulates at large intestine before going into convert into acetyl-CoA connections. Am I right?
No. Actually,
so at this juncture, tell me. Okay, at this juncture, if it is a large intestine, it induces the cancer. So, what are the preventive measures you suggest for that sort of stagnations at large intestine?
You are talking about heterocyclic compounds is a different proposition altogether. What I am talking is that as far as these polysaccharide insoluble fiber or soluble fibers are concerned, the degradation takes place in the large intestine. It is selective and it will not be degraded 100%. There will be selective degradation. It depends upon the microbial species, and all these microbial species are genetically predisposed also. Every human being will have a unique probiotic bacteria in their large intestine. Okay, and we cannot really universal think that these are the microorganisms present in the there are 400 species of microbial species that are present in the large intestine.
And viral admissions are strong antibiotics.
Entirely different, Dr.
Yeah, please.
They're different. What we are addressing is that as far as undigestible carbohydrates, way back in 1950s, they used to think that cellulose is a roughage material, and we don't have any other carbohydrate material other than starch and monosaccharides or disaccharides. They are the only important. Dietary fiber is useless. That is the thing which was the thing in 1953 and 54. So, but a lot of research that has taken place for the past 50 years, they documented beyond ambiguity that they have a lot of beneficial effects. These prebiotics and probiotics are of interest, which is coming for the past 15 years. So, a lot of bacterial species, probiotic bacteria has a lot of benefits. If you scan the literature, you name anything, they say that probiotic bacteria has positive effect. See, you take probiotic curds are coming, they're very popular in all the European countries and as well as American countries. So they take probiotic curds, and they will definitely increase your beneficial bacterial population in the large intestine, so that you will be much more resistant compared to the people who are not taking this probiotic bacteria. And with respect to the oligosaccharides, and you take probiotic bacteria together, it will be symbiotic, will be much more effective than the probiotic bacteria itself or the undigestible oligosaccharide itself. So now if you take undigestible xylo-oligosaccharide or fructo-oligosaccharide, which are documented very well, fructo-oligosaccharides are very good for health. Even you can, that's why people suggest honey also. Honey people take with lemon juice in the morning. That will be very good for the intestine and large intestine health also.
Yeah, thank you. Thank you, JMK.
Thank you, Dr. Elizabeth. Convey my regards to your husband, Nathaniel, and blessings to your daughters and your grandchildren.
I can see.
Thank you so much. Yeah.
I see. ASPR Garu on the line. Prasad Rao Asifu from Leeds.
ASPR, oh, he is such a wonderful teacher. Actually, we'll never forget him, and he taught me carbohydrates.
Please, sir, you're on the line. Sir, hello.
Hello, sir.
Prasad Rao Garu, ASPR Garu. Hello, ASPR Garu. Hello.
Hello, sir.
Yeah.
Can you hear me now? Hello?
Yes,
Yes,
sir.
sir. Yes, sir. Yes, sir.
Okay, it's good afternoon for you, but it's quite early in the morning for us.
Yes.
I missed your talk
like
because
to
I was
see
trying
you.
to listen to it, but the
Sir, we
audio
would
connection
like
was
to
so poor, I couldn't listen to you. Now it is back
Yeah.
again. I switched to a different device. I could listen to it. So I only could follow the last few questions. Anyway, it's very nice to see you. And obviously, I couldn't attend yesterday. It was quite early hours for me. I think it works for both US as well as India. For UK people who are in the middle, I think it is very odd hours.
Yes, sir.
So I couldn't make it yesterday because I also had to go to work as well. I'm sorry about it, but I'm hoping to catch up. I tried to go on to the recordings, but I couldn't access the past recordings. But obviously, I would like to see what people have said about the department. Well, I know the early history of the department, obviously. So I think Professor Siva Prasad and Rama Rao and others must have told you how the department's roots are begun. But there are plenty of stories that could be of use to the younger generation that people like me could share with you. Unfortunately, I couldn't do it. Anyway, it's very nice to see you all. I didn't want to delay, and with my usual long speeches or talks, lectures. Yeah, it's very nice to see you all. I can see a few faces here, about 4. Obviously, you can't have the videos, all the system might crash. Nice to see you, Murali Krishna.
నమస్కారం అండి. మీరు మన మైసూరు వచ్చారు, మళ్ళా ఈసారి కూడా రండి తప్పకుండా.
Yeah, sure. Mysore is one of the nicest places that I have visited in terms of the cities. One of the best cities I have been to, and I think I would love to go there again. Yeah. Sure. The cold has restricted me quite a lot.
You have to go via Hyderabad, sir.
నమస్తే సార్, ఏఎస్పీఆర్ గారు.
నమస్కారం అండి.
ఎలిజబెత్ గారు.
Yeah, my video is not working, sir, because of the weak
connection. Okay.
So I couldn't see you,
I
sir.
can put my video on if that works.
Yes, sir. Very happy to listen your voice, sir.
Yeah.
Your camera will not be available. Okay. I will probably speak to you later in the afternoon sessions when Sridhar joins.
We were never missing classes of Dr. SPR.
I'm sorry?
We were never missing your classes, sir.
I'm so pleased. I'm too old now. See, 68.
Carbohydrates.
Yeah. I'm still continuing teaching glucose homeostasis to our final year students. Give 5 lectures. I mean, the microbial data is very, very interesting, obviously. And I think that is one of the reasons why I always worry about India, where people take enormous amount of antibiotics due to infections. And are we killing a lot of our good bacteria? Is that also contributing to the rise in diabetes? And it is also connected to neurodegenerative diseases as well. So I think India has to be very cautious in terms of prescribing antibiotics. We can't get prescription of antibiotics in this country that easily. We can't buy off the shelf. My daughter who is a consultant, she can't prescribe antibiotics for herself. She has to go to her doctor and get antibiotics. I think the abuse of antibiotics is very, very high in India, which obviously affects the beneficial effect that we have. It doesn't matter if you take probiotics. Probiotics is whether it's a commercial angle to it, so whether they are selling it like statins and a lot of other drugs. I'm a little bit cynical about some of the things that people advertise. But obviously, the kind of food that we take influence the bacteria that we have in our gut, and they have very useful roles in our body. So, like we said, not all bacteria are bad. There are a lot of good bacteria that are helping us. Let us not kill them. Thanks, Murali Krishna. I'm sorry I missed the talk, but I think I followed the last bits of the
Sir, I will share the
conversation.
slides, sir. I will share the slides.
I'm sorry?
I will share the slides, otherwise madam also can,
Yeah.
can send all the presentation to you, sir.
Yeah, I think I would probably rather like to listen to you and see the people. So if there are any recordings are there, that would be great.
Sure,
if
sir.
I can access them.
Sure, sir. We'll pass it to you, sir. Definitely we'll pass it to you.
Yeah. I tried last night. I said, no, I can't. Yeah. Anyway, thanks very much for that offer.
Yeah. Thank you very much, sir. Namaskar.
Okay, you're most welcome. The next session is in what time?
Yeah. Sir, please. Next session.
2:30, na?
What time it starts?
Yeah, it starts at 2 o'clock, sir. 2 o'clock.
2 o'clock. Okay.
Sure, please.
Yeah.
Thank you very much,
okay.
sir. Thank you.
I'll join you later. Okay. Thank you all. Nice to see you all. Okay.
Thank you.
Bye for now.
Thanks, sir. Namaste, sir.
Okay. Bye-bye.
Yeah, and another senior student, I'm Sarada from CFTRI. I think she is also on the line. Madam, Sarada Garu, nice to see you on the line.
Yeah.
Even I am seeing her. I am seeing Dr. Sarada, but no response. Madam, if you are there, please say hello.
Okay. Yeah, okay. Ramchandra Rao?
Yeah.
You want to speak?
Yeah, no, actually, I don't know the Siva Prasad Rao, another one just now. I was confused with D. Siva Prasad because yesterday I was seeing the inaugural session, but this Siva Prasad Rao seems to be another professor, which actually when I joined, he was not there. And actually, I wanted to address some of the questions that he has raised regarding the antimicrobial resistance. Actually, I was one of the member of the Ministry of Health and Family Welfare. Recently, we conducted in 20 medical colleges in all over India on the AMR projects. I was a third party evaluator to see how actually antimicrobial resistance program is being taken place in India. So in that, actually we found that yes, it is a long way to go, but actually not only in India, but India is of course it is over the counter. The antibiotics, whatever the antibiotics that doctor do prescribes or doctor do not prescribe, we can able to get those medicines. And we had conducted that evaluation and we came to know good facts and also the limitations, and we proposed to the ministry and the National Center for Disease Control NCDC Delhi, and the recommendations will soon going to be out. And that AMR program, which is truly important to enact the AMR program in India, one has to be aware about the antibiotic stewardship program, which has to come from not only from the consumers, from our stakeholders, but also from the doctor's angle, so that the prescription should be avoided. The doctor's angle
an hour lunch break.
Fine, madam. Fine.
Yeah,
Fine.
2:15 we'll start.
Fine. And I just wanted to know before that, the sharing of the slides, just like a PowerPoint, just sharing only. I just wanted to ensure that whether it is just I will share my slides and so that it is visible. That I wanted to share, madam. Once I am going to share how we should be shared, because we are giving on the Google. Let me see the sharing and then the slide.
Yeah.
How it should be seen.
Madam, ask admin to speak on that.
Yeah, share option is there now, sir.
Yeah.
Yeah, you can just click on the share option and you can share your slides.
No. That is, sorry, this is something else. Okay. Am I presenting what I am presenting right now? Can you see my slides on the screen?
Yeah.
Okay. Achha, okay. So now if I open, it is visible there, right?
Sir, we could see your slides, sir.
Yeah,
Fine. Okay. Oh, I just want to ensure so that we don't waste the time that time. Thank you. I'm just trying to, yeah.
Dear participants, will join at 2:15 after the lunch break.
Fine, no problem, madam. I am here because today we are off. So today I came to my lab just for presenting this so that it will be quite calm and here. Thank you. I am waiting for that. Thank you.
Okay, sir. Anil, welcome Anil, will join. Hello, madam. We'll join at 2:15 after the lunch break. Okay.
Right. Thank you, GMK. I missed intermittently.
Very nice to see you. Please come to Mysore.
Thank you.
Yeah.
Right, sir. Thank you, sir.
Thank you.
Subhash Nagar.
Okay.
సార్ మురళీకృష్ణ గారు
చెప్పండి రామచంద్ర గారు, ఎలా ఉన్నారు మీ పిల్లలు మీరు?
మేబీ అందరు వెళ్తున్నారు. సో వీ విల్ టాక్ ఆన్ దిస్ ప్లాట్ఫామ్. హ్యాపీ టు సీ యూ మళ్ళీ అగైన్ 2 decades తర్వాత మిమ్మల్ని చూస్తున్నాను మళ్ళీ. ఎప్పుడు 2002లో వచ్చినట్టున్నాను. యంగ్ సైంటిస్ట్ అవార్డు రిసీవ్ CFTRI అప్పుడు ఎప్పుడు వచ్చినట్టు.
అప్పుడు వచ్చారు మీరు, జ్ఞాపకం ఉంది. తర్వాత మళ్ళీ మిమ్మల్ని కలిసినట్టు జ్ఞాపకం లేదు. ఆల్మోస్ట్ 19 years.
రాలేదు నేను రాలేదు.
మీరు ఇండియాకి వచ్చి ఎన్ని ఏళ్ళు అయింది?
నేను వచ్చి 4 years కంప్లీటెడ్ అండి. 2017 ఆగస్టులో వచ్చాను.
వెరీ గుడ్. మీరు ఆల్మోస్ట్ ఎన్నేళ్ళు ఫారిన్ లో ఉన్నారు? 14 ఏళ్ళ?
18 years I was there.
18 years. Oh, you are one of the few exceptions that coming after 18 years.
అది 18 years ఉన్నానండి అక్కడ. సో అక్కడ మా అమ్మాయికి ఇంటర్నేషనల్ స్కూల్, నేను సేమ్ అంతకని గవర్నమెంట్ ఆర్గనైజేషన్ లో ఉన్నాను. సో అక్కడ ఉన్నప్పుడు ఒసాకా యూనివర్సిటీ నుంచి AIST అని నేషనల్ ఇన్స్టిట్యూట్ ఆఫ్ అడ్వాన్స్ ఇండస్ట్రియల్ సైన్స్ అండ్ టెక్నాలజీ, దాంట్లోకి నేను చుకుబాక్ అయ్యాను. టోక్యో దగ్గర చుకుబాక్ ఉంటే అక్కడ మా అమ్మాయి ఇంటర్నేషనల్ స్కూల్ చదువుతూ ఉండేది. కానీ నేను ఇంకో క్యూషూకి వెళ్ళంగానే ఆ సిటీలో లేదండి, పక్క సిటీలో ఉంది. కానీ నేను కమ్యూట్ చేయాలంటే రోజుకు 3 hours పడుతుంది దానికి. మార్నింగ్ 3 hours, ఈవినింగ్ 3 hours అయ్యేటప్పటికి నేను ల్యాబ్స్ లో నైట్ 12 వరకు ఉండే టైపు. ఈ CFTRI ఎలా ఉంటానో జపాన్ లో కూడా అలానే ఉన్నాను.
మీరు అసలు హార్డ్ వర్క్
అట్లా ఉండేటప్పటికి కాబట్టి కష్టం అని చెప్పేసి మా ఫ్యామిలీ ఏం చేశారంటే ఇండియా వచ్చేసారు. వాళ్ళు ఇండియా వచ్చారు కదా అని చెప్పి నో పాయింటింగ్ బీయింగ్ దేర్ అని చెప్పి నేను వచ్చేసాను. సింపుల్.
అంటే మీ వైఫ్ కూడా వర్క్ చేస్తారా?
లేదు.
ఎంతమంది మీకు పాప?
ఒకే ఒక పాప అండి. ఇప్పుడు షీ స్టడీయింగ్ +1, ఇది ఇంటర్మీడియట్ అండి ఇక్కడ ఫస్ట్ ఇయర్. సో తర్వాత నేను ఇక్కడికి ఇండియాకి రావాలనుకున్నాను. I don't know whether I am in good place or not, but I am in the National Institute of Health and Family Welfare and NIHFW ఉన్నాను, మినిస్ట్రీ కింద. సో ఆల్ ఓవర్ ఇండియాకి ఐఎమ్ గోయింగ్ ఫర్ అసెస్మెంట్స్ కి. టర్షరీ కేర్ హాస్పిటల్స్ ఉన్నాయి కదా, ఫర్ ఎగ్జాంపుల్ ఎయిమ్స్ ఢిల్లీ, ఎయిమ్స్ భూపాల్, ఆల్ మహాత్మా గాంధీ ఇన్స్టిట్యూట్ ఆఫ్ మెడికల్ సైన్సెస్ కి, వాటన్నిటికీ ప్రతి సంవత్సరం వాళ్ళని అసెస్ చేయడానికి వెళ్తా ఉంటామండి. సో దాంట్లో ఒక టీమ్ లీడర్ని. సో మళ్ళీ within 2 weeks లో గోయింగ్ టు స్టార్ట్ అనమాట ఈ సంవత్సరంది. సో అంటర్ ది కాయకల్ప అవార్డ్ స్కీమ్ అని. సో దాంట్లో వీ విల్ బి గోయింగ్ ఫర్ ఈ సంవత్సరం 2020లో మేము ఎయిమ్స్ ఢిల్లీని, ఎయిమ్స్ భూపాల్, తర్వాత CNCI కల్కట్టా, నిమ్హాన్స్ బెంగళూరుని అసెస్ చేశాము. సో ఏంటంటే ఇవన్నీ అడిషనల్ టాస్క్స్ అండి, మినిస్ట్రీ ఇస్తుంది, వీ హ్యావ్ టు డూ దాట్.
వెరీ గుడ్, వెరీ గుడ్.
సో ఇలా ఇవాల్యుయేషన్ ప్రోగ్రామ్స్ యాక్చువల్ గా మేము FSSIకి ఫుడ్ సేఫ్టీ స్టాండర్డ్స్ అథారిటీ ఆఫ్ ఇండియా ఉంది కదా, సో దాంట్లో తర్వాత డ్రగ్ కంట్రోల్ అథారిటీ ఆఫ్ ఇండియాలో నేను దాంట్లో కోపిఐని. సో ఆ ప్రాజెక్ట్ ఇచ్చి నేను ఆంధ్రాని డ్రగ్ ప్రాజెక్ట్ ని మొత్తాన్ని, డ్రగ్ సిస్టమ్ ఎలా ఉంది ఆంధ్రాలో అని అసెస్ చేశాను లాస్ట్ ఇయర్ బిఫోర్ కోవిడ్ అటాక్స్ అనమాట. అప్పుడు 2020 ఫిబ్రవరి వరకు విజయవాడ, గుంటూరులో ఉండి దాన్ని తర్వాత హైదరాబాద్ వెళ్ళి ఫుడ్ సేఫ్టీ ల్యాబొరేటరీ ఉంది కదా, ఇప్పుడు మనకి ఆంధ్రాకి హైదరాబాద్కి ఫుడ్ సేఫ్టీ ల్యాబ్ ఇంకా సెపరేట్ కాలేదండి. అది ఇంకా మనకి ఆంధ్రాది కూడా హైదరాబాద్లోనే ఉంది.
వెరీ గుడ్.
సో దానికి ఒక మినిస్ట్రీ ఏమిచ్చిందంటే, ఫుడ్ సేఫ్టీ వాళ్ళు ఇచ్చామంటే ఒక ఈచ్ ల్యాబొరేటరీకి 10 crores అలాట్ చేసాం. ఏంటంటే హై అండ్ ఎక్విప్మెంట్స్, GC మాస్, LC మాస్, ICP మాస్ వీటన్నిటికి. సో దాంట్లో వెళ్ళాలంటే తెలిసిన వాళ్ళే PI కోపిఐ ఉంటారు. సో అవన్నీ నా ల్యాబ్లో జపాన్లో నాకు హాఫ్ డజన్ ఉన్నాయి అట్లా అవి. సో అవి ఇచ్చినప్పుడు దాంట్లో ప్రాజెక్ట్ రాసి వాటిని అసెస్ చేయడానికి హైదరాబాద్ అలా వెళ్ళడం జరిగింది.
సార్ ఎక్స్క్యూజ్ సార్, గుడ్ ఆఫ్టర్నూన్. కృష్ణ గారు, ఇట్ వాజ్ నైస్ టాక్. సార్ డాక్టర్ శ్రీనివాస్, నేను సైంటిస్ట్ ఫ్రమ్ ICMR NIN.
చెప్పండి.
సార్ రామచంద్రరావు గారు, మీరు చెప్తున్నారు కదా రిగార్డింగ్ ICPMS, LCMS, వీ హ్యావ్ బీన్ ఎక్సలెంట్ ఫెసిలిటీ ల్యాబొరేటరీ హియర్ ఫర్ ఫుడ్ అనాలిసిస్, ఎస్పెషల్లీ అనలిటికల్ వేలో.
ఎక్కడండి? ICMR అది NIN లోనా?
NIN హైదరాబాద్ సార్.
ఓకే.
బట్ సార్, టు గెట్ ద ఫండ్స్ ఫర్ ఇన్స్ట్రుమెంట్స్ ఈస్ వెరీ డిఫికల్ట్ నవేడేస్. ఎట్లా సార్ అది? హౌ వి విల్ షార్ట్ ఇట్ అవుట్ దిస్ ప్రాబ్లం? ఐ డోంట్ నో. అందరూ ఉన్నారా సార్, లేకపోతే మనమే మాట్లాడుతున్నామా? ఎవ్రీబడీ ఈస్ ఆన్.
I'm seeing how many members. చాలా మంది ఉన్నారండి.
Sir, if you don't mind, can I have your number? I wanted to talk to you later on.
Sure. ఇస్తానండి.
ఒక్కసారి లేకపోతే మీ మెయిల్ ఐడి నాకు పాస్ ఆన్ చేసినా పర్వాలేదు సార్, ఇఫ్ యూ డోంట్ మైండ్.
ఓకే, ఐ విల్ గివ్ అండి. సో యాక్చువల్ గా మీకే కదండి ఫండింగ్ ఇప్పుడు నా దగ్గర ఉన్నా కూడా నాకు చాలా టఫ్ గానే ఉంది ఫండింగ్ ఇక్కడ. నేను ఇప్పుడు డిపార్ట్మెంట్ ఆఫ్ రీప్రొడక్టివ్ బయోమెడిసిన్ లో ఉన్నాను, కానీ చాలా మాకు టఫ్ గానే ఉంది. కానీ కొన్నిటిని ఇవేంటంటే ఫుడ్ ల్యాబొరేటరీస్ కి, గవర్నమెంట్ ల్యాబొరేటరీస్ కి మినిస్ట్రీ హ్యాండ్ ఓవర్ చేసింది. మినిస్ట్రీ ఆఫ్ హెల్త్ ఇచ్చి ఒక 10 crores ఇచ్చింది అనమాట, హై అండ్ ఎక్విప్మెంట్స్ కోసం. వాటిల్లో మొత్తం ఎలా ఉంది ఫంక్షనరీ, ఆ మ్యాన్ పవర్, యూటిలిటీ ఎలా ఉందని అసెస్ చేయడానికి మమ్మల్ని పంపించారు. అట్లా సో అట్లా నేను హైదరాబాద్ ఆంధ్ర డ్రగ్ లో, అట్లానే ట్రాన్స్ప్లాంటేషన్ ఆఫ్ ఏదంటే నేషనల్ ట్రాన్స్ప్లాంటేషన్ ఆఫ్ ఆర్గాన్స్ ఆఫ్ ది హ్యూమన్ బీయింగ్స్ అన్నమాట. సో పీపుల్ హూ పాస్డ్ అవే అండ్ దాంట్లో చాలా డిమాండ్ ఉంది. ఇప్పుడు ఏదో లో బ్యాండ్ విత్ అని చూపిస్తుంది నెట్ ఏదో. సో వాటికి మొన్న 3 ప్రాజెక్ట్స్ కి జస్ట్ కోవిడ్ స్టార్ట్ కాకముందు ఎట్లానే AMR అయితేనేమన్నా లాస్ట్ 2020 డిసెంబర్ లో కంప్లీట్ చేసాము. అది యాక్చువల్ గా ఒక 6 నెలలు లేట్ అయింది. సో ఇవన్నీ అడిషనల్ గా మినిస్ట్రీ నుంచి వస్తూ ఉంటాయండి. సో మేము ల్యాబొరేటరీ సైంటిస్ట్ అని కాబట్టి, సో ఆబ్వియస్ గా వచ్చినప్పుడు వి గో అండ్ అసెస్ ది కండిషన్ ఎలా ఉంది ఇండియాలో డిఫరెంట్ ల్యాబ్స్ లో అని వెళ్తా ఉంటాం. తర్వాత ఇప్పుడు ఏమన్నా ప్రతి సంవత్సరము మోడీ గవర్నమెంట్ లో స్వచ్ఛత అని పెట్టాడు కదా, కాయకల్ప టెరిషరీ కేర్ హాస్పిటల్స్ అని. సో దే ఆర్ గివింగ్ హ్యూజ్ ఫండ్స్ అనమాట. అటువి వాటిని అసెస్ చేయడానికని వి కీప్ గోయింగ్ అనమాట. గ్రూప్ ఏ హాస్పిటల్స్, గ్రూప్ బి అని ఉంది, 1000 bed హాస్పిటల్స్, బిలో 1000 bed హాస్పిటల్స్ అని. సో వీటిని వీఆర్ గోయింగ్ టు సూన్ స్టార్ట్. డిసెంబర్ 2nd week, 3rd week లో మళ్ళీ వెళతాము. సో అట్లా ఇవన్నీ ఏంటంటే అడిషనల్ అండి. సో బికాస్ ఇక్కడ నేను 2-3 కోర్సులకి ఇన్స్టిట్యూట్ లో పోస్ట్ గ్రాడ్యుయేట్ డిప్లొమా ఇన్ పబ్లిక్ హెల్త్ మేనేజ్మెంట్ అని ఒక కోర్స్ ఉంది.
వండర్ఫుల్ సార్. ఓకే.
సో దానికి ఎక్కడి నుంచో ఎక్కడికి వచ్చామండి, హార్డ్ కోర్ సైంటిస్ట్. సో ఎక్కడి నుంచో ఎక్కడికి వచ్చాము
సార్. ఓకే, ఎనీహౌ సార్, నైస్ టు టాక్ టు యూ. నేను మీతో కొంచెం మాట్లాడతాను సార్ లేటర్ ఆన్. వెన్ ఐ విల్ కలెక్ట్ యువర్ కాంటాక్ట్ నెంబర్ అండ్ మెయిల్ ఐడి ఆల్సో.
షూర్.
రామచంద్రరావు గారు
చెప్పండి సార్.
నాకు మీ ఈమెయిల్, ఫోన్ నెంబర్ ఇవ్వండి, ఎప్పుడైనా తర్వాత మాట్లాడతాను. ఈ ఫోన్ నెంబర్ చెప్పండి.
అలాగే సార్, రాసుకోండి అయితే.
సార్
మీరు కూడా రాసుకోండి సార్, అడిగారు కదా శ్రీనివాస్ గారు.
yes sir
7780
16 15 19. ఓకే సార్, రైట్ సార్.
సో మీకు మిస్డ్ కాల్ ఇస్తానే,
ఇవ్వండి మీ ఫస్ట్
7780 16 15 19. ఓకే, ఆల్ ద బెస్ట్ రామచంద్ర. వస్తాను మీ టాక్ కి, మళ్ళా నేను ఫోన్ చేసి వస్తాను.
మోస్ట్ వెల్కమ్ అండి. చాలా ఉన్నాయి, ఏదో కొంచెం ఇస్తున్నాను. ఇవన్నీ జపాన్ రీసెర్చ్ అనమాట. చాలా ఉన్నాయి, ఇండియాలో కాదు జపాన్ లో చేసినవి. కొంచెం హైఫైగా ఉంటాయి, బట్ ఐ వాంటెడ్ టు లెట్ అదర్ పీపుల్ ఆల్సో నో వాట్ వీ కెన్ డూ. ఇండియాకి ఏదో చేద్దామని వచ్చాను, కానీ ఏమీ చేయలేకపోతా అనే కొంచెం ఫ్రస్ట్రేషన్ లో ఉన్నాను ఇప్పుడు. యాక్చువల్ గా జరిగింది అది. సో బట్ ఎనీవే, బట్ ఐ విల్ నాట్ లీవ్ ఇట్ అప్. సో చూస్తాను. యాక్చువల్ గా మా ప్రాజెక్ట్, మా ఈ రీసెర్చ్ నుంచి చాలా మల్టీనేషనల్ కంపెనీస్ వచ్చాయండి. దానికి ఏది పేషెంట్ స్పెసిఫిక్ మెడిసిన్స్ గా ఈ రోజు చెప్తాను. అవే కొన్ని అన్నిటిని ఇంట్రడ్యూస్ చేయలేను కానీ. సో దాంట్లో ఏంటంటే మోనోక్లోనల్ యాంటీబాడీ, పేషెంట్ స్పెసిఫిక్ మోనోక్లోనల్ యాంటీబాడీకి, సో దాన్ని పర్సనలైజ్డ్ మెడిసిన్ గా, సో దానికి రిలేటెడ్వి అది రీసెంట్ గా వివాలిస్క్ అనే దానికి ఇప్పుడే కాదులేండి, 7 years బ్యాక్ మా టెక్నాలజీని అమ్మడం జరిగింది ఒక 30 million euros కి. బట్ దట్ ఈస్ రియల్లీ సర్వింగ్ ద పీపుల్ ఫర్ ది పేషెంట్ స్పెసిఫిక్ మెడిసిన్ ఫర్ ది ఫర్ అబౌట్ 2 to 3 weeks లో వచ్చేస్తాను పేషెంట్ స్పెసిఫిక్ మెడిసిన్. అదే కొంచెం ఈ రోజు ఆ టెక్నాలజీని ఇంట్రడ్యూస్ చేస్తాను విత్ లిటిల్ రీసెర్చ్ బ్యాక్గ్రౌండ్ తో. సో లెట్ మీ మేక్ ఇట్. చాలా ఉన్నాయి కానీ చాలా చెప్పలేను, బట్ ఐ క్విక్లీ గో త్రూ అనమాట. ఓన్లీ పార్ట్ ఆఫ్ మై రీసెర్చ్.
వెరీ ఇంట్రెస్టింగ్ టాక్ సార్. వి ఆర్ ఈగర్లీ వెయిటింగ్. అనిల్, హాయ్ అనిల్. అనిల్, హాయ్ అనిల్.
హలో సార్, నమస్తే సార్.
యా అనిల్, హౌ ఆర్ యు?
నమస్తే సార్, బాగున్నాను సార్. రామచంద్ర సార్, నమస్తే సార్. నేను మిమ్మల్ని యాక్చువల్లీ ఇంట్రడ్యూస్ చేస్తాను సార్. ఆఫ్టర్నూన్ ఐ వాస్ గివెన్ టాస్క్ టు ఇంట్రడ్యూస్ యూ.
ఓకే, అనిల్ కుమార్, నైస్ మీటింగ్ యు అండి.
నైస్ మీటింగ్ సార్. నా పేరు అనిల్ కుమార్ అండి. నేను ఇక్కడ మన యూనివర్సిటీ ఆఫ్ హైదరాబాద్ లో ఫ్యాకల్టీ సార్. ఐ మీన్ ఐఎమ్ అబౌట్ టు.
సార్ నాదే సార్, మిస్డ్ కాల్ నెంబర్ సార్ ఫ్రమ్ శ్రీనివాస్.
ఒక్క నిమిషం అండి, ఒక్క నిమిషం. ఎవరిదండి మీది? ఎండింగ్ విత్ 4529?
ఎస్ సార్. యా,
మీరు శ్రీనివాస్ గారా?
అవును సార్, అవును సార్. యా,
ఓకే ఓకే, నేను జస్ట్ మెసేజ్ చేస్తాను. ఇది మీకైతే సూర్యనారాయణ గారు తీసుకుంటారు కదండి? సూర్యనారాయణ
సార్. నేను అనిల్ సూర్యనారాయణ ఇక్కడ కలుస్తుంటాం సార్.
యాక్చువల్ గా సూర్యనారాయణ నా క్లాస్మేట్ అండి, బయోకెమిస్ట్రీ.
వెరీ ఇంట్రెస్టింగ్ సార్.
సార్ నాకు సూర్యనారాయణ గారు కో గైడ్ సార్ పిహెచ్డి.
ఓకే ఓకే, నైస్.
పిహెచ్డి చేశాను. NIN లో హెడ్ హాక్ ఫ్యాకల్టీగా ఉండి ఇక్కడ మూవ్ అయ్యాను సార్ నేను.
ఓకే ఓకే.
మురళీకృష్ణ గారు చాలా చాలా వెరీ నైస్ టాక్ అండి. నమస్కారం.
అనిల్ కుమార్ గారా?
అవునండి,
నమస్కారం. మీరు ఇంట్రడ్యూస్ చేయాల్సింది.
నేను అవునండి, రామచంద్రరావు గారిని.
మీరు రామచంద్రరావు, మీ పేరు నాకు పెట్టారు, ఇందులో పెట్టారు ఇంట్రడ్యూస్ చేయాల్సింది.
మిస్టేక్.
అనిల్ ఏ టైం మళ్ళీ? వెన్ వి ఆర్
2:15 అండి, 2:15.
ఓకే ఓకే, రైట్ అయితే దెన్. ఓకే
రామచంద్రరావు, ఇప్పుడు మిస్ కాల్ ఇచ్చాను చూడండి.
ఒక్క నిమిషం సార్, మీది చేస్తాను. ఒక్క నిమిషం. మీకు ఇప్పుడు మీరు డిక్లైన్ చేశాను. ఒక్క నిమిషం, మీరు వచ్చింది. ఒక్క నిమిషం, లాస్ట్ 3 numbers చెప్తాను. ఒక్క నిమిషం. మీది లాస్ట్ 2199 మీదేనండి?
9480382199 కరెక్ట్.
అదే 2199 లాస్ట్. ఇప్పుడు నేను యాడ్ చేస్తున్నాను సార్.
ఓకే అండి, థ్యాంక్ యూ. వస్తాను, రెండుపోకు వస్తానే మళ్ళీ మీ టాక్కి.
అలాగే అలాగే, ఓకే అండి.
ఓకే, అనిల్ కుమార్ గారు నమస్తే.
నమస్తే సార్. నమస్తే సార్.
నమస్కారం.
ఒక్క నిమిషం అనిల్ కుమార్ గారు, ఒక్క నిమిషం.
రామచంద్రరావు గారు, ఇట్స్ గుడ్ టు సీ యూ అండి. ఐ విల్ టాక్ టు యూ అండి సమ్ టైమ్స్. ఓకే,
షూర్ షూర్.
నేను జాయిన్ అవుతాను, మీ టాక్ నేను జాయిన్ అవుతాను.
అలాగే అలాగే,
రైట్ సార్.
మీరు ఒక్క నిమిషం. మురళీకృష్ణ గారు, చెప్పండి సార్. మీరు
యాక్చువల్లీ నేను వెరీ ఓల్డ్ స్మాల్ సార్. యాక్చువల్లీ మాకు మా ఇంత అల్యూమినై స్ట్రాంగ్ అని మాకు తెలియదు సార్. నేను ఆంధ్రప్రదేశ్లో 2002 అంటే 2002 వరకు 2 years ఎంఎస్సీ చేశాను సార్. చేసిన తర్వాత చాలా ప్లేస్లో వచ్చింది, కానీ రాఘవరావు గారు NIL సూర్యనారాయణ గారి దగ్గర ఉన్నది, అక్కడికి వెళ్ళమని చెప్పాడు సార్. అక్కడికి వెళ్ళాను. అంటే ఐ గాట్ CSR అండ్ ICMR. కాకపోతే సూర్యనారాయణ సార్ గారికి ల్యాబ్ ఉండేది, కానీ ఆయన వేరే భాను ప్రకాష్ రెడ్డి గారిని వాళ్ళిద్దరు కలిసి చేసేవాళ్ళు. సో నేను సూర్యనారాయణ గారు ఆయనకు పరిచయం చేశాడు. అక్కడ PhD చేశాను సార్. చేసిన తర్వాత యూనివర్సిటీ ఆఫ్ కాలిఫోర్నియా ఇర్వైన్లో చేశాను సార్ ఫస్ట్ పోస్ట్ డాక్. బట్ అబౌట్ 1 year. దెన్ యూనివర్సిటీ ఆఫ్ మిషిగన్లో ఒక 4 years ఉండి దెన్ ఐ గాట్ వెల్కమ్ ట్రస్ట్ ఎర్లీ కెరీర్ అవార్డు.
వెరీ
మీ
గుడ్.
దగ్గర THSTI ఉంది కదా సార్? THSTI అని మీ దగ్గర ఫరీదాబాద్ లో ఉంటుంది సార్ ఒకటి. అక్కడ
అదేంది త్రిస్టీ అనేది ఉంది కదా, త్రిస్టీయా?
అవును సార్, త్రిస్టీ. దాంట్లో సైంటిస్ట్ వచ్చింది సార్ 2012 జనవరిలో. ఎయిమ్స్ లో వెల్కమ్ ట్రస్ట్ ఎర్లీ కెరీర్ వచ్చింది. కానీ మీరు చెప్పిన బేబీ రీజన్ సేమ్ రీజన్ సార్ నాకు. అంటే మై సన్ బోర్న్ ప్రీమెచ్యూర్ అండ్ ఈజ్ హెడ్ షేప్ వాస్ స్కల్ షేప్.
కన్స్ట్రైంట్స్ వల్ల మీరు అక్కడ ఉండిపోయి అంటారు.
నేను వచ్చేసాను సార్ హైదరాబాద్ లో. అంటే మా మిస్సెస్ దగ్గర పక్కన పల్లెటూరు సార్. సో ఆవిడ ఇక్కడ ఉండమంటే ఇక్కడ ఉస్మానియా ఫ్యాకల్టీ జాబ్ ఇచ్చారు సార్.
వెరీ గుడ్.
అంటే అక్కడ వెరీ ఫన్నీ సార్. నేను ఉస్మానియాలో సెటిల్ అయ్యడానికి బైఫర్కేషన్ 2014. So MSc, though I did PhD from Usmania NIN, but if you do a master's in other state, then you are non-local. Then నేను ఇక్కడ జాబ్ వచ్చింది సార్. I was fortunate that.
So now you are in University of Hyderabad కదా, very nice place.
Very nice place, sir. For competitive place, sir. I don't say it's nice place, but it's a very competitive place. That's good part, sir, actually. And
what do you mean by not I don't say nice place and competitive means highly competitive, you mean to say?
Competitive, we can go very good, sir, scientifically. Challenging, like lot of good people are recruited, sir. Nice place means, sir, we have to live 12 long years to become associates. Like I just finished 10 years, sir. Like, and they don't consider postdoc experience, no promotions, especially. And I backlog appropriate part, sir. Otherwise, like other categories, you can't given any promotion, sir.
మీరు ఎప్పుడయ్యారు ఇక్కడ MSc 2000 to 2002?
Yes, sir. Yes, sir.
సో మేము అయితే మీకు మాకు 1 decade గ్యాప్ ఉంది.
Yes, sir.
మేము
చెప్పాను కదా సార్. I stayed with సూర్యనారాయణ గారు for almost
92 మాది. 1990 to 1992.
సార్ మీ క్లాస్మేట్
నేను చెప్తాను మీకు మా క్లాస్మేట్స్ మేము 14 మెంబర్స్ 5 బాయ్స్ 9 గర్ల్స్. సో ఇప్పుడు దీంట్లో నేను, సూర్యనారాయణ, విజయ్ కుమార్, హరిబాబు, వెంకట చౌదరి అని ఉన్నారు. మేము 5గురు. తర్వాత అమ్మాయిల్లో ఇప్పుడు నువ్వు ఇక్కడ రాధిక ఉంది కదా, ప్రొఫెసర్. నౌ షీ బికేమ్ ఏ ప్రొఫెసర్. సో అండ్ సూర్యనారాయణ ఇది మిగతా వాళ్ళందరూ నాకు టచ్ లో లేరులే, కానీ ఒకసారి రాధికాని ఎప్పుడు చూసాను. ఇంకెవరు మీరు వీళ్ళు కాక ఇంకెవరిని అడుగుదాం అనుకుంటున్నారా?
యాక్చువల్గా మీ కొంచెం సీనియర్ సార్. ఆవిడ హేమలత మేడం గారి క్లాస్మేట్ సార్. తను ప్రొఫెసర్ డి శివప్రసాద్ దగ్గర PhD చేశారు సార్. ఆవిడ టెంపరీగా చేసి తను జస్ట్ షాక్ అయ్యాను సార్. మెంటల్ బ్లాక్ అయింది.
శారదా గారు అక్కడ CFTR చేశారు PhD. డాక్టర్ శారదా
అంటే యాక్చువల్లీ షీ డిడ్ PhD, సపోజ్ టు బి డిడ్ PhD శివప్రసాద్ గారి దగ్గర రిజిస్టర్ అయ్యారు సార్. హేమలత గారి క్లాస్మేట్. బట్ ఆవిడ కొన్నాళ్ళు మైసూర్లో వర్క్ చేసి అంటే టు రీడ్ సమ్ ఎక్స్పెరిమెంట్స్ అండ్ కేమ్ బ్యాక్ సార్. ఆమె మా కజిన్ గారు చేసుకున్నారు సార్ మ్యారేజ్. ఐఎమ్ జస్ట్ షాక్ అయ్యాను సార్ మెంటల్గా. ఎందుకంటే చాలా ఆల్మోస్ట్ 10 years అయింది సార్ ఆవిడ కలిసి. న్యూయార్క్ లో ఉంటారు సార్, రోడ్స్ ఐలాండ్ లో ఉంటారు తను.
ఓకే
అది చెప్తాను సార్, జస్ట్ ఐ విల్ గెట్ హర్. సో తను మా బంధువు సార్. యాక్చువల్లీ తను చెప్పారు. ఎస్ సార్.
సో మీకు ఇక్కడ సెంట్రల్ యూనివర్సిటీలో ప్లాంట్ సైన్సెస్ లో మన ఎవరు మన రాజగోపాల్ అన్నాడు కదా, ప్రొఫెసర్ రాజగోపాల్.
రాజగోపాల్, ఎస్ సార్.
నేను CFTRIలో ఉన్నప్పుడు మా ల్యాబ్ కి ఆయన తిరుపతిలో SV యూనివర్సిటీలో PhD చేసి టెక్నిక్స్ నేర్చుకోవడానికి, ఆ స్పైరులినా గురించి చేసుకోవడానికి CFTRIకి మా దగ్గరికి వస్తే నేను టీచ్ చేయడం జరిగింది ఆయనకి. అంటే నేను కూడా అప్పుడు స్పైరులినా మీద ఒక పార్ట్ గా చేసేవాడిని. ఐ మీన్
ఆయన స్పైరులినా మీద చేస్తున్నాడు. కాకపోతే ఆయన ఒక నిష్ లో ఆయన హీ ఎవాల్వ్డ్ యాజ్ ఏ టాప్ క్లాస్. ఫోటోసింథసిస్ చేస్తాడు సార్ వాటిలో. ఐ థింక్ సమ్ ఆల్గల్ లో చేస్తాడు. హీఈస్ ప్రొఫెసర్ నౌ. ఈజ్ డూయింగ్ వెరీ గుడ్ సార్. ఈజ్ డొమైన్ సార్. యాక్చువల్లీ ఆయన కూడా జపాన్ కొలాబరేషన్స్ ఉన్నాయి సార్ బాగా. సార్ ఈ DHR ఉంది కదా సార్, డిపార్ట్మెంట్ హెల్త్ రీసెర్చ్, అది మీకు అసోసియేషన్ DHR డిపార్ట్మెంట్ హెల్త్ రీసెర్చ్ ఉంది కదా సార్, అది ఎట్లా సార్? అది మీకేమైనా కనెక్షనా సార్?
డిపార్ట్మెంట్ ఆఫ్ హెల్త్ రీసెర్చ్ ICMR లో కంపెనీ. మీకు చూడండి, ఇప్పుడు మినిస్ట్రీలో 2 divisions ఉంటాయి. మినిస్ట్రీ ఆఫ్ హెల్త్ అండ్ ఫ్యామిలీ వెల్ఫేర్ లో మేము వచ్చేసి డైరెక్ట్ గా అటానమస్ ఇన్స్టిట్యూట్ అండర్ మినిస్ట్రీ. మా యొక్క ఎంప్లాయ్మెంట్ మొత్తం జరిగేది హెల్త్ మినిస్ట్రీ ఆధ్వర్యంలో జరుగుతుంది, హెల్త్ సెక్రటరీ ఆధ్వర్యం.
ఓకే సార్.
సో మాకు అన్ని ఆర్డర్ మా యొక్క ప్రమోషన్స్ అన్ని వచ్చేది హెల్త్ సెక్రటరీ సైన్తో వస్తుంది.
ఓకే.
సో ఇఫ్ యూ వాంటెడ్ టు లీవ్ అబ్రాడ్ ఆల్సో, కానీ అవుట్ ఆఫ్ స్టేషన్కి వెళ్ళాలంటే పర్మిషన్ హ్యాస్ టు కమ్ ఫ్రమ్ మినిస్ట్రీ నుంచి రావాలి అనమాట. సో మిగతా మీరనేది ఏంటంటే ఇంకో డివిజన్ ఉంటుంది దాని కింద, DGHS అని. DGHS అంటే డైరెక్టరేట్ జనరల్ ఆఫ్ హెల్త్ సైన్సెస్. దాని కింద మీరు చెప్పేది వస్తుంది
అది గాట్ ఇట్ సార్. యాక్చువల్గా ఐ పబ్లిష్డ్ రీసెంట్లీ సార్, ఇక్కడ నుంచి లాస్ట్ ఇయర్ సెల్ డెత్ అండ్ డిసీజ్, JBC అట్లా పబ్లిష్ చేశాను సార్.
మీ ఏరియా ఏం చేస్తారు ఇక్కడ మీరు యూనివర్సిటీ ఆఫ్ హైదరాబాద్ లో? మీరు
నేను డయాబెటిక్ నెఫ్రోపతిలో చేస్తాను సార్. అంటే లైక్ ఆ సెల్స్ ఉన్నాయి సార్, పొడోసైట్స్ అని. ఆ సెల్స్ దే అండర్గో సెవరల్ డిఫార్మిటీస్ డ్యూరింగ్ డయాబెటిక్ నెఫ్రోపతి. అవి గేట్ కీపర్ సార్, బ్లడ్ యూరిన్ బ్యారియర్, దిస్ పొడోసైట్స్. So those cells are terminally differentiated cells and they undergo a lot of injuries. I am interested to understand how several insults like hypoxia kani, high glucose, advanced glycation end products, elevated growth hormone levels, how they tamper these podocytes biology. And also we investigate how and why these podocytes shed in the urine. అంటే సార్ లైక్ కంచె చేను మేస్తే అంటారు కదా, ఈ పొడోసైట్స్ కాపాడాలి సార్. యాక్చువల్లీ అవి గ్లోమ్యులర్ క్యాపిలరీస్ చుట్టూ టైట్ గా కనెక్ట్ చేసి ఒక మెష్ లాగా హెల్ప్ చేస్తాయి వాటి యొక్క సెల్స్ యొక్క ఫుడ్ ప్రాసెస్. ఆ గ్యాపుల్లో యూరిన్ బయటకు వస్తుంది సార్, లో మాలిక్యులర్ వెయిట్. కానీ వైడనింగ్ అవడం వల్ల వాటి యొక్క ఫుడ్స్ వైడన్ అవడం వల్ల కానీ ఈ సెల్ యాక్చువల్లీ డిటాచ్ ఫ్రమ్ ద బ్లడ్ వెజల్, అంటే క్యాపిలరీస్ డిజాల్వ్ అయితే దెన్ లాట్ అమౌంట్ ఆఫ్ ప్రోటీన్ కమ్స్ ఇన్ ద యూరిన్. సో ఐ ట్రై టు like explain why these podocytes undergo deformities.
First time I am listening, interesting, because it's a medical
I joined
component.
around 7-8 years back, around 7.5 years back, sir, and they gave me service. What I did was Mania. It's 10 years I completed, sir. As assistant professor, 2 more years we had to wait.
Acha, 12 years you have to wait
sir,
for the assistant professor to become associate
teaching
professor.
I teach. So you asked about teaching, sir. Kuch internet flux ayin sir actually.
Yeah, మీ వాయిస్ కూడా మధ్యలో బ్రేక్ అవుతా ఉంది అక్కడ మధ్య ఏదో నాకు
ఏం లేదు సార్, 12 years కూర్చోవాలి సార్ అక్కడ.
ఓకే, we will talk on the regular.
I think signal నా దగ్గర కూడా ఫ్లక్చుయేషన్ ఉంది సార్.
జనరల్ ప్లాట్ఫామ్ మీద వద్దు, సో తర్వాత మాట్లాడదాం. ఓకేనా? సో గుడ్ టు నో అబౌట్ యువర్ రీసెర్చ్. సో నా రీసెర్చ్ ఇప్పుడు ఇండియాలో
థ్యాంక్ యూ సార్,
గెట్ లాఫ్ అన్నమాట, బికాజ్ చాలా యాక్చువల్ గా మీరు చెప్పిన ఈవెంట్స్ ని మీరు వినండి నా టాక్
సార్ క్లాస్ బికాజ్ హి వాస్ లైక్
no
one of my like brother kind of thing from NIM. We used to sit next to each other for like 7 years, sir.
Very good, very good. So please
Annapurna sir.
Annapurna, okay, maybe she is so senior kada, we don't know. Maybe we might have heard, but I do not know.
Yes.
మేము యాక్చువల్ గా మాకు ఇమీడియట్ సీనియర్స్ ఎవరంటే రాఘవరావు గారు.
అవును, ఓ తెలుసు నాకు, మా గురువు సార్. He taught us, he's a mentor. Actually we came from very small hamlets. We don't know really anything, sir. I got second rank. I don't know how I got second rank in the entrance examination. I still know, I don't know.
That is called
nobody got problem.
That is called life, na? Because even same is the case here. So because finally actual ga na interest anta it has come from the department of biochemistry. Let me tell, I actually had to go to the Andhra University biochemistry. అక్కడ మనకి ఎవరు మాకు ఉంది, శివప్రసాద్ గారు, రమణ గారు, రామారావు గారు, అండ్ అఫ్ కోర్స్ TMR అండ్ TG రెడ్డి గారు అండ్ శ్రీరాములు గారు అండ్ హేమలత మేడం. So these 7 to 8 staff who are taught to us.
Yes, sir.
So tarvata then I came to know that I should not stop here.
Have some.
I should go for further అని మీలాగా అందరిలాగానే అప్పుడు ఆంధ్ర యూనివర్సిటీ బయోకెమిస్ట్రీ డిపార్ట్మెంట్ ఇప్పుడు ఎలా ఉందో తెలియదు కానీ అందరూ దే రైట్ ఫర్ CSR రాస్తారు, గేట్ రాస్తారు. ఆ రోజుల్లో అప్పుడు 10 మంది రాస్తే 10 మందికి వచ్చేది, 15 మంది రాస్తే 15 మందికి వచ్చేది.
తగ్గిపోయింది సార్, తర్వాత స్లోగా తగ్గింది. మా బ్యాచ్ లో పాపం నాకు ఒక్కడికే వచ్చింది సార్. నా ఫ్రెండ్స్ నాకన్నా బ్రిలియంట్, దే కేమ్ ఫ్రమ్ వెరీ గుడ్ సిటీ కాలేజెస్, బట్ దే కుడెంట్ మేక్ ఇట్. బట్ చాలా మంది పిహెచ్డి అయితే చేస్తున్నారు సార్ కంప్లీట్ గా మన డిపార్ట్మెంట్ లో. లైక్
వెరీ గుడ్.
చాలా మంది చేశారు సార్ నా క్లాస్
రోజుల్లో. సో దెన్ వి వెంట్ ఫర్ అన్ని ఐఐటీస్కి వెళ్ళడం జరిగింది. అదొక పెద్ద స్టోరీ. సో దెన్ ఫైనల్ గా వీ కేమ్ బ్యాక్. టిఎంఆర్ సజెస్టెడ్ వై యూ వాంటెడ్ టు గో ఫర్ MTech. Why don't you think of like PhD itself? That will become a waste of time for you already postgraduate. అంటే
మా టైంలో లేదు సార్ మా టైంలో సార్ యాక్చువల్గా మాకు ప్రొఫెసర్స్ అంటే మీరు చెప్పిన సార్ అప్పుడు ఆయన పక్కన గీతంకి వెళ్ళిపోయాడు సార్. ఇక్కడ రిటైర్ అయ్యి గీతంలో ప్రొఫెసర్ గా చేస్తున్నాడు.
ఎవరు?
ఆయన ఒక రెడ్డి గారని ఒక సారు. బట్ మా దగ్గర సెలెక్టివ్ గా ఉన్న వాళ్ళు శివప్రసాద్ గారు, టీజీ రెడ్డి గారు రిటైర్మెంట్ దగ్గరగా ఉన్నారు సార్. ఆయన కూడా యాక్టివ్ గా పార్టిసిపేట్ చేయలేదు సార్. టీజీ రెడ్డి గారు రిటైర్మెంట్ దగ్గరగా ఉన్నారు మా టైంలో. శివప్రసాద్ సారు, ఆతోటి సార్ కూడా కొంచెం ఉండి తర్వాత వేరే యూనివర్సిటీలో వైస్ ఛాన్సలర్ అయ్యారు సార్, ఆతోటి రామారావు గారు.
ఐ కేమ్ టు నో నిన్న యాక్చువల్ గా ఐ కేమ్ టు నో నిన్న ఇనాగరల్ అడ్రస్ లో రాఘవరావు గారు, రామారావు గారు ఎస్కే యూనివర్సిటీ ఫార్మర్ వైస్ ఛాన్సలర్ అంటే నాకు తెలియదు యాక్చువల్ గా. నిన్న నాకు ఫస్ట్ సర్ప్రైజింగ్ అనమాట ఈ న్యూస్. సో నేను యాక్చువల్ గా అంటే ఐ వాజ్ ఇన్ డిఫరెంట్ కాంటాక్ట్ అంటే డిఫికల్ట్. యూ ఆర్ ఇన్ యువర్ జాబ్ మీన్స్ యూ విల్ బి టోటలీ ఆక్యుపైడ్. సో కానీ దీన్ని రావడం వల్ల ఐ గాట్ టు నో. సో నాకు బట్ రాఘవరావు గారు తో ఒక 4 సంవత్సరాల క్రితం 5 సంవత్సరాల క్రితం కాంటాక్ట్ ఎస్టాబ్లిష్ అయింది. సో
సార్ మనకి యాంకర్ అందరికి అంటే చిన్నవాళ్ళకి పెద్దవాళ్ళకి హీజ్ అంటే సుభాష్ని గారు తర్వాత బయట అంటే తను చేశారు కానీ తను మేము ఉన్నప్పుడు లేరు కదా సార్, తెలీదు. యాజ్ వెల్ మేబీ చిన్నవారు మీకన్నా. రాధికా గారు మేబీ మీ క్లాస్మేట్ గారు కానీ తను అసలు మాకు అసలు పరిచయం లేదు సార్. మాకు షీ వాజ్ నాట్ దట్ టీచర్. బట్ లేటర్ when I become faculty at TCR, she used to when Professor Raghav invited me for a couple of lectures, I used to interact. But otherwise, Professor Raghav is a big anchor for us. He's like a focal point. Sir, I think, sir, I'll change my settings, sir. I'll rejoin because here internet fluctuation undi.
So
when I'm talking, when you're giving lecture, I have to listen.
I will also rejoin because there is a fluctuation. Voice is cracking అవుతుంది. ఇక్కడ బాగా ఉన్నట్టుంది. మీ సైడ్ నుంచి కొంచెం లో బ్యాండ్ విడ్త్ వస్తుంది, మెసేజ్ వస్తుంది. చూస్తాను.
That's what I noticed it, sir. See you, sir.
Okay.
Namaste,
Okay,
sir.
then.
See you, sir. Yes.
Yeah. Thank you.
హలో అనిల్, బాగున్నారా?
నమస్తే మేడం.
నమస్తే అండి, బాగున్నారా?
బాగున్నా మేడం. ఎలా ఉంది మేడం? చాలా యూఆర్ ఆర్గనైజింగ్ ఇన్ వెరీ గ్రేట్ వే.
ఓకే, థ్యాంక్స్ అండి. వైజాగ్ వచ్చినప్పుడు రండి.
అయ్యో భలేవారు మేడం, నేను వైజాగ్ వచ్చేదే మన డిపార్ట్మెంట్ కోసం. నిజంగా వేరే పనే ఉంటుంది మేడం వైజాగ్ వస్తే. అంటే వీ హ్యావ్ టు కమ్ ఫర్ అవర్ స్కూల్. యాక్చువల్లీ నేను చాలా సార్లు ఐ లాంగ్ టు కమ్ మేడం, బట్ ఏంటంటే కరోనా వచ్చిందని రాలేదు.
అవునవును. లెట్ us wait for 5 more minutes. స్పీకర్ జాయిన్ అయినట్లేరు సార్.
విన్నానండి.
సార్ యు ఆర్ దేర్ లైన్. వెల్కమ్ సార్.
థ్యాంక్ యూ అండి, థ్యాంక్ యూ. బికాజ్ మీ లాస్ట్ టైం నేను 2017 లో ఒకసారి వచ్చినప్పుడు మిమ్మల్ని కలిసినట్టున్నాను, గుర్తుందా?
అవునవును, రాఘవరావు సార్ దగ్గరకు వచ్చానట్టున్నారు.
అవునవును. యాక్చువల్ గా నేను 92 వదిలిన తర్వాత డిపార్ట్మెంట్ కి ఒకసారి 2006 లోనే, సారీ 96 లో ఒకసారి వచ్చానండి. డిపార్ట్మెంట్ శివప్రసాద్ సారు, రమణ సారు దే హావ్ ఆర్గనైజ్డ్ ఎస్బిసి మీటింగ్ ఆర్గనైజ్ చేశారు.
ఓకే సార్.
డిపార్ట్మెంట్ చూడటానికి కోసమే 96 లో వచ్చాను. సో తర్వాత అప్పుడు ప్రజెంట్ చేయడం జరిగింది. మా అప్పుడు రీసెర్చ్ స్కాలర్స్ సిఎఫ్టీఆర్ లో పిహెచ్డి స్టూడెంట్ గా ఉన్నప్పుడు. తర్వాత 96 తర్వాత నాకు 99 లో ఐ మూవ్ టు జపాన్ వెళ్ళిపోయాను. సో 2002 లో నాకు ఏదో మళ్ళీ యంగ్ సైంటిస్ట్ అవార్డులు వస్తూ ఉంటాయి కదండి, సిఎఫ్టీఆర్లో వస్తే ఏదో అవార్డులు ఇచ్చడానికి వచ్చాను, తీసుకున్నాను. మళ్ళీ 2000 తర్వాత విశాఖపట్నానికి 96 తర్వాత 2017 లో రావడం జరిగింది.
Yeah, I met you actually when you met.
అది నా ప్రైవేట్ విజిట్ మీదకి వచ్చాను. జస్ట్ ప్రైవేట్ విజిట్ మీద వచ్చి ఐ వాస్ దేర్ అబౌట్ 5 hours ఉన్నాను, 5 to 6 hours ఉన్నాను వైజాగ్ లో. సో నేను నెక్స్ట్ డే నాది నెక్స్ట్ డే నాది మళ్ళీ జపాన్ కి నా ఫ్లైట్ ఉంది. సో అలా రావటం జరిగింది. సో వచ్చినప్పుడు అది మిమ్మల్ని తీసుకెళ్ళాడు ఇక్కడ ఇప్పుడు అని చెప్పారు, మీ పేరు చెప్పారు, గుర్తుంది. తర్వాత వెంటనే నేను ఆ రోజు మళ్ళీ వచ్చేసి వెళ్ళిన తర్వాత మళ్ళా ఇప్పుడు మళ్ళీ మీతో ఇది చెప్పంగానే ఆ రోజు యాక్చువల్ గా రాఘవరావు గారిని తర్వాత నేను బయటకు వస్తుంటే ఒకసారి అదిగా కనపడింది, నా క్లాస్మేట్. సో తర్వాత అలా కలిసేసి అలా జరిగింది. మళ్ళీ ఇప్పుడు మళ్ళీ నేను మళ్ళీ జపాన్ నుంచి రావటం ఇలా జరుగుతుంది మళ్ళీ. మేడం బాగున్నారా? బాగున్నానండి. ఐఎమ్ ఫైన్ మేడం. ఎస్టర్డే ఐ హర్డ్ మీరు అదే మీది రిటైర్మెంట్ అని విన్నాను నిన్న దీంట్లో చాలా, కానీ అప్పుడే రిటైర్మెంట్ అయిపోతున్నారా? అవును, అప్పుడు గుర్తొచ్చింది, మేము కూడా 50 దాటాం కదా,
నమస్కారం మేడం. హేమలత మేడం నమస్కారం. రాఘవరావు సార్ నమస్తే సార్. అయ్యో మీరు ఎందుకు సార్ నమస్కారం పెడతారు?
అనిల్ ఉస్మానియా యూనివర్సిటీయా, సెంట్రల్ యూనివర్సిటీ కదా?
సెంట్రల్ మేడం. స్టేట్ సపరేట్ అయిన తర్వాత బయటికి పంపించేశారు మేడం, అందువల్ల ఇక్కడికి వచ్చాను. సుభాష్ మేడం మీరు ఎప్పుడు చెప్తే అప్పుడు స్టార్ట్ చేస్తాను మేడం. మోర్ ఎక్సైటెడ్ దాన్ రామచంద్రరావు సార్.
ఓకే
మేడం, కాసేపు నేను చేయలేకుంటాను పక్కన.
భోజనం అయిందా రాఘవరావు గారు?
ఇప్పుడే.
అవునా?
నమస్తే సార్ రాఘవరావు సార్. నమస్తే సార్, బాగున్నారు సార్? గుడ్ సార్, బాగున్నాను సార్. The things are going well సార్, నో ప్రాబ్లం, బానే ఉంది.
ఈ శ్రీనివాస్ అనేవారు డ్యూ టు ల్యాండ్ వీడియో ఈజ్ నాట్ కరెంట్లీ అవైలబుల్ సంథింగ్ ఏదో ఎక్కడ ప్రాబ్లమ్? డి శ్రీనివాస్ దగ్గర మీ సైడ్ నుంచే మెసేజ్ వస్తుందండి.
నా సైడ్ నుంచా సార్?
ఇది క్వశ్చన్ అది ఎక్స్క్లమేటరీ మార్క్ వచ్చి కొంచెం బ్యాండ్ విడ్త్ అని మీరు నుంచి కొంచెం లోగా ఉంది వీడియో.
సార్ ఎవ్రీథింగ్ ఈజ్ క్లియర్ ఫ్రమ్ మై ఎండ్, బానే.
ఓకే రైట్ నో, అంటే ఇంటర్మిటెంట్లీ ద మెసేజ్ ఈస్ క్రాపింగ్ అప్.
ఓకే.
మీరు ఏ బ్యాచ్ అండి శ్రీనివాస్ గారు? ఇక్కడ ఆంధ్ర యూనివర్సిటీ.
మాది సిల్వర్ జూబ్లీ బ్యాచ్ సార్, 96, 98.
96, 98.
Yes, sir.
Raghora garu, no, Raghora ma ఇది 97, 99 కదా మీది రాఘవరా గారు?
Yes, sir. Raghora sir, my teacher, sir.
ఎలా అవుతుంది నీకు? నువ్వు ఎప్పుడు?
నో నేను మనం 90 to 92 మాది. కన్ఫ్యూజ్ అయిపోయాను చాలా.
రాఘవరా సార్ మా టీచర్ సార్.
నేను షాక్ అయ్యాను సార్.
ఇప్పుడే షూట్ చేసుకున్నారు కదా, అందుకే కన్ఫ్యూజ్ అయ్యాను.
దేశంగా దేశంలో ఏదో 20 సంవత్సరాలు ఉన్నాము, అందుకని కన్ఫ్యూజ్ అయిపోయాను.
Shall we start అండి?
మార్నింగ్ ఏఎస్పీఆర్ గారి గురించి చెప్తున్నా కదా, తెలియదన్నావు కదా.
ఏంటండి?
శివప్రసాద్ ఆయన టీచర్ ఇక్కడ, మనం జాయిన్ అవ్వక ముందే టీచర్.
ఓకే ఓకే ఓకే.
రినౌండ్ టీచర్ లైక్ శివప్రసాద్ రావు, చాలా మంచి టీచర్.
సార్ ఇప్పుడు ఎక్కడ ఓవర్సీస్ లో ఉంటారా?
లీడ్స్ లో ఉంటారు సార్. లీడ్స్.
ఓకే ఓకే.
యూనివర్సిటీ ఆఫ్ లీడ్స్. one of the great teachers of this department.
Okay, so we missed because I don't know. అందుకని తెలియదు, వినే ఉంటాను కానీ పేరు. సో అందుకని మార్నింగ్ మాట్లాడినప్పుడు కన్ఫ్యూజ్ అయ్యాను. డిఎస్పీ గారా అంటే ఏఎస్పీ అంటున్నారు. సో
సుధా, హాయ్ సుధా.
హలో రఘురావు, ఎలా ఉన్నారు?
బాగున్నా అండి, మీరు ఎలా ఉన్నారు?
బాగున్నాను.
Sudha madam, where are you right now?
Boston అండి.
Boston.
హలో మేడం, గుడ్ ఈవినింగ్.
హాయ్ సుభాషిని, ఎలా ఉన్నారు? బాగా ఆర్గనైజ్ చేస్తున్నట్టున్నారు.
ఫైన్ అండి, ఫైన్ మేడం. విత్ ఆల్ కోఆపరేషన్ ఫ్రమ్ alumni.
But you are there in the center, right? You, Raghuram, Hemlata madam.
Sir, we'll start.
Most welcome.
Okay, welcome back. Good afternoon. My name is Anil Kumar. I'm a alumni of Department of Biochemistry. I did my master's from 2000 to 2002. Then I moved to National Institute of Nutrition, Hyderabad, and I did a couple of postdocs at UC Irvine and University of Michigan. And presently, I'm an assistant professor at University of Hyderabad. I feel it is extremely like privileged to introduce today's speaker, this session speaker, Dr. Ramachandra Rao Satuluri, for various number of reasons, because he's our great alumni and also has a very inspiring story. Dr. Rao received his master's from our department in 1992. Then he moved to the CFTRA for his PhD. He received his PhD in 1999. Then he moved to University of Japan under very prestigious fellowship called JIST, that is called JSPS Post Office Fellowship. In 2000, initially from 2001 to 2003. But interesting fact is that he spent almost 18 years in the Japan at various stages, from scientists, senior scientists at various institutes. So one of the important thing about Dr. Rao's contribution to the medical world is that they designed a single cell microarray. They developed the single cell microarray microfluidics device for the development of monoclonal antibody. It led to the beginning of a venture business company called SC World. And later, the SC World company was taken over by Vivalis. It's a France-based company, and they are tailoring this one, patient-based monoclonal antibodies. And as I said, Dr. Rao moved from Osaka University. Then he moved to the National Institute of Advanced Industrial Science and Technology at Tsukuba, then Kishu Center. Then at Saga University, served as visiting faculty. So altogether, 18 years he spent various designations in very highly competitive world in Japan. Then he returned to India in the August 2017, and he started his journey as an associate professor at the Department of Reproductive Biomedicine at the National Institute of Health and Family Welfare. The interesting fact is that Dr. Rao is worked at single cells, microarray microfluidics and nanomaterials. And also the nanomaterial interaction with embryonic stem cells and iPS cells, and development of
nano needle array-based single-cell sorting system, and microreactor-based metal extraction and recovery. These are very application-oriented science he pursued during his research career. So he has more than 60 publications. He trained like more than he has supervised five PhD students and more than 10 master projects. He's serving as associate editor for journal Health and Population Perspectives and Issues. It is a NIH journal, and also section editor for Journal of Indonesian Chemical Society, Indonesia, and is serving as a review editor for or editorial board member for several journals of the Frontiers, which is based upon Switzerland. With this less is more. With this less introduction, I would like to request Dr. Rao to deliver his talk. It's all yours, sir. Welcome.
Yeah. Thank you, Dr. Anil. If I understand correct.
Yes.
For the nice introduction, and I'm very happy, first of all, to thank you so much, Dr. Subhashini and Professor Raghav Rao, and for inviting me to deliver my contributions, what I have done so far. But what I have done so far, it is 1 hour, it is very short. So I thought that I will give you only 2 or 3 components. And after I moved to, CFTRI, I have done my PhD over there. That was mainly on the plant cell cultures and mainly for the biotransformations. And after that, I moved to Japan. So there I was doing many of the today forenoon also we heard, in another angle, that is polycyclic aromatic hydrocarbons and its metabolic pathways. So mainly I have done it's the biomineralization and application side, and then biometabolic pathways and so on. So it was not exciting me when I went to Japan in the beginning, because what I know in India, I am doing the same in different system altogether. So I felt that I should switch over to the area which I don't know anything. But based on the biochemistry and the background that I have and those things, then I was thinking that is the area is that, because when we do any of the analysis, any of the assay, we do like a offline, each one reaction, endpoint, measurement, and then we put together, we corroborate, and we try to say so and so phenomena is leading to so and so things. So, but when I see that, why don't we put together all the things in one go to see in a more like a real-time fashion, so that we would be able to understanding things much more betterly and closely and better understanding. So with that, I thought that we should see and we should aid of because we do reactions and assays and things in the milliliters and because huge volumes we do. In the real sense, we do not need to that level where we will be doing and hours together, days together to do any of the reaction or any of the assays. To be frank, only the reactions just takes place at the very, very quick level. So, but we are doing actually most of the phenomena that we are actually not addressing, because it's also the knowledge that already public domain is also very meager. So therefore, if we go into those things and if we aid any of at the like a molecule level, at the single atom level things, so we would be able to understand the biological intriguing phenomena in depth. So therefore, we thought that because our department in way back in 2000 in Japan, and it is more of like working on the micro fabrication and nano fabrication, where we aid the tools of micro and bio devices for various biomedical and bio applications. So we call as a bio devices or bio chips, you would be calling them. So those actually, some of those findings, what I have done in Japan, I am going to give you 3 or 4 here, and to see some of the success stories. And what I am doing here right now, I am not giving, because I had to get some more time to truly make some of the outstanding results yet to come. Okay, and with this background, and before getting into the things, I should thank the faculty who taught me in the Department of Biochemistry, Andhra University, starting from Professor TMR and Professor TG Reddy. I do understand from yesterday's that they were no more, and they let them be, rest in peace there. And then I think Professor Ramana also, Sir also was actually no more I understood from the Professor Raghav. And yesterday I was very happy to see Siva Prasad sir and Ramarao sir, and of course Hemalata madam. I just had a brief talk now, and Sri Ramulu sir. So these are the faculty who used to taught us during my 1990 to 1992 in my stay in the Andhra University. Actually, Andhra University, I should say that I should owe a lot. That is the place where I learned and more interest for the higher, the research. Actually, the seed has been, it has come into my mind. So that's why I pursue my career. And where I am today, it is after that the biochemistry department and CFTRI Mysore and my. Therefore, I am here with this thing. Let me share my slides. Is this one? Am I sharing my slides? Am I audible, please? Somebody can answer.
Yes, you are. I can see the slides.
Okay, right. So with this, actually, today I actually try to say my presentation is on nano and micro devices for biomedical application. It is too vast. Okay, and but today I will quickly go through. And if you are, if the time is not permitting, you are most welcome to reach anytime. And my details are with the Department of Biochemistry. And if anybody interested for any collaborations, most welcome. Okay, and with this, as I told you in my introduction, is that any of the things like we have here in this slide, we have healthcare and we have the food and we have the agriculture and environmental. Anything if you wanted to any of the chemicals and the biomolecules or the cells and so on, any of the biomolecules means let it be like a DNA, RNA and proteins and so on. Any of the things that you wanted to measure, those you wanted to identify, to clarify any of the mechanism underlying the pathways or anything you need to detect. The detection should be rapid and should be quantitative and preferably should be low cost and one can easily do, and it should be user friendly. At the same time, put together all the things should be high throughput, so that you would able to see many of the reaction put together in one go and more concrete manner, more visualized, more envisaged manner, so that we can try to understand. But putting together all the things, it is not that easy. Okay, and therefore, so here I am going to tell in the beginning few slides before going to mostly today my talk is on the how actually these devices have been employed for the single cell research and actually how it has been aid for our investigation that you will be seeing but in the beginning I'm going to say something about how actually the DNA are the small molecules, any of the peptides and any of the things that you can identify, detect much more rapidly and quickly, and to identify their signaling pathways and so on for various reactions that you are going to see that. Okay. And here, let me say that here, as you know that you all know that if you want to do any amplification of the DNA assay, because actually these things we do for the, you might have heard POCT, patient care, point of care diagnostics, and that will be near the bed. The things will be analysis can be done, the diagnosis can be done very rapidly, and the results will be momentarily. It can be shared to the doctors, and the treatment can be given. So in that angle, we always thought of doing the reactions very quickly, not compromising the detections or the sensitivity or any of the investigation. So therefore, when you do the PCR or when you do the RT-PCR, you always do either by any of the 20 or 50 microliter assays, and you do for 3 hours by doing all the reactions, and then at the end point you try to measure, and then after that you go on to see whether like agarose electrophoresis, whether the target that you are aiming for, the gene that has been amplified or not, and then you quantify and then you go for so on and so forth for further analysis. But it is actually, in my view, that it is very long, and so therefore we thought that why don't we develop, and you need like a plenty of volume. It's because if you are looking for that, if you are looking for any of the assays, many of the things that you are doing, it should be at the nanoliter and the picoliter. I mean, so that you can do in a microliter volume, you can do hundreds and thousands of assays can be done without compromising the things. For that, if you wanted to realize that, we need to develop the platforms wherein you can do the assays very efficiently, effectively things. So here, as I told you in this cartoon, as I'm showing here, device is there. Why they advanced? Here it has been shown that when you do in the bulk, if it has been amplified, it has been in the diluted. So therefore, you would need actually more and more volumes, longer time to assess. But if you do your platform is very tiny, and when you amplify things, and therefore it put together, it adds up to the much shorter times, and you can do the detection much quicker. So therefore, we have here, and this is the platform wherein we have used like a micro chambers. It is came out, I mean, we fabricated from the silicon wafer. Silicon wafer, we use like a fabrication, means you use like a various kinds of photolithography or any of the things that you will see in the next slide. I do understand, please understand that most of the details you might not be following it, but you will come to know when you see that, okay? And then what we do here, and when you wanted to, I mean, you can do about the nanoliter level reaction. If you wanted to handle that, you need to have the robots wherein you have to do that. Also, we developed later on, and in the beginning, we actually commercially we procured, and how it can be done quickly for doing about like a, I mean, for about 40 liter microliter, where you can do one reaction, you can do about more than 1,000 to 1,500 assays, you can do that. And to do that PCR mixture, and this is the place, this is the way we have done, and you have the silicon wafer. It is about 4 inches wafer, and it will undergo various kinds of steps as we see here, and oxidation and photolithography, and you have the pattern that you wanted to print, and that you will make with either like using like a CAD. CAD software and AutoCAD, and then you print it by using a photolithography, nothing but a UV, ultraviolet source, you will use it, and then you have the pattern, whether it is a negative resistant, positive photoresist, and so on, and then you can go into a couple of reactions, and you will have the one which you are seeing at the down. You can see this is the size, slide glass size, and it has about 1248 microchambers are there, and each chamber you can see here. And it is like, it is interestingly is that only the chamber inside is hydrophilic, and chamber outside where you are seeing here, and these are the hydrophobic, which means that when you are doing each chamber, each reaction chamber serves as a one container, and without no compromising of the no cross contamination. So therefore, we have used this platform for very many applications, how you can do for the DNA, and you can do for the, small molecule, how you can do the drug discovery screenings and its potential, and also for the small molecule identification for the, for the alternative to the nerve growth factor for the blood-brain barrier that can cross, that you will be seeing that, okay? And this is the way where I see, I will mute it. So this is the robot that you do. You have the reaction here, and you put the chamber here, and it takes the whatever the target from here, and it comes over here. And because here what we do for the DNA means we put in the beginning mineral oil. Mineral oil is the one as a serves as a lead, and then it will just the pattern in which we want. It will dispense it, and it will go whatever the pattern you want. And once you have that, after the assay or the PCR and the reaction, you can put it, this is the one which you have the scanner, because if you want to read out whether the PCR has taken place or the amplification has done or not, and you have to have a readout. So generally we take that and we go for agarose electron. You don't have to do that. And here, since it is a fatty now, 20 or 40 nanoliter reaction, and there is no, the drying effects are there, because this Cartesian system nano dispenser, it has inside the sterility, and it has a humidity is maintained so that the samples will never get dry. And so then you can see here, and you can put in this place wherein you have the inlet, and you can see the readout. It can be done whether it's amplification or not. So this is just to show that in the beginning we put the primers, and then you get dried up, and then we put our targets, and we serve as a mineral oil is there. Mineral oil serves as a lid over that, and you can go for the like your PCR reaction, and if you wanted to do the reaction, either Tachman chemistry or any of the things that you can do, and you can find out whether the amplification has done in the nanoliter chambers, and those chambers you can do by the cyber green chemistry. If you are using Tachman chemistry, you can use cyber green chemistry, or if you are using for other, you can take, or sometimes you can withdraw, you can retrieve a sample from there and you can do for further analysis, and you can do electrochemical based sensing or any sensing that you can do. It is up to our target how we can do that. And we have applied this is for various kinds of beta-actin and SR and the RHT, rhesus D genes, various kinds of templates that we have tested, and you can see the real-time fashion also these genes after 23 cycles. In the beginning, when we tested, it's about, it needs, you don't need about 3 hours time. You need about less, like just 40 cycles is enough to get your target to reach the amplification, and you can find out, and you can see how it has been. You can negative target, you put that, whether you can also proof of concept. We have tested how actually these devices are applying. This is just to show the another thing. Of course, these are all has come publications in good journals, obviously, because once you do any research will lead to a publication, otherwise it will not see the light of the day in the public domain. Okay, and then after that we also have seen, this is to see like electrochemically, and this is also a microfluidic chip wherein you have here, and we use like a micro channels are there here, and through micro syringe pump we will put the micro PCR reaction, and inside there is also but I should say that mineral oil is there to serve as a, not to evaporate, because this is made up of with a glass or PDMS device, and wherein you will not have any evaporation. You have like a 95 degrees, 65 degree, you have the things, and once it will get amplified and you can collect it, and you have the collecting chip, and you can directly put onto the your depth chip that is called electrochemistry chip. You can put it here and you can identify the reaction, whether amplification has done or not. And of course later on the products have come out from our laboratory and it is commercially available. So, and it is like a screen printing electrode, and you can see what not you want. And the electrochemistry, what we have is that when you have because when you see the DNA, you have the grooves are there like a double helix. When you see that you have ATGC are there, and you have wherein you can use the what I should say that the intercalator. Here we have used the HEXT H33258, and where it will go and bind with the where the double, I mean in the duplex where adenosine thionos 80 places, and where the intercalation occurs, it becomes like an aggregate and the signal comes down. So the signal coming down means in electrochemistry, which means that amplification took place, and you can see the very nicely the, I mean quantitatively you can also detect it, and it has been actually, this has come as a palm-sized device wherein you can put it in your pocket and you can go to the on-site monitoring and you can take the sample and you can do this reaction over there itself. It is available right now from our company that is called Bio Device Technology. Later I will introduce that. Of course, those days I say it is my company, our company, but it's not now because we handed over to the Japanese colleagues, so they are running the company. Okay, and same thing as I showed you, this platform we have used for the small molecule, small chemicals. For example, you have any of the plants or any of the natural or synthetic or the chemistry based synthesis, when you identify, if you want to identify their potential, I mean that is like anti-cancer or any of the effect things that you have to do, we have used these things is that in the beginning. So sorry, and we have put the here, and we have tested for the anti-cancer, means see this one we have tested for the mainly for the carotenoids that we have tested, because this knowledge which I acquired from the CFTRI. So there we used to do many of the bioefficacy, but here I have employed into the where we found what I should say that from the actually bacteria, we identified some of the chemicals, and that happened to be anti-cancers, and we found out some of the leucines and astaxanthins and so on, and different stages, and we have studied metabolic pathway. That is another part of studying which I am not addressing today. And then we identified, and we see the HL60 are like you have the any of the cancer cell lines, either like it is like MCF7 cells, and those cells we also put it by nano dispenser, and you put your targets, and you can put it in the incubator, because since it is a cell based assay, and then we have seen the cell proliferation, how long that it takes, and then after that you can put into the readout, and you see based on the intracellular calcium, and by using the calcium assay, and you can see that whether cells have been proliferated or not, and either has been dead, whether it has been dead, how it has been dead, like it is been like apoptosis or necrosis, and you can do that. And we compared this assay with the regular MTS assay. You all know that is a dehydrogenase, and here we are doing calcineamase. This is mainly for the esterase, and you can see the membrane integrity, which means that cells, whether it has been compromise their life, death, you can identify this way. So, and similarly, we have used the same platform to identify the alternative peptide. Alternative peptide means it is alternative to the nerve growth factor. You all know that nerve growth factors are known for to identify the neurite outgrowth factors in the Alzheimer's and Parkinson's disease, where you have the DOPA, or where you have the degeneration of the neurons takes place in the brain. So therefore you have the Alzheimer's or you have the Parkinson's disease. So, but when you wanted to treat these the patients, there is NGF is there, nerve growth factor, but it is having a very difficult, and you cannot give like IV because it get degraded and it cannot pass through blood-brain barrier. There are many difficulties are there. This study we studied in the 2003, let me tell you that. So then this is one of my PhD student. Just I am showing in this slide, and wherein you have we have made combinatorial peptide library of the pentapeptide. We decided to make the pentapeptide by using a split base synthesis, and then once you make the one bead and split base peptide synthesis, you would not be knowing that what amino acid comes next. So, but you would know that you are sure that all the amino acids we have taken like 5 amino acids mainly for, and then we found out like millions of and hundreds and thousands of peptide beads we have did. And what we did was, and those beads in these chambers we have transferred, and then parallelly on the functionalized glass slide, either you can do the lamine or you can use the collagen coated place, wherein you have put the pheochromocytoma cell, that is a PCT cells, and we made like a smear like this, and we combined these things like this device, and we put into the incubator, because the reaction wherever you don't know that which bead contains what pentapeptide. Okay, and then we allowed it for a couple of hours, and then we realized, since we have this is a glass place, we can see through microscopically, wherever the cells has a close proximity with the peptide bead, the bead on the peptides, which has like a billion, huge concentration of the peptides are there on the bead, and we found out few chambers wherein you can see the neurite outgrowth, but not all the things. So then we wanted to know what those peptides, and which means that these are the peptides which can induce neurites. So, and then we identified what are those peptides, and those peptides were taken do the sequencing we have done from the beads, and then we identified what their sequence, and we made the couple of various kinds of peptides, and that I am telling this, though it is been patented, and we have already made some small technology also. And finally we found that methane and valine isoleucine phenylalanine found to be the best among the 4 peptides that we found out. And then we identified wherein you can see the neurite outgrowth are there, and then those beads can be taken out. We subjected to amino acid, and we found out these are the things. And then we also to test whether it is truly, either it is the bead, like sometimes you have the surface chemistry also gives you some of the results. So then we to control, we made the control peptide like a valine like that, and then we have tried, and NGF is a positive control, and we have the non-treated, and we allowed these things in the petri dishes, 96 well plates, and we see that how the growth have taken place. And then where we found that you have the nerve growth factors, of course, because the NGF is 122 amino acids, wherein you have the TRK, ERK pathways there that I'm going to tell you later. So then we found out that how actually the identified peptide can induce the nerve growth factors. And then we were actually skeptical to see that whether it is those nerve growth factors, are they truly functional, are they releasing DOPA or not. And then we also quantified the dopamine levels in these actually neurite outgrowth that has been come upon the treatment of the MIBF peptide, and then we compared with NGF. Obviously, it will not to the level of NGF because it's a pentapeptide. NGF is 122 amino acids. Then we saw that how actually pentapeptide can go, and its signaling we are interested. And you all know that this is a NGF means it goes into TRK, ERK signaling pathway, and you have much more reactions. Of course, those days it is not known. Now most of the pathways, many of the secrets has been unraveled. Okay, and then what we did was that whether it is a TRK mediated or ERK, and then we actually inhibited the TRK by using its inhibitor K252A, and then we see that how actually the neurite has been stopped, and then we coined the pathway how it has been compared with the with NGF. And then it's also followed the neurite outgrowth inhibition by the TRK pathway, and then after that we also compared with the ERK cascade also by using its inhibitors, and we ensured that it is TRK, ERK, and that was actually. But otherwise, if you have the peptides and if you have the millions of peptides, I mean peptide beads, how would you able to do the conventional based? You cannot. We cannot able to identify, but using this, aiding this platform, this setup, we could able to do and identify, and like this very micro devices can apply for various things. So now you have seen, I'm not going to other details, and you have seen the how actually DNA can be used, the small molecules can be screened and efficiently also for the identify the nerve growth factor by the peptides also, and their applications you have seen that. But from now onward, the abstract what I shared is mainly for the single cell screening. So how we have used is that because this is the what wherein it can everything decides the reactions, because what we have is the primitive, that is like you're seeing the DNA or the protein or you're seeing the peptide and the reaction small molecules in that. But if you see the whole cell, because if you see the single cell, why single cell is truly important means always we do reactions when you go to the clinic or hospital, they take your blood and they do the assay and they say that so and so you have or you have not. So, but most of the diseases that you have, like hepatitis B or any of the diseases that you get, is that you have like those positive cells in your body in the peripheral blood. You have about 10 power of 5. You have 1 or none. So therefore, when we targeted that, and how do we develop this aiding these devices? And that's why my talk I said is that mainly I use microchambers for high throughput antigen specific single cell analysis, and how actually I use microfluidics, another platform for the single cell screening. Actually, these two researchers have led to the company of the SC World. SC World stands for Single Cell World, and this actually has led to the, you may call it as a spin-off, you may call it venture business laboratory, you may call it startup. But that has actually, the startup has led to the worthwhile business, and actually this was taken over by the, we can develop for the patient specific, not only disease diagnosis or the prognosis, also for the monoclonal antibody. For him also we have done the T cells also. To begin with, we tested on the B lymphocytes, and then we can make the antibodies based on the information, the disease that we can get from this. And later I also see, because this you will see on the cell surface markers, and later we have tested for the, because when you take any of the cell component, you have the intracellular also. So many markers are there. If you wanted to do for the intracellular markers, you will have to sacrifice the cell, so that you either you have to do like any of the histochemical or the fluorescence staining, so that the life of the stage, you identify some results, there it ends. But we wanted to use based on the intracellular based, where can we able to develop any of the cell sorter or in the microfluidic outside, and how actually the current like a flow cytometers have a difficulty. So those things we have seen that in the couple of slides you will be going to see these are things. As I told you that I'm not going to details, as I already told you that, because when the cells are having a heterogeneity, even if you take from the same B lymphocytes, it has like a various levels of B lymphocytes or T lymphocytes, or any of the cell population never be the same. Though it is the same cell type, the cell types are always different, because they are at the different levels and they have the different targets. So therefore, if you do the assays collectively, you would not be able to address, or you would not be able to hit the gold mine or the things that you wanted to unravel the secrets, you will not be able to identify. So therefore, there is a great need is that when the disease manifestation or things comes from the single rare number of cells which are there, you know that is called circular tumor cells, CTC, in the body. So there are many circular tumor cells are going in the blood, going on, because that is actually is a tumor cells which come from there, and it will go and it's going to another place, and it will go into the bind another organ. There you have a metastasis. So actually CTC, after this research, my colleague was working on the CTC, circular tumor cells, using that, and we have done a huge, the leaps and bounds of things have come. So therefore, in CTC, you will have 10 power of 8 cells. For about 1 crore or 10 millions, more than that cells, you have 1 or none. And so when you have this kind of low frequency cells which are a target for your things, how would you able to identify them, to identify the disease or prognosis, or to identify any other things? For example, as I told you, hepatitis B and hepatitis C, you have like a very less number of things. That was our first targets when we started this project, and it is funded by the Mon Bouchon. And it's about 25 billion yen program, not the, of course, dollars, 25 billion yen, which means 2.5 billion US dollars. And we have got in that, actually, we have developed various kinds of tools, and how actually these tools can be used for the development of the identifying the disease diagnosis and single cell analysis and so on. And when we started this research, there are no tools, there are no cameras, there are no manipulators. So we had to collaborate with many of the companies like Hitachi, Toshiba, and many of the the companies which can pick up the cells, and we developed many of the, like in one go, CCD, CMOS cameras, and many of the tools which we have developed. That's why this whole technology has led to the establishment of a company, and that company has finally taken over. By that, you will see in the next slides. And this is to show in a nutshell here. This is like about how we screen the high throughput way of the antigen specific single B lymphocytes. Here it shows that. And you, for example, you have mouse, or you have mice, or you have the human blood, anything you have that. And our target is to be in the beginning, it is for us like a spleen, or where you have the source of B lymphocytes, either thymus, you have the T cells, you collect the cells and you take them, and you, of course, you have to little purification has to be done by using, and you have the, of course, you have a blood. If you treat with ammonium chloride I forgot that one concentration, and you have the rupture of the blood cell happens, and you have the BNT is there, and once you have BNT, you have almost like a syringe type columns, or any of the, you just put it there, you have the B lymphocytes comes out. So there, that is serves as there for us a source of investigation. Once you have a B lymphocytes, what we did is that we have made the micro chamber array, which this chamber array is totally different from the other, which what I said it, because there we have here, it can accommodate only one cell at a time. It cannot be done 2 cells. So for that, we have, this is about this slide that you are going to see here. One square, this is slide glass. It is like it's a plastic, and this device comes for about 10 rupees or $1. So of course that is like it just become a technology. Now we realized that in the beginning when we started doing that, we tried for various kinds of substrates, and it is like it's a polypropylene actually, and it is like it doesn't have any autofluorescence, because when we are dealing with this fluorescence analysis, you have to ensure that your substrate should not have any autofluorescence. And of course those things parallel we did it, and once you have that, and once you have the cells are there, and after that and encapsulation in the chambers attached, and you can treat, because assume that you have taken from a patient and you know the patient information, and you can you can treat with the suspected disease of the causative agent, and you can identify the cells which in this population, the patient specific cells are there. You can identify how many times these fluorescence will go increase. That how we do? It is based on the intracellular calcium. Intracellular calcium, how we have done with various kinds of calcium indicator, that is called fluo4, and it will actually do about the excitation and emission. You can beautifully see this as you are seeing slides, and you can see in subsequent slides also. And by using a scattered plot, you can see the what is the population of, you have 10 times or 5 times of population increase, and you can identify. You will not be knowing at this stage, are they really that? And by analyzing these things, you will know that the stage of the disease, whether you have First, you can identify the so-and-so, whether he has or not. You can use this information for cell diagnosis, and this information you can take out later on your target cells, what you identify, and you can go for RT-PCR, because when you take a single B cell, your DNA is very less. Whereas an RNA, you have RNA is 20 times more. And so therefore, we do the RT-PCR, and then you can identify the sequence and build the antibody. Okay, that you are going to see that. Sorry. Oh, where is that, my slides? Okay. And to do that, and this is, you have the slide, don't worry that we have used, this is called Liga, and this is the process wherein we can develop our devices, and we have developed by using this this process. And you have the, like this device, you have got it here, where you can see here, and it is about having about 200,000 chambers, and each chamber is demarcated, and it is been clustered, conglomerated. You can know which chamber has while you are scanning that. And then you have each one, and it can go and accommodate a single cell. There is no chance of two cells, because B lymphocytes is about 8 to 10 micrometer in size. So, and it can accommodate, and we have tested that. And then we have seen the, don't go into details, and we have optimized the substrate and what is the, treatment wherein you have best, wherein encapsulated cells can go and accommodate in the chambers. And we have seen all the things, and these are the places you can see. I hope you are seeing on the screen. And here you have the, where the cells have been in the each chambers are there. And once you have that, each block is about, you have in each block 900 chambers are, it has 32 blocks are there, you have about this many chambers are there. And we found that single cell encapsulation in this, it will go based on the simple gravitational force, and it's over 80%. And of course later on we increased. And once you have, I'm showing you in each single block, once you have that, you just add by your pipette and the target that you are suspecting. And as we target the calcium, the intracellular calcium goes, wherein you have those cells, you have that, and with that the titers will increase. By using that, can be done by scanner, where we have the Hitachi soft we have developed with our collaboration. The tool has come, so there you can put it and you can scan, all the messages of the things will come on the screen, and you can know which cells. You can just click on that. When you are analyzing, you will know that so and so place you have so and so, and this you will see as a scattered plot, and how many times you have. These are the population, it doesn't have reacted, and where you have the red population is a 5 times, and the blue color dots are there, you have 10 times positive. And when you identify that, total about this, out of 32,000 plus cells, we identified 0.29% of the cells have data suggested 10 times activity, which means that you can identify these are the supposed to be. At this juncture, you will not be knowing that are they truly causative or not. That we have investigated further, and it is much more 10 times. And those cells which you get limited population of cells, and then we identify and we take them out. This is what the data shows, that 0.29% of the cells. And then after that, we take them out by using the micro manipulator. And of course, the manipulator also we developed later on. In the beginning, we didn't have the efficient one. And then we after retrieval, we have developed their sequence, and we built the heavy chains and light chains, and then and so on. And finally, you will have the monoclonal antibody cDNA analysis was done, and then which, of course, I am showing a part of things later on. And we have tested these antibodies, so-called, are they we tested back with those antigens when we tested them, and then we identified these are the things for the causative, and these are the disease at which the status are there. Otherwise, if you don't do by this kind of analysis, you will always get is that negative. But the time you will become a truly at the metastatic level, 4th stage or 3rd stage, you will say that we are not able to save the patient or save the lives. So that's why these kind of things, and later on we have used for the various other assays. Yes, obviously these results will come into various kinds of publication. I'm not showing all. We published in Nature Medicine and analytical chemistry and many other things like cytometry. Many other journals we have published from these papers, and also we have patented this technology. Obviously, that's why we got into a company and so on. And this is later that what you have seen is the micro chamber array. But what we realized there, when you wanted to do the PCR in the same chamber, if you see here, and what we did is that we have retrieved from each chamber and we did the PCR outside. So we realized that why don't we develop a platform wherein there itself we can do the PCR. So that's why we thought that why don't we develop another platform that is called microfluidics. It is a micro channels. Micro is a single channel, and you can put it to the cell suspension, and which can be bifurcated into small drops by using a two-phase, and then there the rest of the story. And then we have used in one small channel, you can do hundreds and thousands of samples can be segregated and identified, and you can screen it, and then you can also do the PCR, and you can collect the DNA out, and you can do the further analysis. That is called, because this is actually impossible at the micro chamber level those days. Okay, the knowledge that we have those days. What we did is that here, this is called, I say that it is like a Y-shaped micro channel. This is just cartoon, I'm showing the illustration. And wherein what we have done is that we have put from here the suspension, cell suspension, the target B lymphocytes, and here this is like aqueous, and it has been labeled with the already the calcium indicator already in the beginning, because, and you have the oil. This oil we have used the mineral oil. I will show you some of the movies in the next slide. You will understand when you introduce together at the same time, and these things, because of the surface tension, viscosity, and interfacial tension between at the micron level, the micron size, because these are all 30 microns, 15 micrometer width and height. These are the depth of the channels. So what you will get is that you will get beautifully liquid oil, liquid oil. Like you will get is the compartments. You will get the drops. So by manipulating the these your flow rates of the liquids, you can ensure that a single cell be encapsulated in each drop. That is the beauty of the system. Though it is a single channel, you can do that. By doing so, you know that because it's already been treated with calcium indicator, so you know that each one is a fluoresce. But these fluoresce is various, because you would not be, you have your cells are there. So when you are aiming for the antigen, that you are searching for antigen specific cells, that antigen in between you can introduce into the channel, so that will go and react with your cells, aqueous chambers. So then you can identify wherever you have the positive cell is there, that calcium efflorescence increases several times. So
Sir,
yes.
Sir, excuse me, sir, your slides are not visible.
From when, madam? From when? I'm sorry.
Since 10 minutes back.
No. 3 slides back, sir. 3 minutes back, probably.
Oh, sorry.
You explaining video. We thought your video will come. That's what we're waiting.
Okay. Am I? So hope you. That is the. If you miss the beauty. Did you understand this? Did you see this one? No.
No.
Yes, sir. Visible. Now it is visible.
I will tell you that. So this is micro chamber. Once you do the reaction, where the calcium will go, and whenever the slides see, though we are in the 21st century, the electronics, the difficulty that we are seeing, still we are facing that. Okay, please do remind me, because I would not be knowing that whether you are listening or not. Please remind me. Otherwise, if you don't see the slides, I am 100% sure that you will not get it anything. That's what I just wanted to remind you. Okay, and you see that once you have the specific cells, after that you will be scattered analysis. You will identify the 5 times and 10 times of the cells. And then once you have that, because when you see that, you will find out the less than 0.5% specific cells you identify out of, just to show that, 0.29%, those cells, since we cannot do the PCR in the same chamber, because the chamber is only for the one cell it can accommodate. So then what we did was that we took out the target cells, and we did the RT-PCR, because as I told you, hope you understand, because each cell has having a less amount of DNA, and then you have the RT-PCR we done, and then after finding out that we found out like a heavy chain, L chain, and we did the DNA sequencing, and we constructed the monoclonal antibody, and then we tested its specificity against the in vitro for that antigen, and then we identified this could be the target, and we developed the antibody, and we tested the patients and so on, not the patients at the in vitro level, and now government of Japan has given permission to do that. Okay, when we wanted to test on the human beings, it's not that easy, so we have to undergo for many things are there. And like finally you will get the antibody cDNA analysis we have done, and we constructed the things, and then we actually tried the things of, as I told you, we published couple of papers comes, and since we have a limitation in the the PCR we cannot do in the micro chambers, we thought that we have to develop the microfluidics. Microfluidics is the one. I hope you are seeing the slide right now, right? So you have the microchannel is there, and once you have the microchannel here, you will put the B lymphocytes, and you will put the here mineral oil, because the both are the two different phases. They will never get mixed up. So by using this phenomena, we have used this microfluidics and surface, that interfacial tension and this viscosity, all the things. When you introduce together, you will have not the laminar flow. You will have a slug flow. You can have a plug flow. You call it as, that is, you call as a water oil, you will have a slugs. You will have a slugs, and you will have a continuous flow. So though it is a single channel, you can make number of cells can be sorted, and you can identify because these cells already been treated with the calcium cell indicator. Am I audible?
Yes, sir. Audible, sir.
There is a noise is coming from others. One can mute those sides so that the disturbance will not be there, right? So once you have that, and once you have the cells are there, you can same thing, or one of the another inlet, you can treat with the antigen that you are suspecting, what you are looking for the disease specific cells, and you can identify those cells in those population, 10 power of 6 or 10 power of 7 B lymphocytes are there. You will find out only few cells, the fluorescence increases, that is intracellular calcium increases. So therefore, you can identify those cells on the chip, before and after, which is not possible by flow cytometer. No, impossible. So, and fax it cannot be done. So that's why we thought of this kind of platform. So because therefore, and in the same device, you can also introduce the cell lysis reagent, and you can do the PCR. And you can do the PCR, and you can collect the DNA. Of course, we have done up to the lysis level. PCR, we have done the same platform separately, because we had a difficulty doing that. So this is because in a single chip, you can do this, you can do the sorting, you can do the screening, you can do the analysis, you can do the lysis, you can do the PCR. That is, we call this device is called as a lab on a chip. You can do all the laboratory reactions on a chip. So this is, you might have heard, we have also, I have published a couple of papers in the lab on a chip journal. So it is also highly impacted journal, and that is we call as a micro total analysis system. So that's why most of our papers comes under the microtask journals. Microtask means micro total analysis. Many modules of the assay you can do on this simple device, which is of your palm size. And of course, the microscopic setup is there. Other than that, you can actually take your chip in your pocket, and you can actually move on. So of course, this is the things, and the reaction, as I told you earlier, intracellular calcium, and then you identify based on the increase in the calcium level, and mainly for the calcium signaling. And here we have done the single cell screening, sorting, and PCR we have done. So, and to do that, don't go into details, and we have used here, unlike silicon, we have used like one polymer that is called PDMS. PDMS means polydimethyl siloxane. It is like two monomers are there. Once you add these things together, and once you treat with about 80 degrees or 100 degrees, they get polymerized. So that becomes very transparent, and it is not so expensive. I'm not saying that it is not cheap, but it's not so expensive. One can bear any of the research laboratory, and then immediately you can. So how do you do? We use this material. We actually did whatever the pattern that you want. Either you can do by Adobe Illustrator or the CAD. the CAD software, you can do the micro channels, all the devices, everything. And once you put on that, you have the platform. You can use the photoresist. Like olden days, we used to, nowadays all the digital has come. Earlier days, we used to take a film camera and then we give it for film development in the black room. Similar principle here, but high precision. Okay, and since we do the photolithography and the same pattern can be printed there, as you see the channel here, and you can do the what angle, what depth you want, all the things. That is called by using this is what the steps are there. That is the negative MOS called SU8. SU8 is the SU8. We really don't know what XU8 because it's like a trade secret, not by my trade secret. It is a trade secret of all over the world. So then we actually develop it and then we make it, and then we will put the ports, inlets and outlets by using a tubes. Then it will serve as a chip. So here, as you've seen that next, maybe next one I show you. Okay, here is there. You have the PDMS and we put the tubes, and you have the pump if you put it, and you will see that. And this is the platform. Next, you will see the video. You will understand that. You have the syringe pump is there, and you are connected to oil and liquid to see the principle, and you can see the CCD camera is there, and you can identify the things. This is the setup. It is like a Olympus inverted microscope where you have the micro syringe pumps are there, and you will see the things I'm going to show here. Okay, and you see here. I hope you are seeing that. So you have the from here. You have the one which is seeing it is a liquid, and here ones which are not there, it's oil. So, and we are seeing that you have the liquid oil coming here, and this is the zoom. Okay, this is the zoom wherein you have the cross section where you have the Y joint or these actually Y are 120 degree, 90 degree, 60 degree. We all optimize these things when we develop the things. So this is the place where you will get the see here the liquid and oil, like this what you have seen in my illustration. So like this you have on single channel you have hundreds and thousands of cells can be separated. That's what I wanted to say. And since it is been flowing reaction, and it is like not only the velocity and this fluid dynamics in the each one is very beautiful. It is like a chaotic mixing is there, which you will see briefly. I'm not going to fluid dynamics. So, and that is the why you can do the reactions takes place in the millisecond rate. In this microsecond, millisecond rate, the reactions takes place. The beauty of the system. Okay, and then of course don't go into details. We have isolated and we have characterized the device, blah blah blah, all the things, and we identified that at what level you can make it to the picoliter chambers can be made, which means that a single cell is there in a small reaction chamber, one drop. When you do the lysis, when you do the PCR, the dilution will never hamper your assay. That's what I want to address. Okay, and then we have seen that, and in the beginning to do that, we have seen that when you have so for example, when you have aqueous, a lot of beads reaction, whether these phenomena exist or it will rupture. So we have identified this is like a fluorescence beads we have done, and when you see that it is been happening, and also we have seen before going to cell, I tested that if you see, because I have used 6 micron FITC beads, the fluorescence beads. So it is a model to the B lymphocyte. Lymphocyte is also 6 to 8 micrometers. So I thought that once you do here, as you are seeing here, you have the loop is broken. So you have each chamber. Some chambers you have one, and some chambers you have none. Yes, it will be there. For that, how to improve, we have done that. Okay, and we have tested how the angle, whether it's a depth, it is a height, everything we worked out and we have tested that. Okay, and then when you have the sample is in one row, this is the way beads goes into this is the aqueous, and you have oil, then it will form one. And when you have the more, you have got the more in one in droplet, you will have the more cells. That's why we optimize which size of the sample inlet, cell inlet is the best we optimized. Okay, and then we have tested the cell adhesion. Some of the anything blacks, the cells get adhesion. Then we have identified some of the thing that wherein it will make that cells will not attach to the PDMS and glass chip. It will not attach, and so that we have done. And this is the B lymphocyte, which you can see that. This is the cell. and you have the cell. And the cell, some places you are seeing that it doesn't have. No, the cell is there because it's about 30 micron height. So you have the cells can be in any height of the drop. So that's why we have used special system that is called confocal microscopy. So there itself, high speed camera, that is about 25 millions. Hence it was the equipment. So that I made with another company. We have developed the system. When we started this project, the technology is not there and we had to start. That actually it was tough and it has become a boon to us because the whole technology is ours. Because there is know-how, not only know-how, it is no tools also available. So we could develop all those tools and you can identify each cell when it is processing that, you can see its signal. Okay, like that you can see. So we identified that how many single cells are there, how many two cells are there. Some chambers we find out that there are two cells. How can we avoid that the cell camera, all quantifications, everything has been done. So, and I told you in the beginning that yes, you identified cells are encapsulated in a chamber. And then how do you do? While they are going in a device, how do you test? By using another reagent, that is your antigen. So it should go and bind, it should go and mix with only the aqueous droplet. So there only you have cell. So that we have done again here we have done this by using this kind of device. And this is the real device, and here you can see that, maybe where is that? Okay, here it is the device wherein you have, and here you have Y shape 120-degree angle, and here you have things. I am going to show you the movie. So here I have tested to show the proof of principle. Sorry, to show the proof of principle is that you have here the red one is neutral red aqueous reagent. Assume that it is a cell. Okay, the one which doesn't have, it's an oil. So assume that you have a cell over there, whether your green, that is like a methyl green, I have put the indicator, assume that it is antigen, the target that you wanted to see, whether those cells are specific cells are there in that or not. Cell is there. So interestingly, this will go and mix with only the aqueous one. It will never go and form because if it goes and mix with the oil, it will form only green one, right? So these are the fluid dynamics, all the things, and this angle, everything, this diameter, everything we have optimized at the minute level. And though it looks simple, but it is not that simple because we toiled in the clean rooms. These are all cannot be done in the normal rooms. It has to be done in the clean room facility. So there we used to have a special attire, and then once we go and you have all the things and so on, and you have that. So you have beautifully, it will go in that. And this is the things what you see in one go. You can see the cell, which is like a before reaction and after reaction. Then we realized in the beginning that this thing is quite far away. Then we have remade it. This is I took the screenshots. Where is that? And then we found out that this dynamics in each chamber, this is like aqueous. This is oil. This is oil. As you are seeing that the dynamics are vortex mixing, like a double vortex. When you do vortex this way and the same way also it does this way. So like this. Okay, like this it does. Like what I should say that you have the water tank is there. It goes on the belt and it progresses and the belt reverses, right? It progresses, it belt reverses, same dynamics. So that is called double vortexing is there. As you see the arrow marks are there. So therefore you have the reaction takes place. That for that we have done the dynamics using microparticle image velocimetry. This is called vector plots, wherein you can see where the speed, the red one shows that the speed at which the mixing is higher, the blue is the less at the walls places, and the center which is in yellow color. So wherein you have the mixing capacity and it is a mixing dynamics. Because to understand the phenomena, you need to do that. Once we understand, what we did was, before going to this thing, we thought that let us try any of the bioassay to see that whether it is happening or not. What I did was the simple conventional what you do the laboratory, the beta galactose assay. If you use the FDG, that is the substrate, we use the fluorescent one, fluorescein dibeta galactose dyes. It is like it has a 2 galactose will be there, and you have a galactose dyes, and it will cleave one, next it will cleave other side. So then fluorescence releases. It will become fluorescein, it will, right? The fluorescein moiety will come, it will. Then I thought that I tested on the 5 to 10 microliters in small Pendorf tube, and then I see that its fluorescence with the time 180 seconds you got. Then I compared with their quantity. Then I see that let me try on the device if the reaction is taking place such a quickly. Yes, the fluorescence should come because this reaction should take place in no time. So that's what we have realized this, and this is the screenshot. I'm going to show the video. I hope you will be able to watch the fluorescence that comes because it is a small volume. The fluorescence do comes. Okay, let me try that video. And because see, this is the video, and assume that you are getting it here. The cells, the beta galactosidase substrate. From here, you are getting the enzyme. Okay. And no, sorry, here this is the enzyme, this is the substrate is there, and then wherever it goes and mixes in time less than few seconds, you should get the fluorescence. Okay, you will see that, and it will switch over to the fluorescence now. I think we have identified this one. I hope you will, because I don't know whether the room is bright enough, you will not be. Already it started forming here. This is the place where you can see that. Okay, the fluorescence. So we have seen at the thing, and if you go a little further, you have like this. So this is what I showed in the here, the fluorescence, and then we quantified that, and how it is very, very different from the bulk to chip. And I hope I have a time because I am running short of time. And then identified, then bulk and chip. Then what is the reason is that it is because of this fluid dynamics, the reaction takes place, and then that is what we said. And then we found out actually those days there was no CMOS cameras which can be do high sensitivity single cell imaging was there in 2008. This year also we didn't have. So I had to do for many companies for this thing, and finally that year we got from the Zeiss, Carl Zeiss, Axiocom. So they have the high sensitivity camera with the time limitation. They have without time limitation they have done. So this is called chip. It looks this is the chip. PDMS. It is like a transparent, and it is like a, since we are seeing the glass, we have used this cover glass thickness down because we are using on the inverted microscope, wherein you can see from the bottom and you can see the cells. And once you have the cell here, it is there is a screenshot. You will not be able to identify because it goes such a fast. That's why you see the cell as a streak, because the speed at which the cells goes in that it is faster than the high sensitivity camera that we had those days. Okay, and then you identify the same cell because we can track where the same cell and the same cell, so you can, because the system has been developed like that so that we would not miss this is the specific cell or not. So then positive cell, when it comes, the signal goes. In the beginning, signal is like this here, then after that here signal is like this. Like this we identified, just this is like a proof of study. We have done for many cells, and when you have that here, as you have seen in the micro chamber array, the specific cells you have done 10 times and 5 times, and the cells which do not have responded. And we also have used the control is that PBS. If you do the PBS in place of the your antigen, whether the cells reacts or not, also we tested. So that because I mean that control, we have used the PBS in place of anything, and then we identified. And obviously with this system we could establish a company that is called Bio Device Technology. Sorry, it is in Japanese, don't worry. Okay, Bio Device Technology. And then from this technology we have had, this is the pamphlet in Japanese, and from that we together there is a called SC World. This is called single cell world. SC World means single cell world, and it is the thing, it has been there in the Toyama. And because I was there in Ishikawa Prefecture that time in Japan, because I was affiliated to Japan Advanced Institute of Science and Technology, and this project was given to us for the Toyama Prefecture and Ishikawa Prefecture to our institute and Toyama University with the medical doctors. And because as we are getting from the patient specific, all the cancer related things are the cells, so we have collaborated on this one. And this technology has been taken over by the Vivalis. As in my introduction you have heard, I think I have not put the Vivalis, and then later on I heard the Vivalis actually they bought for 30 million or something, 30 or 40 million euros. And this later on I came to know when I was checking a couple of days back, it was taken over by the Sanofi Patcher they have taken over by this thing for further collaborations. Once you give, and in Japan also we have the Vivalis Toyama still is there, and it is flourishing and they are producing. And yesterday night I have seen that Vivalis is also producing, that is called as a Valvia or something name is there, and they are also developing for the COVID-19 vaccines, which I yesterday have seen, and many progress that they are doing yesterday night I have seen. And now you have seen that how this microfluidics and micro chamber platforms you can do for the single cell analysis, so as to identify the disease diagnosis and to make the antibody. Also how microfluidics you can do on a same platform, very many applications, and how it has been led to the know-how, and it has been to the commercialized technology. And that is mainly based on the, you have seen the cell surface markers. Obviously you have IgG and IgM, you have about the cell surface. But we thought that, of course, that was project, and we were thinking that because of intracellular, of course, this is the place where I have actually moved to another organization. Also, it is a government only. And then we were thinking that why don't we develop intracellular based? There are many markers are there, but non-invasive, but the same cell has to be used for further analysis. So our target is that, because you have the, in 2008, 2007, you all know that in place of embryonic stem cells, Professor Yamanaka from Kyoto University, he developed by using like IPS cells, that is induced pluripotent stem cells, and from the skin fibroblast. It is not the, because of ethical issues, he taken over and he put the ACT4, SAS, and 4 transcription factors, and he ensured that he developed the fibroblast into a stem cells like cells, which is same as stem cells. That is called induced pluripotency, has induced pluripotency in the fibroblast, I mean normal cells. So that's what it is, stem cells. So that was, it was actually, you all know that in 2000, in 6 years, he has got a Nobel Prize in medicine, right? So I had a lucky to work with him, with his cells, while I moved from to Osaka University, where I was in the applied physics department, because our group, all our group, we department moved from the Japan Advanced Science and Technology to the Osaka University. There I have developed using the, how actually IPS cells, the undifferentiated cells, when you do the differentiated, undifferentiated cells, these things can be separated based on the device that I have developed already earlier, single cell technology. So then I had collaboration with the cardiovascular doctors in the Osaka University Hospital. They have collaboration with the Professor Yamanaka, Kyoto University. So therefore, the cells for that he has got Nobel Prize. Those cells I had worked and developed some of the small molecules and made those small molecules into neuronal cells and the cardiomyocytes. So when I was doing these things, the differentiation never be 100% understood, because that is about to, luckily we have got 76 to 84% we have got in my, of course it has gone couple, I'm not presenting those details. So
sir,
I'm, yes.
Slides are not moving, number one, sir. Second thing, your slides are not moving.
Now?
Yeah, it's visible, sir. It's visible, yeah.
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 Is it moving now or not?
Yeah, it's moving.
Okay, I'm explaining in between. I understand that. So therefore, I thought that when I was doing that, I should develop a platform wherein I should not sacrifice these cells that I have developed for the patients. That is cell-based therapy. What you have seen earlier is another one, and these cells itself you can use for the treatment of either any of the things like cells for the patients. So I thought that we should have to develop something. That time, the other group, when I gave a couple of presentations here and there in all over Japan, and some team said that we need a group for collaboration to have an experts to develop some nano needles. Nano needles are the high aspect ratio. They are at the when you pierce. I am saying non-invasive. I am saying at the same time pierce. No, because once you pierce, these order of the nano needles are the nanopore order. So what you are puncturing, it is not truly invasive. It is a minimally invasive, truly scientific manner. But the cell repairs in no time. Okay, so that we have, then we thought that why don't we target intracellular, many markers are there. So those things, by using this nano needle, and you can identify the cells, whether they are, yes, whether it is like undifferentiated, like stem cells, or at the already terminal cells, like anything. Because once you start beating, you will know the cardiomyocytes. But the cardiomyocytes, once you treat them, they will be in spherical shape only. But they start beating also, right? That we have done. Electrophysiology, everything that is like a cardio chips, we have developed. That has been done, and we have found out some of the drugs, all those things. That is another story. But I wanted to see the same cells. How can I identify, remove the undifferentiated from the targets? I mean that your targets are either neuronal or from the cardiomyocytes. That was my target. So therefore, we thought that here we aimed it, as you know, that iPS cells are come, and these cells, once you have a specialized cells, either you can use it for the cell transplantation, that is cell-based therapy, or these cells you can use it for the special drugs, because you treat in the cell level of the patient specific, and then you develop a drugs, a patient specific drugs, right? So like this we have done, but how to do that? And we want it to be high throughput, it should be rapid, and it should be non-invasive, because we wanted to use the same cell after identifying for the treatment. We don't want to terminally we identified, not that we sacrifice them. No, we wanted to use that. So that's why we have tested. This is like a mechanic, we call it as a cell sorting based on the regenerative application. And I am not going to that, because you know that facts I have already said earlier, because it can do only the extracellular, intracellular it cannot be done. So therefore we have tested like as a model system. In the beginning we have taken nestin. Nestin is an intracellular filamental protein. So then we targeted the neurofilament. If once you have the neuron, you have the neuroproteins, and you have the undifferentiated stem cells, you have the nestin. So to begin with, we targeted that. And then how do we do that? As I told you that you need to have a platform. So this group, actually what they have asked for the collaboration, they have the group, this was work before I started a collaboration with them. What they did is that you have the cantilever, you know, the atomic force microscope, and you have the cantilever. The cantilevers are that, and by using this cantilever, using like a, that beam etching, you can cut it. Because initially this will be like this, and he cut it, and he will make the needle, and they have tested, and it has come in the nano letters. It has covered in the BBC and so on. They have done quite a bit of research how you can do the intracellular mRNA detection can be done, molecular beacon based. Either you can do all the DNA delivery. They have done. What the thing is that what they have tested is that they have done good progress. They have done, but they wanted to do, but it is only one at a time. One at a time. How can you do for the your proposal of the cell-based therapy to identify the target cells? So you need high throughput, but the same technology. So that was the target. So then they asked me, can we have this thing? I said, yes, in principle, it is easy, it is possible, but we have to work it out. So, and what we did is that nano needle fabrication, this is like what you have seen micro, this is another level of the another level of this is also you need all the clean rooms, and of course wherever you are seeing all are being protected by the patents, as you see that, and we developed by using that, and you can see all it will go many things like a development photolithography, deep reactive ion etching, and things, and you will have here you are seeing that it is when it is in a micron level, this much is the 20 microns length, and you have that, and finally you have like a single new nano needle or the triplets, like a triple nano needle we have done, because as we have to penetrate into one cell and to see those cells, based on that we wanted to lift the target cells, but the same cell we wanted to use for the further treatment also. So that's why we have seen, as you have seen the beautiful nano needles you have seen here, and it has undergone, and we have toiled, of course. It is not that 1 day or 2 days. It took 1.5 years to develop this platform, but not by going to hi-fi, but is a low-level technology. We have demonstrated to the world and how simply it can be done, this technology for that, to make that. Of course, to make that, it is not that easy, and these are the things you have in the clean room equipment, where you can see the yellow rooms are there with lithography, and the white rooms are there, you can do the wet research. So that's why you have the lights variation, you have the spin coater, And hope you are seeing the slides. Are you seeing the slides, please?
Yes, sir. Yes, sir. Visible, sir.
This is like aligner, and you have the, this is called deep etcher, dry etching, which is, it runs in several crores, don't mind, because this is high technology. And you have the field emission scanning electron microscopy. Anything you want to see that you can't visualize by naked eye, you have to see the scanning. And this is like, if you wanted to treat the surface with oxygen plasma, it is called oxygen plasma equipment. And then it is for the, if you wanted to make the silicon wafer into oxidizing, hydrophilic, you have to treat with the oxidation. Oxidation means it will go into 1200 to 1500 level temperature. Systematically, it will do that. So this is the one. And then, of course, we have used like an ultra temper and so on, and like this. And then we found out once you have that, for example, yes.
Sir, actually, we have to little bit, because next speaker is lined up from Boston, sir. So please.
Sorry. Can you give me
exciting talk, but yes, please.
5 minutes? Can you give me 5 minutes? Is okay.
Kindly, sir, please.
Okay, so then we developed it. Once you have that, and we diced each device by using like a laser stealth dicer, you will get the device. And to operation, we have developed one more that is like a manipulator, where the device that you have developed, and where this is with the another university collaboration. And here wherein you will put the nano needle array, which is about 0.5 centimeter, and it has having about hundreds and thousands of nano needles are there. And then like this fashion, and we use on the petri dishes, and how you can introduce into the chambers. And in the beginning, we have used to test whether it is in vitro. And we have also seen that by using confocal laser scanning microscopy, and whether the cells are, whether nanoneedles are going into the cells, wherein you can see here on the screen. And of course, we have various kinds of things. And finally, we have seen how it can be plasmid, can be introduced in the cells, and the efficiency, and like a real-time fashion, how actually the compare with the liposuction nanoneedle one, how the relative fluorescence will be increased without compromising the things. Here we have used for the PCS2 Venus, the plasmids that we have used in these cells. And at the end, we have also tried for the CRE recombinase, wherein we have, this is like a proteins also we can introduce by using this platform. And this is the where we have tested this micro nanoneedle array for the delivery of the micromolecule, that macromolecules without compromising the cells. And then we have used the same technology for using the separated cells, and by optimizing the adhesion force and antibodies on the nano needle, you can easily separate the things lift by using these are confocal, how the antibody, this thing. And this is the way wherein you have the cells are there on the platform, and where you have the targets for the nano needles, and it will go on multiple times, it will penetrate, and wherever they have intracellular proteins are there, their affinity, and it will go and they lift, because the adhesion force is lesser than the affinity of the force. And then we transfer into another platform where you have seen here, and another plate, and we assess and we collect the cells, and we estimated like what are the forces you need. And the same thing, we have used the Micron platform to stamp single cell on the place. We have developed one biocompatible membrane, and wherein you can put and you can stamp on the petri dish, and you put the cells over it, and you wash it, only the cells will come. Wherever the needles are there, cell will go one cell and one needle, and at the same place we have done. And then I use for the separation of specific cells using an nestin positive. Yes, and we have succeeded, but the efficiency is about 40 to 50%. We are not happy with the real application, but of course we have published papers in the nano letters and so on, and a couple of patents are there. So with this, I come to an end, and of course with different collaborators. We have the Professor Tamiya from the Osaka University, Japan, JACE Japan Advanced. With him, I have associated with him 8 years, and then another my PhD students and collaborators, and nano needles with Chikashi Nakamura. Of course, funding from the JSPS and Mon Bushro and JSPS next generation programs. All the funding we have done. I think that's it I have. Thank you for your attention. If you have any quick questions, I will not waste your time. As I promised you, in 2 minutes we can wind up. Thank you.
Hello.
Yes, sir. Actually, next speaker, Dr. Sudha, please excuse us because it is run up. You have any questions, audience? Could you
Yeah,
one or two
Subramaniam
questions
here.
could you?
Yeah, I could hear.
Yeah, this is Dr. Subramaniam, PhD Biosciences. Wonderful technology, wonderful presentation. I don't have any, but you have too many slides. I mean, it's very difficult to follow that many slides for anybody.
Sorry, because
Okay, that's my criticism. But my point, I want to make it very clearly, is that biology is too complicated. Phenotype is very, very dynamic. You cannot have a single cell related to the entire biology. Yeah, it could be used as a diagnostic method, but if you want to make extrapolations to biology, biology is organ to organ communication, cell to cell communications, or the reason biology works. So single cell analysis, we cannot extrapolate it to the cancer biology. That's my okay. So that's my major problem with this type of assays. What we're doing is we're taking cancer steroids from patients and them in microfluidic channels because you need the entire organs steroids with the cell to communication has to be organ to organ communication. We are building a microfluidic channel combined, tagged into different organs along with the steroids. We don't take single cells. In humans, the cancer biology is very
Yes, sir.
important.
Sorry to interrupt. If you want to identify, this is the way. Yes, because when you want to see at the organ level, yes, that is a different. We have the organ on chips also. We are doing parallelly. That is different. But if you wanted to identify what is the causative of the so cancer research means find, for example, the examples that I have given, and you would have actually limited options unless until you go into the just peripheral blood based analysis. So that was our realization those days about decade ago, and then we have actually demonstrated this is just not only for the lymphocytes, and you can apply for many substances.
Okay. Yeah. I mean, we'll talk later. I'll give you a call. I'll get your number and I'll talk to you more.
Sure. Thank you.
Okay. Sir, actually, with the interest of the time, I think I would like to conclude, Dr. Rao. It's really it's an application, it's intense biology application, and application at the best. Actually, biology plus technology and application. You had done extremely great job, and also you work with the pioneers like Nobel laureates, and also more important thing, you brought the application to more to the life. Thank you so much for.
Thank you.
In fact, actually, I have a line of questions that definitely I'll write to you after this talk.
Sure.
I hope we'll meet sometime.
Yeah.
With due respect, extremely, sir. I would like to conclude this. Thankful for you for your time. Since it is a Thanksgiving weekend, Dr. Sudha is waiting, so I would like to hand over it to the Dr. Srinivas, who is next chair to introduce the speaker.
Anyway, I should say maybe I have taken a little bit more time. Sorry.
No, sorry, sir. Because we're the family, we're department, we're family, we respect everybody's time. So thank you so much, sir. No sorries among us. And Dr. Shubhashini, thank you. Now the connection to Dr. Srinivas.
Yeah.
Srinivas, please take over.
Srinivas, just wait a minute. Sir, thank you very much. Ramchandra sir will give another platform with more time actually. Because of paucity of time, we could not give you more time. Because two other speakers are there. Madam is also there. And one more speaker is also from USA.
Yeah.
Thank you, sir. Thank you, Anil, for chairing the session. Thank you. Before we invite Srinivas Rao garu, I'll ask Apranji will introduce Srinivas Rao garu. Then Srinivas garu, you take the chairperson.
No need, madam. Just will introduce Madam Sudha, madam.
It's okay. Anyway, Apranji is there now.
Hello,
madam. Madam, hi, Srinivas Rao garu. Good evening. And first, I would like to introduce Srinivas Rao garu, who is chairing the session for Sudha Talk. And Srinivas Rao garu is a nutrition biochemist and micronutrient research specialist in the division of food composition and nutrition analysis, and at National Institute of Nutrition,
Madam, you are not audible.
We can't hear you. Hello?
Madam, you are not audible.
You are not audible, madam.
Somebody
చెప్పండి మీరు.
somebody should remind her, otherwise she is reading.
నాగరావు గారు చెప్పండి.
Madam, you are not audible, ma'am.
Apparanji, I just sent you.
Hello. Madam, you are not audible.
Yeah, they're looking after. There's a problem in the head's room. They are looking after. They just went. I just sent a person to Madam. Some technical problem, just.
Just wondering what is the time for Sudha now in US?
It's 5:20 in the morning.
Okay. You are used to get up early, so no issues for you. Yes.
I get up early, but today I was up from 2:30. I think I was hearing Dr. Murali Krishna's talk, but not before that. I am hoping I can hear the recordings.
So that don't worry. This is Subramanian.
Yeah, వినిపించలేదండి మళ్ళీ ఇంకోసారి చెప్తారా?
Sudha never sleeps, that's what I said. I know you better than anybody else, right? So
Apparanji is back, I guess.
Yeah, there is a problem, Sudha.
But she is not audible. Sir, she is not audible, sir. Apparanji madam is not audible.
Yeah, that's right. They are looking after. Admin, I just sent admin to there. What time, Sudha, it is there?
It's 5:22 now.
Oh.
Good morning.
Good. That's why you're looking fresh.
Actually, no, I was yawning a few minutes back, so pardon me if I do that during my talk.
there is a general, don't take me wrong or anything. If you have a 45-minute presentation, do not have more than 20 to 25 slides. People lose attention of you because if you put 87 slides into 90 slides, nobody can pay attention to all those slides because you're not even spending half a minute on the slide. So don't put slides for a 45-minute presentation. You should not have more than 25.
Also, don't make the slides very busy.
That's good suggestion, sir.
There is too much of text and too many figures. It's
yeah.
very hard to follow.
It's very difficult to follow.
Actually, from the noon itself, we are having a backlog. I think we are running short of time.
Already we are running schedule behind now.
Could somebody step in and introduce the speaker in the interest of time?
So,
can we start, sir?
So I request Srinivas Rao Garu to introduce speaker Sudha madam.
Thank you, Dr. Subhashini. It's a great privilege to me to introduce Dr. Sudha. And before going to introduce Dr. Sudha, I was a student of Biochemistry Department of Andhra University from 1996 to 98. That is the Silver Jubilee batch. I am very happy that I would like to introduce Dr. Sudha. Dr. Sudha Chivukula is an experienced researcher in the vaccine research and development space with profound scientific expertise and business acumen. She has a PhD from Department of Biochemistry from Andhra University, India. And she has done her postdoctoral research from University Putra, Malaysia in area of genomics and transcriptomics, and is graduate from Indian Institute of Management, Lucknow also. So during Dr. Sudha's professional pursuit, she has worked on multiple viral vaccine projects, including human papillomavirus, rotavirus, influenza respiratory syncytial virus, the present problematic virus SARS-CoV-2, on combination vaccines as well as on mRNA vaccines. Dr. Sudha has held scientific and strategic leadership positions impacting translational research and taking vaccine candidates from conceptual phase through pharmaceutical and clinical development phase. Sudha has authored several scientific articles in reputed scientific journals and is currently leading the RNA vaccine technology at Sanofi Pasteur with a mission to build and integrate strategy and development of a mRNA vaccine platform at her organization. It's very interesting brief biography of Dr. Sudha. Now, I would like to request Dr. Sudha to give her talk. Thank you. Dr. Sudha.
Thank you, Dr. Srinivas Garu. I'm not sure I recall you, but I'm sure we overlapped in the department. Good evening to everyone at AU Biochemistry. It's an immense pleasure to be amongst the AU Biochemistry fraternity today to celebrate the Golden Jubilee. I first of all like to thank Dr. Subhashini Garu, Dr. Raghav Garu, Hemlata Garu, for inviting me for this meeting and seminar today. I'm pretty sure the department is celebrating a very proud, alumni. Listening to the talks today and some yesterday, I can tell you that the department has produced hundreds of scientists who are contributing to healthcare. And I think all of us at this moment should, feel good about celebrating the Golden Jubilee. I want to take a moment to bow down to my teachers, Dr. D. Sivaprasad Garu, Dr. Hemalata Garu, Dr. Ramarao Garu, Dr. Ramana Garu, Dr. T.M. Radhakrishna Garu, Dr. Govardhan Reddy Garu, and Sri Ramal Garu. I think I covered everyone, hopefully. And then, I would like to share my title today for my presentation. And hopefully I have only 15 slides. So Subramaniam Garu, I hope I'll not disappoint you. Can you see my slides, everyone?
Yes, Dr. Sudha ma'am, yeah.
Yeah. So today I'm going to spend some time with you all to share my experience on the mRNA vaccine platform. You probably have been hearing a lot of this in the last 1.5 years. And the title I chose for today is mRNA vaccine and a paradigm shift in pandemic preparedness. I think this pandemic has really changed the vaccinology in a perspective that is really moving to think of preparedness for future pandemics, delivering vaccines in a very rapid time, and that's kind of a new era in the vaccinology. The pandemic is really resulted in some new arsenals to fight infectious diseases. If you have seen this couple of vaccines based on novel technologies like the adenoviral vectors, the mRNA vaccines, these are even, I think, the DNA vaccine, all these have been first time in this pandemic. So we are really looking at a different era and a different way of dealing with infectious disease. And I'll focus on mRNA vaccines today and what we have done in this space. Today in my discussions, I'll be covering a little bit on the background of mRNA vaccines, just in case there are some students, I'm sure most of you know, but I'll touch base on what they are and how they are relevant to a pandemic and in future and current. A standard development path for a vaccine and how these platforms, these novel platforms like the mRNA vaccine platform really impact the development and why they become more and more relevant in the future fight to infectious disease. And again, looking at certain designs of elements of mRNA vaccines that really drive the potency or the efficacy of the vaccines. MRT is the technology that we developed, which we call as the mRNA therapeutic technology. That's we at Sanofi Pasteur in collaboration with a company called Translate Bio, which we recently acquired. We have developed this technology and we've started, using this technology for not just the pandemic, but also for other infectious diseases. And I'll give you some examples of the data that we have generated from this technology. Some disclaimers that I work for Sanofi Pasteur and okay, so what is the mRNA vaccine all about? Typically, a vaccine is either a live virus, a live attenuated virus, or an inactivated virus, a whole virus, or a whole bacterium, or a protein derived from that virus or bacterium. And I'm not sure if you can follow my cursor, but usually what we have seen, all these traditional vaccines are based on either proteins, inactivated, live attenuated, VLP, or the virus-like particle-based vaccines. But what we are seeing right now is the nucleic acid vaccines, or as a new generation. If we go back to the central dogma of molecular biology, the flow of genetic information is from DNA to mRNA and protein. I don't need to tell this to you all, but just to use this cartoon to say that while traditional vaccines have been based on the proteins, which is called the subunit vaccine, that means express these proteins in a recombinant host. That means you need large bioreactors to actually grow the recombinant host in case of subunit vaccine, and then purify it in large scale in manufacturing facilities requiring large, clean room footprints. Now, in case of mRNA vaccines, what we do is use the mRNA, which is actually coding or the message, or having the message for that particular protein, which is the target for this vaccine or for this infectious disease, as to use this mRNA to deliver, and this mRNA is taken up by the host cell, and the protein, which is again what we need for raising the antibodies, is actually produced within the cells. So our cells are going to act as bioreactors, and we don't need to do a large-scale production of protein. Yes, we need to make the mRNA, and this can be done by synthesis. And so that's kind of the concept of mRNA vaccine. Slightly different from DNA vaccines, where the DNA or the code or the gene that is coding for that antigen of interest is delivered into the cells. Remember, the mRNA is transcribed in the cytosol, whereas the DNA has to go into the nucleus for transcription to mRNA, and then in the cytosol, this will be the mRNA get translated, meaning that when you deliver mRNA into a cell as a vaccine candidate, the expression to protein happens in the cytosol. There is no need to cross the nucleus or the nuclear membrane, no need to cross the barrier. When you give a DNA vaccine, there is a chance that there is an interaction with this exogenous DNA with the host genome. That's kind of something you'll remember as one of the advantages of mRNA vaccine. Now, what is the vaccine made up of? We've all been hearing SARS-CoV-2, Pfizer vaccine, Moderna vaccine. There are many other vaccines, but what is this made up of? Let's imagine that this small cartoon of the SARS-CoV-2 virus, and we all know the spike protein on the surface of the virus is the target for this vaccine, for the SARS-CoV-2. Now, in mRNA vaccine, what we do is we've taken the gene of interest, which is a spike protein sequence. We made a plasmid DNA using, some vector or backbone, linearize the DNA. That means get the template for an in vitro transcription to an mRNA. This is a synthetic step. It just can be done in small, bioreactors, which just do not use any host cell. It's just a synthesis reaction. This mRNA now, which is produced from this in vitro transcription, is our vaccine candidate. But we all know mRNA is prone to the RNase degradation. It is very labile, and so it needs to be protected. Number two, this exogenous RNA needs to cross the cell barrier and enter into the cytosol. So that is where the concept of the lipid nanoparticle has come in, which is a protective, lipid shell. Let's look at it as an outer covering, where the mRNA is embedded in that lipid nanoparticle. The lipid is not only protecting the mRNA from potential RNase degradation, but is also helping to cross the cell membrane. And I'll tell you how it's crossing into the cell in a minute from now. Coming back to the mRNA itself, which is now our payload, which is the one actually that's carrying the message, can be again, presented in different formats. An unmodified mRNA, where means that the coding sequence is natural, uses natural nucleotides, sorry, I have a mistake here. A modified, unmodified, and self-replicating. A modified, where the coding sequence, the nucleotides, especially the uridine, is modified to pseudouridine. The uridine is known for causing some kind of innate signal in the human body, meaning that there is a possibility of an extreme inflammatory response, which leads to pain or fever or chill at the time of vaccination. And because of that, the uridine is replaced by what we call as the pseudouridine. It's a technology to reduce the innate signaling due to the mRNA. So these are the two formats, but there's another format called the self-replicating RNA, wherein apart from having just the gene of interest mRNA, there is also some replicon, which are the non-structural proteins from certain viruses, like the alpha viruses, that can be packed into this cassette, so that they help for a couple of cycles of subgenomic particles, which will increase the expression of the protein. So meaning in the self-replicating RNA, you not only just have the gene of interest, just reiterating, you also have a replicase cassette, which helps to make some more mRNA in the cell, and because of which you have more expression of protein, hence you have more antigen produced. And so you expect to see a higher immune response. In all, what we see now, if this is the vaccine that is, the SARS-CoV-2 vaccine using the spike protein package, made into a mRNA, single-stranded mRNA, and then packaged into this lipid nanoparticle, the expectation is that in a clinical setting, when immunized to humans or even in preclinical models, you see that there is induction of both T cell and B cell memory. And I will T cell and B cell immunity, and I will show you some data around that one. So what is, I described what is the mRNA vaccine all about, but what's the utility of these vaccines, especially in a pandemic situation? Let's come back and see that. So the mRNA, as I described you, the synthetic process and the form and the encapsulation, the synthesis of the mRNA, encapsulation into lipid nanoparticles, these have nothing to do with the antigen sequence. The process has nothing to do with the antigen sequence. Irrespective of what the sequence is, the process is going to be the same. But imagine if you were traditional vaccines where you're making some live attenuated virus or subunit, depending on what the protein is and the characteristics of the protein, example, size, glycosylation, PI, whatever, the process to make that particular protein will change. But in this case, it's going to be a flex, what I would say, a broad manufacturing process that can be applied to more or less all antigens that need to be made. So this actually simplifies new product industrialization. And why this is important, new product industrialization. SARS-CoV-2 was a new product 1.5 years back. It was something new, and we saw that today it's completely industrial with billions of doses being sold. So that kind of, because there's only one process for any antigen, it's very fast to go into that product phase. So the R&D gets shortened. As I described it's the mRNA vaccines, the expression of the protein happens in the cytosol. There's no interaction with the genome, the host genome, and so there is no risk of any integration of this mRNA vaccine or the gene coming from the mRNA vaccine into the host. This is something which I can say the mRNA vaccines have advantage over the DNA vaccines. This is a reusable technology, meaning that now I put this in comparison with the adenoviral or viral vector vaccines, where you could see anti-vector immunity, which means that the backbone of the adeno, like for example in certain of the technologies which we have seen, I think it's a ChEDOX technology, we have seen that there is an adenoviral vector that's carrying your SARS-CoV-2 DNA, and there is an immunity or an antibody response to that vector. So after second dose, or even after the, when you're going for a booster, maybe the third dose, you're going to see some reduction in immune responses because the anti-vector immunity. And then the next advantage of the mRNA vaccines, and I can tell you from my own work that we're able to make mRNA vaccines either be transmembrane attached. That means you design the antigens to have a transmembrane domain and have it, with attached to the cell membrane. You can secrete them, and you can make the flexible design. We are able to express the most complex antigens de novo using mRNA vaccines. Now when I say complex antigens, some of the bacterial vaccines, they use the outer membrane protein, bacterial outer membrane, as a vaccine. They are certain of these outer membranes which are so complex that they are not only structurally complex, but they also have a lot of what I would say integration with their host cell membrane, because of which making them in bioreactors really makes it very complex. So with mRNA, we're able to use our host cells to make these complex antigens while having the right translational modification that is needed for the immunity or the right kind of immunity. Finally, we get the adaptive immune response to both B and T cells. And again, as I said, I will show some data around that one. So what did I mean by saying that it's reducing the time, that the vaccine development time? Traditional vaccines, and I have seen that in my own career, we have taken 15 years to bring a vaccine to the stage of market or licensure or registration. Meaning that once we know what is the target, and I take the example of SARS-CoV-2, the spike protein being target, we would have to do some preclinical work to say, okay, this is the way I'll optimize the target, be it prefusion, for example, spike protein, we all know that it's a prefusion stabilized protein. That means certain mutations were included so that the protein is expressed in the conformation of the spike, which is before the virus fuses into with the cell membrane. So it's kind of a prefusion stabilized. In nature, that's a very metastable conformation. But in this case, we have done some mutations to make it prefusion stable. And so there's a lot of the target optimization and preclinical work that is done usually. And as I said, the manufacturing process has to be developed. If it's a traditional vaccine based on, let's say, protein or any other, and then a clinical study which goes on for a long, multiple years, clinical phase 1 followed by 2 and 3. These are more, I think most people are more familiar with this now with this pandemic, but this is the traditional time taken for licensure of vaccines. Phase 1 for safety immunogenicity, phase 2 for dose optimization or even schedule optimization. We know that every one of us got 021 or 028, but normally we try to do, is it 028 schedule for immunization, or that 0 1 month, 6 months, 6 months, their schedules are usually worked out. And then you do the efficacy trial where you actually see the efficacy of the vaccine in endemic places. That's the usual time. But what happened in the case of this pandemic, and especially due to technologies that really reduce the development time, I think for the spike protein, one of the advantages was the data from SARS-CoV-1 about the prefusion stabilization and probably even the MERS. That data was used as soon as the sequence was available for the SARS-CoV-2. Within 43 days, the phase 1 had started, meaning that from the time that we know the sequence of this virus to making a clinical phase 1 GMP lot, which is really the, covers for this piece. What I'm talking about is optimizing the preclinical and the phase 1 process development and manufacturing. CTM is clinical trial manufacturing. This phase was really cut down to 43. Normally it's 3 years, 4 years. It's really cut down to 43 days by some companies. And then, of course, there was definitely in a pandemic a shortened clinical development. But this is a period where we are gaining. And what I'm trying to say is that if a company could do in 43 days a start of clinical trial, that means that in the future pandemic, this can get even shortened. We can have preparation for pandemics by having libraries of the mRNA for zoonotic viruses, let's say, which are potentially going to be the future pandemic strains. We can have them stocked, the mRNA libraries, like it, which means what? It's just the linearized DNA, which I showed in the first slide, and it's no need of a host. It's very safe. Just store the linearized DNA in some freezers and for potential pandemic strains, and then we can be faster to catch up with the pandemic situation. And why was this possible? Again, I want to just reiterate standardized manufacturing process, because any sequence, you follow the same process to manufacture. Standardized QC methods. Why QC method is important? Because for regulation, for FDA, for WHO, for DCGI, there is to be a standard package to be submitted for the registration or for even for clinical studies, and they need to have proper methods. And we've seen the previous talk on how deep analytical methods can go into. Validation of those methods is important to make sure that we are getting the right quality and safety of our product into humans. So this is very important for regulation, and these things can be already ready-made if we have a standardized process. And then, of course, we don't have to use any pathogens. We don't have to grow any bugs. It's all synthetic. And we don't need dedicated facilities. That's critical. We don't need to build specialized facilities for certain live attenuated viruses or certain pathogens or certain recombinant antigens. We can use, because this is a synthetic process, we can use any facility, and there is no need to dedicate because of certain use of some BSL-2 or BSL-3 kind of facilities in this case. So that's kind of one of the areas where these platforms made a huge impact in pandemic. I want to switch gears a little bit now to the mRNA vaccine itself and how we design this vaccine, what goes into the design of the different elements. The mRNA vaccine is essentially comparable or is the same as the fully processed mature mRNA in the eukaryotic that is found in the eukaryotic cell, meaning that it will have a 5' cap, a poly-A tail, a 5', 3' UTR or untranslated region, and a coding region. Now, I just would like to share what are the efforts we put to get our mRNA into the right design, which will help us get the right level of immunostimulation, meaning that we want to have the good immune responses to the vaccine candidate, but we do not want to have any inflammatory response that lead to pain or fever or chills or headache. And if any of you have read about the mRNA vaccines, I think this is one of the limitations that, yes, we do see pain and in the mRNA vaccine. This is an area for improvement. And I can tell you what goes into the design to avoid those kind of inflammatory responses. Let's start with the coding region. This is the region which is coding for the gene of interest. If again, we can take the example of SARS-CoV-2, we can take the example of influenza, hemagglutinin, any of the protein that we want to express. This will have a signal peptide. It could have a transmembrane domain to have it, expressed as a transmembrane protein, or it can be secreted, depending on what the antigen is. And most importantly, we can use either, as I said in the first slide, the modified or the unmodified mRNA format. The modified, in which the uridine is replaced by pseudouridine, and the unmodified, which is natural, just like you see in a natural mRNA. What we used is the unmodified, which is the natural. And our argument was that in a vaccine, we want immunity, we want some inflammation. Unless we see kick off the innate signal, we need that innate signal to get some, vaccine immunity or good antibody responses. But we don't know what is the level of innate signal that can go to the danger. So it's like a Goldilocks, not too high, not too low. We wanted that right level. We were expecting that the unmodified will be good, but certain companies like Moderna, BioNTech, both use the modified. Well, it's a different situation that we don't have the patent, and there's a patent war for the modified nucleotide. And for all those reasons, we went for the unmodified nucleotides. And again, we did not use the self-amplifying. We just used the regular mRNA format. Coming to the 5' cap, the cap is like any eukaryote, the 7-methyl guanosine cap. The cap is important because, one, it protects from the exonuclease digestion, and second, it promotes the translation by binding of the initiation factor. What is important here is to have a cap which is similar to the multicellular eukaryote. Reason being, if the cap is not similar, which is we have different cap features, I won't go into that, cap 0, cap 1. The cap 1 is the one that is similar to what is in humans. And to design that cap and to have that cap feature is important to reduce, again, a danger signal or recognition of this mRNA as a foreign mRNA. So we mimic the cap to look as human-like as possible, in simple words. Coming to the poly-A tail, the poly-A tail usually gives, stability to the RNA in vivo, and it also helps in the initiation of translation. The way we add the poly-A tail can be two ways. You can put it in the plasmid. Remember, in the first slide, I showed that there is a linearized DNA template, and that comes from the plasmid, right? You make a plasmid with a vector backbone and the gene of interest, and then linearize that. So that you can put in the poly-A in that template itself, or post-transcriptionally, you can add the poly-A. The cap is usually added post-transcriptionally. Co-transcriptionally is done by few companies, but we do a post-transcriptional cap and a post-transcriptional tail, and there are some advantages to it. It's different platforms, different companies claim differently, but I can tell you that for the poly-A tail, the longer the poly-A, 200, 300 nucleotides longer, we've seen better stability. If you add poly-A in the plasmid beyond 100, it just cannot be, it throws out the E. coli or the host will throw out that plasmid and it will not retain. So retaining a plasmid with a poly-A in an E. coli host become a challenge. So what we do is that we safely do the poly-A addition enzymatically after the transcription reaction. We spoke about cap, tail, and now the UTRs. Again, the 5', 3' UTRs are again very important regulatory elements. They're important for translation efficiency. The 5' UTR engages with the ribosome. for initiation of the translation, and choosing the 5' 3' UTRs again based on a lot of data that we generated, which UTRs to use. That some work went into that. And so what I can tell you is that we defined each of these elements based on some data that we generated and based on some patterns that we have to avoid crashing into, and we designed this construct. Ultimately, what I'm trying to tell you is that if you had a coding region with unmodified, there is a chance that you have the innate signal. If you have a cap that's not the Cap1-like or the human-like, there is a danger of the innate signal. If you had a poly A which is shorter, there's a danger of losing the stability of the RNA. Every of these elements is important. Every of this has to be optimized carefully to get the right level of translation efficiency and the right level of immune stimulatory profile for that vaccine. Now I move into the lipid nanoparticle. I explained about the mRNA. We move into the lipid nanoparticle. And this is kind of a cartoon, and you probably saw in my agenda slide an electron microscopy of the picture of the cryo-EM picture of the lipid nanoparticle that we made in our lab. But this is kind of a cartoon where you can see that the lipid nanoparticle actually has a multilamellar layer, like at least in our case, it's multilamellar layers. The mRNA being in the core, and the way mRNA is negatively charged. The components of the lipid nanoparticle, mainly the ionizable lipid, is positively charged, and that's why you have the mRNA being encapsulated by these lipids. Now, there are 4 kinds of lipids in this lipid nanoparticle: the ionizable lipid, the cholesterol, the helper lipid or the structural lipid, and the PEG lipid. The role of the ionizable lipid, as I said, is because of its charge, the positive charge it encapsulates the mRNA. It also helps to release the mRNA from the endosome. And I will tell you, I probably missed the slide, or it's there in the next slide. I'll tell you how the mRNA gets internalized, but there is a step of endosome escape for that lipid nanoparticle to enter into the cytosol, and it's a cationic lipid that helps, because the cationic lipid normally in a physiological condition, physiological pH is neutral, but once it's in an endosome where the pH is lower, 6-ish, it actually is protonated, and because of which it pushes out the lipid nanoparticle out of the endosome, which is called the endosome escape. The cholesterol is adding rigidity to this LNP. The helper lipids also form like a membrane and layers for this lipid nanoparticle, and the PEG is the outermost layer, which actually acts as a shield for this particle. Now, the way the mRNA and the lipids, we call this drug product, that's the final product. How they are made, mRNA is called the drug substance. As I said, has a negative charge, it's in an aqueous phase, whereas the lipids are in an organic phase. The two are actually mixed at a certain flow rate and at a certain concentration, flow rate ratio. And this helps them form, because when the aqueous and the organic phase come together at a specific flow rate, you get these nanoparticles formed. I'm simplifying this, but this can be done using a cross flow, a T mix or a Y mix. We've seen some of those videos in the previous talk. It can be done by microfluidics, but at the end, it's about mixing the aqueous and the organic phase to form these lipid nanoparticles. The flow rate, the process at which they are done, will determine the size of the particles, determine the shape of the particles, and ultimately the efficacy of the particles. So again, all of these components are the ratios at which they are present, as well as the process at which they are made, will define the potency, the translation efficiency, and the immunostimulatory. Remember, when I say immunostimulatory, not only the mRNA is giving you the immunostimulation, but also the lipids, because the lipids can also cause some kind of immunomodulation. And we've seen that lipids do contribute, and they are adjuvant-like, and they behave like adjuvants, and they can skew the immune responses in a certain specific direction. I talked about endosomal escape in the previous slide, and I'll tell you exactly what happens at the cellular level when a person receives mRNA vaccine. Let's say this is mRNA vaccine the person is receiving. As I said, there is a cationic lipid and a helper lipid. The helper lipids are fusionogenic. They can fuse with the cell membrane, and they can, and then they facilitate the entry of the lipid nanoparticle into the cell. The process is through what we call as an endocytic pathway. An endosome is formed. An endosome is carrying now the lipid nanoparticles. Inside the endosome, you have the TLR recognition, the TLR7/8 receptors, which actually kick or trigger the immune responses in a specific direction. They are the pathogen recognition receptors over there. And then you have this endosome moving in the cytosol, egress through the cytosol. The mechanism of the endosome escape is not fully defined, but it's definitely, spatio-temporally limited. The endosome carrying that lipid nanoparticle, while it's moving in the cytosol, goes through some osmotic changes, ion pH changes, and actually releases the LNP into the cytosol, and actually the mRNA into the cytosol. That process is defined by the main lipid, that's the ionizable lipid. And so the choice of the ionizable lipid actually determines the dynamics for this endosome escape. And what is known is, of all the lipid nanoparticles that are immunized, let's say 30 microgram dose or 100 microgram dose, only 1% of it escapes the endosome. That's kind of the, I'm not sure it's a theoretical value, but it's well described in literature that it's 1% of the lipid nanoparticles that actually escape the endosome and are expressed as the target protein. Now, once there is a escape of the mRNA, it gets through the ribosomal machinery. You have the protein expressed, and depending on whether you designed a transmembrane, intracellular, or secreted protein, that protein is expressed and then ready for antigen presentation to get to trigger those immune responses. So with that background on the technology itself, I would like to share some data from our work. We have 2 ongoing clinical trials based on this platform that we generated or we developed. One is the mRNA COVID-19 vaccine, and the other one is the mRNA seasonal influenza vaccine. I'm extremely proud to be a part of this group. Although our COVID-19 vaccine may not ever go into a market, because we have another COVID-19 vaccine, which is in a fairly advanced stage for booster indication. The company won't like to have 2 products for the same indication. So we will probably land up using this data to understand our technology as a proof of concept for our technology, as well as go back to design in improving our technology. So again, coming back to those 2 candidates, I can't share with the clinical data yet. Probably it will be out on December 1st. We have some press release, but I can tell you that these 2 are, now we have some interim data coming out of these 2 trials. But I can share the preclinical data. This is kind of the antibody responses to the SARS-CoV-2 vaccine, which we call as MRT-5500. This is monkey data, and I can just share with you what we have done. If you look at the cartoon on the right top panel, we had administered the vaccine on day 0 and day 21 on monkeys, and we collected blood at different time points. And from the sera, we did the pseudovirus neutralization assays, or the pseudovirus assays or the neutralization assays. We've done a dose a study with 1545, 135 microgram immunized at just two administrations of those doses. And it's a busy slide, but carefully look that you can see that if you follow my cursor, 0, 15, 42, and by day 42, you see good immune responses, and these are functional antibody responses on both panels. We didn't see a great impact of the dose. We just thought that 15 micrograms was as good as 135 microgram. We didn't have to give that much. So in a way, it was a good news for us that we don't have to go very high dose. And then this one in the dark dots is actually the convalescent sera. That means sera from patients who were exposed to SARS-CoV-2. And you can see that their geometric mean of their neutralizing antibody is somewhere around 10 power 2, but we can see much higher with our vaccine after 2 doses. So that's kind of what we saw in our SARS-CoV-2 vaccine monkey model. I'll move on to the influenza vaccine monovalent. Again, this is H3N2. We call it MRT 5400. Here we had 2 doses again of the vaccine. Usually influenza is a single dose vaccine because there's a lot of pre-immunity to influenza. Every year, every season, you get a new strain. And so humans have pre-immunity to influenza, and so 1 dose is expected to be enough. But since this is a naive monkey, we used 2 doses. And you can see that it was day 0 and day 28. Again, we did 15, 45, 135, 250 microgram dose. And I think we are showing here day -6, day 14, day 28, and day 42 data. And you can see that by day 42, we have good microneutralizing titers. Now, what is that vaccine design here? It's the influenza HA or the hemagglutinin, which is the main target for influenza vaccine. This HA's role is it plays a role in attachment to the cell. And so what we're trying to do by creating antibodies or inducing antibodies to this protein, we are stopping the attachment of influenza virus to the cell. So that's why this is very critical. And you'll see that this data, we are getting good neutralizing antibodies to the H3N2 virus. I spoke about now on the left panel, you saw the B cell responses or the humoral responses. I spoke about mRNA vaccine inducing the T cell responses, and we took the PBMCs at day 42 to test for the interferon gamma and IL-13 as examples of Th1 and Th2 type of cytokine. And you can see that clearly we saw a Th1 bias with the mRNA vaccines. Usually, if you look at in this lower panel, even you can see that it's a Th2 type, and you can see the recombinant HA, which is a subunit vaccine as our control, did show more of the Th2 compared to the mRNA. So what I'm trying to say is that if you look at the ratio of Th1 by Th2, mRNA is definitely on the Th1 side. Which is something that probably is useful for most viral vaccine. I come back to the previous slides were all about influenza or SARS-CoV-2 neutralizing functional antibodies. In this particular one slide, I'm trying to show some challenge data in influenza, and this is kind of like an efficacy model. And you will see that we immunize, sorry, the needle is, the immunization should have been on day 0. And then we have bleeds on day 28 and day 42. And you can see that we have tested a couple of lipids here, which is the ionizable lipid, which we call lipid A, lipid B, are two different formulations. To keep it simple, an MC3 is a formulation that has been from an island that has been in literature and even in clinical trial, and there is an siRNA product based on this formulation. We use that as a kind of a benchmark, and we can see that we are getting good titers in mice to that HA antigen, which was immunized in these animals. That's the California is the strain, California 09 is the strain, and HA is the antigen or the mRNA that we immunize. See the dose is 0.4 microgram in mice, and we can see that we're getting the titers. Now, what are these titers doing? When we challenge those mice at day 100 something, we see that those mice which were immunized with either the lipid A or lipid B or even the MC3 formulations of mRNA survived, whereas those which were given the diluent actually had a terrible weight loss and they had to be euthanized. And you can see that this vaccine is not only inducing those functional antibody titers, but also protecting from a viral challenge. In this panel, you see a similar data with a second antigen of influenza, which is NA. And this is something very unique, and what we have done, and probably we are really making big claims here that this could be the first time that we are seeing an NA, which is the second target for influenza. Is not a vaccine yet, but there's a lot of work going on. Neuraminidase, which is actually an enzyme which cuts the sialic acid on the glycan. So basically, the virus has this neuraminidase chopping off all the sialic acids on the glycans on the mucus and facilitating the virus to reach the target cells. And this is the second target for influenza vaccine and is expected to give more broad protection to influenza. And so we use this antigen, and again, we have given 2 doses. We have tested it at the antibodies or the NA-specific antibodies, functional antibodies after dose 1 and after dose 2, and you can see that we're getting very high titers even after single dose. And we challenged the animals which got single dose or which got 2 doses, and you can see in this weight loss or cartoon on the right that those animals which got the neuraminidase, this strain is Michigan 15, which got the neuraminidase at either 0.4 microgram or even at 16 nanogram, actually got protected. And you can see that even whether it was a single dose or 2 doses, I think here we lost 1 mouse, but in general, we can see that even 16 nanogram could lead to some protection compared to what we saw in a diluent or a non-relevant mRNA. And so what we think is the mRNA vaccines work very well, not just for SARS-CoV-2, but also for seasonal influenza, at least for 2 target antigens that we tested. What we went on to do, and probably this is going to be my last slide or nearing to the end, what we did next was, okay, influenza is usually a quadrivalent vaccine, and I don't know how many of you have taken an influenza vaccine. It has 4 strains incorporated in it, at least in the Northern Hemisphere, which means that there are multiple HAs in the vaccine. That means if you're giving mRNA influenza vaccine, you need to accommodate at least 4 antigens, maybe more. Unless until there is some universal influenza vaccine candidate, we expect at least 4 antigens to be in one formulation. So what we did here is, and carefully look at my arrow or the cursor here, we have H1, H3, N1, N2. It's kind of a prototype formulation where we did 2 HAs, H1 representing the H1 and H3 what I would say, subtypes, and 2 NAs representing the N1 and N2 subtypes. So what we have is 4 antigens in one formulation, and we had representative controls where we made only H3N2 or H1N1 or only H1, meaning we had bivalent or monovalents, in the controls to really test our concept. Can we put 4 different mRNAs in a single LNP formulation? Here, literally, we have to imagine that the encapsulation of the mRNA in the LNP is actually multi-type. And so within the LNP, you have multi-type RNA. When they express, they need to express and assemble. HANA are not monomeric proteins. They are trimers. HA is a trimer, homotrimer, NA is a homotetramer. So they need to assemble to trimers or tetramers. And we don't want chimeras over there that HA of one type and the HA of other type form trimers, and we don't have right level of efficacy. So what we try to do is make sure that there is no interference between those HA and NA. There is no misassembly between those different multivalents, and we get the right level of immunogenicity. So when we put them in quadrivalents, H1, H3, N1, N2, and you can see in this box, the green dots represent the titers of California, one of the strains. And in the same way, the blue dots represent the Singapore, the second strain. When you had them as quadrivalents, or you had them as bivalents, or you had them as monovalents, you have the same level of titers, meaning there was no interference. So we are recovering all the titers of a monovalent vaccine, even in a quadrivalent formulation. And same holds true for the 4 antigens tested: California, Singapore, Michigan NA, and Perth NA. So we did 4 antigens. All of them recovered the full immunogenicity, whether they were in monovalent or bivalent or quadrivalent formulation. With that, what we did is we say is that yes, seasonal influenza vaccine multi-type is possible. I just got from NPJ that my paper is accepted. Hopefully, you will see this published very soon. So to conclude, the mRNA LNP technology, not just from us, but from several other companies, has demonstrated efficacy and speed in vaccine development. We have developed the mRNA vaccine, generated preclinical data for several candidates. I showed 2 candidates, but we have so many other candidates, including complex bacterial antigens. So this technology is here to stay, but there are certain challenges. We still store these vaccines at -80, which must be a nightmare for logistics in places in low income and lower middle income setting. We need to improve the tolerability, or we do hear a lot of pain after taking mRNA vaccine. I think that needs to be improved. We have seen that there is now a global footprint for manufacturing mRNA vaccines. This can only improve with time, with billions of doses being produced, which means that we are creating the infrastructure for producing vaccines for a potential future pandemic. And that's why this could be an answer for the future pandemics. And then, as I said, there are more opportunities, and we are looking from our clinical data and comparing it to our preclinical data. We are seeing that there's scope to increase the design elements, like how do we increase the expression? How do we improve the endosome escape? How do we target specific cells, like maybe the antigen presenting cells? So I think that's where I would stop. I would like to thank, first of all my teachers at Andhra University. I graduated from this department. I have my complete education in India, especially Andhra University. So thanks to everyone there. And thanks to everyone who supported this.
Yeah, it's very nice and excellent work that you are doing, Dr. Sudha Madam. Of course, it is very essential at this moment to entire globe in this pandemic. So anyhow, the time is very short. We are running behind the schedule, and just we would like to invite just a couple of questions, if anybody.
Yeah, I have a question.
Sudha, I have one question.
Yeah, Subhashini Madam. Yeah.
Yeah. Aparanji, go ahead.
Hi, Sudha. How are you? So I have one question. What about MHE binders? Did you check the MHE binders? Which class of MHE binders bind to the expressed mRNA expressed this thing?
Yeah, we are actually doing that work. Apparently, we have not generated data in that space.
Okay. And another question is regarding this Th1 and Th2 cytokines. You said elevated Th1 response and suppressed Th2. And how do you explain that with respect to humoral and cell-mediated? mRNA expressed protein is, stimulating cell-mediated or humoral?
Actually, it is giving you both. You have seen the antibody responses, right? You have seen the antibody. So it's not that it doesn't have the humoral response, but in addition, it has a strong Th1 response, which probably is helpful, in the efficacy. And it's very difficult to evaluate at this moment.
In fact, it should have, cell-mediated, good potential vaccine should have cell-mediated.
Yes, but to really separate those two and compartmentalize and see how much each is contributing to efficacy or protection at this moment is difficult, but we definitely think the CMI kind of responses will be, the T-cell responses, the Th1 responses will be of benefit, but we'll have to prove that in a clinical setting. We can show that in animal models, but showing that in a clinical setting will be a challenge.
What about adjuvant? Did you use any adjuvant during formulating vaccine?
No,
adjuvant.
no adjuvant. No.
Only
no
lipid nanoparticle with mRNA. Okay, excellent.
Yeah.
Okay.
Yeah, the lipid actually behaves as an adjuvant by itself.
Oh.
Any other
Thank you.
Yeah, this is Subramaniam. Again, wonderful presentation, so that really gave the basics to everybody to understand. My question is regarding, you already said that DNA vectors actually have the immunogenicity and autoantibody production. Is that the reason J&J vaccine was pulled out of the market? And how about the others, the vaccines with the DNA vectors? Have similar problem. And I'm not sure if the RNA also directly RNA generates any autoantibody production.
Yeah, I think right now we've seen one vaccine with the adenoviral. We'll have to wait and see what happens when the boosters come. Definitely there will be vector immunity. If this becomes, hope it does not, but if this becomes an annual vaccine, then, and if you have vector immunity, then that probably will become an issue, because we could see a drop in the antibodies because of vector immunity.
The volume is still low now. But anyway, the other one about the quadrivalent. This is already existing vaccine, right? The quadrivalent vaccine against flu.
Couldn't get the question, Andy. Can you just repeat?
The quadrivalent vaccine that H1N1, H1, H3, N1, N2. That's already existing vaccine, right?
So yes, you do have flu vaccines. Right now, there is in the US, and you probably know, there's annual vaccination with flu, but those are based on egg. I didn't speak of that, but the egg-based vaccines could sometimes get adapted to the glycosylation patterns. When vaccine is produced in egg, it's quite different. And because of the egg adaptation or the glycosylation patterns of egg, we may actually not get the right level of efficacy. And if you've seen some of the news, I think 2 years back, the vaccine was only 20% efficacious. And so egg has become a problem. And second problem with egg is that if there is a bird flu, then you don't have eggs to produce egg-based vaccine. So it's always been a risk to have an egg-based. We've survived with that for several years. It's been a very robust technology, but there's always a fear of egg adaptation, or even a bird flu kind of a situation that will deprive us of availability of eggs to produce vaccine. And so mRNA can become a replacement for the existing egg-based vaccines, either in pandemic or in seasonal setting.
Thank you.
The last question we will attend from Anil. Yeah. Anil.
Dr. Sudha, actually, excuse me for my ignorance. Suppose the cholesterol what you are using to pack that mRNA, do you have any possibility that it interacts with is there any left promoter regions or any material of the mRNA? The cholesterol interaction with the nucleic acid. Do you anticipate any kind of thing in the long run? You may not see such adverse things, but
yeah, so they could, the data is so limited at this moment. We have not, probably there is no literature around that to my knowledge. The role of the cholesterol is to, pack the mRNA and give rigidity to that structure. At the same time, there are different types of cholesterols that can be used, plant-based or, and even modifications of synthetic cholesterol. There is work going on in studying what is the impact of, let's say, changing one cholesterol to another. But I'm not sure if there's data on adverse effects, but these are slowly coming out. I mean, I don't know if some of you seen, there is a report of myocarditis related to mRNA vaccine. And we don't know what that can be. It can be a component of the lipid nanoparticle, or it can be the spike protein by itself doing something. But these are just data coming out. And that's why I said more work needs to be done in terms of safety and reactogenicity.
Yeah, thank you and congrats for your MTJ paper.
Yeah, thank you, Dr. Sudha Madam, for a wonderful and excellent presentation. And hope that you may come with the positive results soon to address this pandemic problem at a global level. Also, I would like to express my sincere thanks to the Department of Biochemistry, present existing staff, for giving me this opportunity to involve into the Golden Jubilee celebrations. And I hear that Professor Hemulata Madam is going to be retiring. Madam, I wish you a very happy retirement life. And thank you very much. Now over to Dr. Subhashin. Subhashin garu. Hello.
Madam coordinators.
Am I audible?
Yes, ma'am. Yeah.
Okay. Thank you, Sudha and Srinivas Rao. And next session is I welcome Professor D. Govindarao Garu for chairing the session. For speaker, Professor T. Sridhar Rao Garu. First, I would like to introduce Professor D. Govindarao Garu. Dr. D. Govindarao, currently working as a professor, Department of Biochemistry and Molecular Biology in Central University of Kerala. He is Dean, School of Biological Sciences and Director of Research in Kerala University, Central University of Kerala. He obtained master's degree and PhD degree from Andhra University. He was awarded with Best Teacher as well as Best Researcher by Geetham University in 2015 and 16. I welcome Govindarao Garu to chair the session. Sir, please.
Govindarao sir, are you there?
Thanks.
Yeah.
Hello,
Welcome sir.
Hello sir, how are you?
Sridhar garu, welcome sir.
Thank you.
Hello sir, Sridhar.
Yeah, thank you very much. Good evening to all of you, my beloved, respected teachers and eminent speakers and alumni and friends. Good evening to all of you. So I am privileged to introduce the speaker of the session, Dr. B. Sridhar Rao Garu, who is currently works at American Type Culture Collection, Virginia, USA. Dr. B. Sridhar Rao graduated from Andhra University, Department of
Sorry,
Biotechnology.
sir, your face is only visible half. We can't see you. Okay, now it's better.
Dr. B. Sridhar Rao is graduated from AU Department of Biochemistry in 1977 and obtained PhD from Department of Biochemistry, Indian Institute of Science, Bangalore. And he went to work at Kansas State University, subsequently worked at NIH, National Institutes of Health, Bethesda, and Johns Hopkins University on DNA replication. He also worked at CFTRI Mysore before he finally settled in USA. As I said, he currently works at ATCC in Virginia, USA. So with this brief introduction, I invite Dr. B. Sridhar Rao sir to deliver a talk on role of homopurine and homopyrimidine sequences in DNA replication. Dr. Sridhar Rao sir.
Very much, Professor Govindra Rao garu. Is everybody able to hear me? Hello?
Yes, sir. Yes, okay. Sir. You're audible.
Okay. Thank you very much, Professor Govindra Rao garu, for the nice introduction. And I feel very honored to be invited back to my alma mater to give this talk. And I thank Professor D. Sivaprasad garu, Subhashini garu, and Hemalata garu, who are all instrumental in encouraging me to come and share my experience with faculty as well as students and other dignitaries who are all alumni of the Department of Biochemistry. I was there only for a couple of years, but those 2 years have been very pleasant, and I still cherish the memory of those associations and friendships even today after a very long time. Okay, thank you very much. Let me share my content. Everybody able to see my slide?
You are, yes.
Everybody is able to? Yes, okay. Thank you very much. Thank you very much. Now I can go ahead. The title of my talk is Role of Homopyrimidine Sequences in DNA Replication. Okay. These sequences are unusual in the sense that they are long stretches of purines or pyrimidines in one strand, that is purine rich or pyrimidine rich in one strand, and the corresponding pyrimidines or purines in the other strand. Normally we see all A, T, G, C distributed all over, and in these sequences you see only purines in one strand and pyrimidines on the other strand. So as soon as you look at it, you know that these are different. These are not normal. Okay, here is one example of a similar sequence, homopyrimidine sequence with G's and A's in one strand and C's and T's on the other strand, alternating guanosine and adenosine sequences. I'm presenting this because I worked on this one. There are also other sequences which are slightly different from this, but still from this one, but still they are purine-rich sequences. There are some exceptions also. Okay, these are unusual not only in the sequence but also in the structure because they have a potential to form an unusual structure that is different from the normal double-stranded DNA. Okay, which we all know about. This can form a triple helical structure, that is a 3-stranded structure. Okay, how that happens is in this DNA sequence, a part of that sequence can unwind, this DNA unwind, and you see instead of a double-stranded DNA, you can have 2 single-stranded DNAs. One of these strands can fold back and then base pair with the double-stranded portion of the same molecule. Okay, the way that happens is shown in the next slide. So this is the same sequence that I presented in the previous slide. What happens is the DNA can unwind here And one of these two strands can fold back and make a triple helix, okay, or three-stranded structure in this. You don't see helix here, but it's a three-stranded structure with the single-stranded sequence sticking around. Okay, it can happen in many different ways, like the DNA can unwind here, and one of the two strands can pair. Okay, either this strand or this strand. They can go back, form a three-stranded structure. And it can unwind on this side too. So one of these two strands can fold back and form a three-stranded structure. So you have, all these structures are possible. Okay, now how does this happen? We know that DNA is double-stranded and it is all Watt-centric bonds, ATGC pairs. So how do you get a triple-stranded structure? That is because, see, you can see the three strands here. This is the vertical bars here are the Watt-centric bonds. Okay. The next bond is these dots here. They are what are called Hooke's-Steen base pairs. Okay. In the major groove of the DNA, there is enough space to accept some more groups or bases. Okay. So it is possible. It is possible to have a triplex structure. So what is a Hooke's-Steen base pair? This is just one of the examples. It can happen in many different ways. This is the standard Watt-centric AT base pair. Okay. Now, on this side, one more base can come along and make hydrogen bonds here. So instead of 2, you have AT base pair. You can have TAT. 3 bases. Okay, that's what is Hooke's-Steen base pairing. And I'm showing here TA and T, but you can also have CG and C here. Okay. It is also possible to have another purine coming along here. So in one of those structures, you can have AAT or GGC. Those things can happen too in these structures. So this is a local structural, this triple helical structure is a local structural variation. You have the double-stranded DNA all along, but locally there may be some triple helices here and there. And this kind of unusual sequences, they're not very uncommon. They're widely distributed among eukaryotic genomes, and we have tested different genomes. There is some variation from species to species. Rodents have the highest content, followed by primates and avian species. They're also present in yeasts and plants and least represented in prokaryotes. You don't normally see them in prokaryotes. Okay. Okay. They're also associated with some diseases. Okay, several diseases. Not necessarily by the sequence that I have shown, which is the alternating guanosine and adenosine sequences, but there are other repeats. It can be a dinucleotide repeat, it can be a trinucleotide repeat. Occasionally, you can have even a tetranucleotide repeats. They're all associated. They all have a propensity to adopt unusual structures and give rise to diseases. Okay. Very hard. They can be found in hot spots in chromosome and can cause chromosomal translocations, the breakpoints, and all those things that can cause many cancers. And they can expand. I mean, these sequences can expand, and that is not necessarily related to chromosomal duplication. So even in cells where the cell division is not very rare, they can still expand locally. And then cause many neurological diseases, etc. Okay. Now, we are interested in this sequence. Okay. To give a background, we were working on eukaryotic DNA replication origin sequences. The way you work with those sequences is you take any origin eukaryotic replication origin sequence, and you can monitor their replication in animal cells. What you do is you take any sequence and put it in a plasmid and grow it in bacteria, isolate them, and put them back in animal cells and see if the origin replicates or not. Okay. If the origin replicates, you can monitor the replication by looking at the state of methylation of this DNA. I'll come back to this later. We'll touch upon this methylation a little later. So when we are working on this one of our collaborators has found that he was working on polyoma virus, and he was working on a polyoma virus transformed rat cell line. Okay, the polyoma virus gets integrated into the chromosome of the rat cell line, and then the polyoma virus gets amplified many times. So what they found is, when they're looking at the amplified polyoma virus sequence, the polyoma virus sequence amplification stops at a particular point. It doesn't happen infinitely. It has to be controlled, it has to be regulated, because if gene amplification keeps on going forever, it will lead to chromosomal duplication rather than gene amplification. So when they're looking at this sequence, and they were also sequencing the flanking cellular sequences, and then they found an unusual sequence there, an alternating guanosine adenosine sequence. We thought that probably this sequence can have an effect on the DNA replication. We are interested in DNA replication, so we thought we would test this one. So what we did is, the first thing, easy thing is our model system is SV40. So let us clone this sequence into SV40 and see how it affects SV40 DNA replication. Okay. And then we took that sequence and then cloned it in SV40. I call this SV40 genome, GA genome, because this is wild type, basically wild type SV40 with the hatched area is a cloned sequence. Okay. This is a short sequence, about 50 base pairs. It can be easily accommodated in SV40 without any problem. So this is the sequence, and these are the various restriction fragments and sites that we have used in the subsequent experiments. Okay, now the question we have to ask is, when we put a sequence like that in SV40, how does SV40 replication is affected? SV40, simply put, is a single double-stranded DNA molecule. The DNA replication starts here, which is the origin. Okay, I hope you are all seeing my cursor. At the top of it is the origin sequence. That's where the replication starts and then goes bidirectionally at equal rates, and the replication forks move 180 degrees away from the origin. And that's how, when the two forks meet here, that's when the replication is terminating. Now, you are putting this unusual sequence here, and you want to see the effect on the replication fork movement through that sequence. So now what you would expect? There are several possibilities. One is the fork just happily moves to the cloned insert without any impediment, and the SV40 replicates happily. That's one possibility. The other possibility is it can stall here completely. Okay. What happens if it stalls here completely? The termination is, origin is sequence specific. The termination is not sequence specific. So the replication fork from the other side can move through this region and come all the way around and meet this clone sequence here, and the replication can terminate here instead of here. That's one possibility. In that case, what do you see? The terminus is at the clone sequence and not at the usual sequence. Okay. The other possibility is it can slow down. It can just impede the movement of the fork, but the fork ultimately passes through the barrier and then continues. In that case, what happens? SV40 can divide, but the terminus is not here because this on this side the fork is delayed. The other fork passes the usual terminus region and comes along here. So they meet somewhere in between, and the DNA replication gets terminated here. In that case, what will you see is a mild shift of the terminus. Okay, it's not a complete shift of the terminus, but little bit of shift in the terminus. So when we clone this sequence, or will the virus die? That is the point. Okay? Because it is an unusual sequence, the virus lives. So this is what happens. The virus is stable, was replicating, and it is growing slowly, and it has smaller plaque size and lower titers. Okay, so that's what I summarized here. The GA sequence is cloned in SV40 and is stably maintained. Okay, the virus grows slowly than the wild type. It has smaller plaques, exhibits lower titers, and it is also stable in mixed infection experiments. Even if you do a mixed infection experiment, it still grows in the presence of the wild type, but though the wild type grows more rapidly than the mutant. It's a variant, it's an altered virus, okay? And then all these effects, okay, can be explained in many different ways. Okay, anything can happen in a living system. But one thing is, if the replication is affected in any way by this sequence, these effects can result. Because the virus can ultimately divide and replicate, but this kind of slowing of the biological process can be explained by the slowing of the replication fork movement through the cloned insert. Okay. How do you check that? How do you check that? So if you think the terminus is shifting to the side, what experiments do you do? One experiment you can do is pulse label the replicating DNA. If you pulse label the replicating DNA with radioactive nucleotides for a short while, where does the radioactive nucleotides, where does the label go? The radioactive nucleotides, where are they incorporated, and how do you do the experiment? Briefly, to keep in mind, this is I just showed this is SV40 genome with the insert, but briefly recapitulate. This is standard. Everybody knows, or a lot of people know about this, how the DNA replicates. But still, in order to revise one more time, you have the SV40 DNA here. I'm giving an SV40 DNA example, but it is true for so many circular DNA molecules like plasmids or whatever, which have a single origin of DNA replication. If you have multiple origins, you have a different story altogether. So this is SV40 DNA with a single origin of replication here, and it grows bidirectionally. There are cases where the fork bubble grows only, the replication grows only unidirectionally, but in this case it goes bidirectionally. So this is the replication bubble which is blown up here. The two strands separate and the nascent strands start getting synthesized. Now the bubble keeps growing, and you get almost a catenated dimer, almost, which gets separated, and you get daughter molecules, two daughter molecules. Now, if you pulse label, where does the label go? The label goes everywhere because they're all replicating, they're all incorporating nucleotides at any given time, so they go everywhere. But what you do in the experiment is you isolate these daughter molecules and get rid of all those intermediates. When you do that, where do you see the incorporated radioactive nucleotides? Okay, they go in the terminus region, because by that time you pulse label for just a minute or two. Okay, by that time the SV40 DNA has almost completed this one. The daughter molecules have almost completed the replication, like this stage, And then at that time, it is the terminus portion that is still being synthesized, and that picks up your radioactive DNA, most of it. So when you do that, most of your radioactive nucleotides end up at the terminus. Okay, that is shown in the next slide. Okay, this is the pulse labeling of the SV40 DNA experiment, and the one on the left is the wild type SV40 DNA. And when you do the restriction digestion and measure the radioactivity in the fragments, this is how it looks like. This is the origin, this is the terminus. Okay. Now, the right side is the mutant or the variant which we have constructed. In the variant virus, almost the same. So it's not really stalling the replication fork at the cloned insert here. Okay. And it is continuing, but so it is almost the same, but there is one minor difference between the two pictures, and that is this fragment, the variant fragment is incorporating more label than the wild type. There is some spillover into this one. So, and it happens reproducible. Okay. Why does that happen? What you are seeing is probably it's not terminating here, but slightly to the side. So there is some activity showing up here. It happens consistently, and you can see a time course also by increasing the length of the pulse, time course of the pulse. Okay, and here we are, I need to tell you, but this is a tritium labeled thymidine incorporation. The cells are chronically labeled with C14 thymidine and pulse labeled with tritium labeled thymidine. And what we are looking at is tritium by C14 ratio in order to get rid of all the back. Now, when you do a time course of pulse, this radioactive nuclear incorporation in this fragment keeps on increasing. Okay, these are the same fragments. This is the fragment on the left of the terminus, and this is the fragment, the second one, the one below that, is the fragment on the right side of the terminus that is closer to the cloned insert. This is the wild type. This is the wild type. These two fragments are of importance. So theoretically, this is how it looks like in the wild type SV40. Now, if you have a cloned insert, the one on the right side starts incorporating more. As you increase the pulse, it incorporates more and more because the terminus is shifting toward that side. So this is probably causing a slowing of the replication fork movement or pausing of the replication fork movement at the cloned insert. So that fragment is attracting more label because the terminus is being shifted toward that side. So now, what we have dealt with here now is completed molecules. As I said, we got rid of all the replicative intermediates, but isolated completed daughter molecules. Now, what about the replicative intermediates? These were a challenge for quite some time because you get different kinds of molecules, different kinds of populations, and all sorts of sizes and shapes. So how do you study those? Okay. So what happens during replication is this. You have this SV40 DNA here, and the replication starts here, grows bidirectionally, and then gets terminated here. So if you take such a replicating molecule, or the completed molecule, you can take the completed molecules also. If you digest, for example, at this site at the terminus with one restriction enzyme, what do you get? If it is a completed molecule, you get a linear molecule. SV40 is about 5 kilobases, so you get a linear molecule. But if it is a replicative intermediate with a bubble growing, you get a linear molecule with a replication bubble right in the center. Okay, but if you cut at the terminus as well as the origin, you get two fragments half the size of the genome. And these have replication forks at one end. So you get this kind of a molecule, a fork, a Y-shaped molecule. On the other hand, if you cut it right in the middle of the origin, but don't cut anywhere else, what do you get? You get a full-length molecule, linear molecule, but these have replication forks on both sides. Okay. And another possibility is, okay, you cut here. I hope everybody is seeing my cursor. Am I audible and my cursor is fine?
Yes, Dr.
Okay, thank you. Thank you. Thank you. Sometimes people are complaining that they are not able to see anything or see the slides, etc. So I'm just confirming that I'm not talking to myself. Thank you. Okay. If you cut with the restriction enzyme on the right side means sort of asymmetric with respect to the bubble. You get again a linear molecule with a bubble, but this time the bubble is asymmetric. Okay, it's not situated in the center, but to a side. Okay, these are all different kinds of molecules you can get when you isolate when you take the replicative intermediates and then digest with different restriction enzymes. But what we have to keep in mind is these are not uniform sets of molecules. If you cut with one restriction enzyme, you get this bubble, but it's not just one molecule. It is a series of molecules, a continuum of molecules with increasing bubble size. Okay, if you look at it, the bubble has all sorts of sizes. Okay, similarly, this fork here keeps on growing, keeps on moving. So any one restriction enzyme, you won't get one population of molecules, but a population of molecules that are of different kinds. So how do you analyze those? So this situation is explained more in the next slide. Some of you are probably familiar with this technique, but I need to explain in order not to lose the audience who are not familiar. So, okay, this kind of replicative intermediates are a challenge because when you run in a gel, there you get what you get is just a smear. Okay, because they're not only of different sizes, they're also of different shapes or conformations. So, for example, if you look at a situation like this, a Y-shaped molecule, it is first a unit length molecule which keeps growing and growing, and suddenly the replication fork traverses the end of the molecule, so you get unit size molecule again. Here it is twice the size of the unit size. But what happens is when the fork is moving, the molecule is not linear anymore. It starts assuming a conformation, a three-dimensional shape, and that delays its movement through the gels. And the same situation happens here in the bubble. Initially, it almost is like a linear molecule, but then the bubble keeps growing. So it is assuming a conformation. It is moving slowly through the gel. And when it ends, suddenly it becomes a linear molecule. An almost similar situation, the double Y, where the forks are coming from opposite sides. Linear, the shape is slowly increasing, twice the size, and suddenly unit size. This and this are almost the same, the way the size and shape are changing. These can be studied in a two-dimensional gel electrophoresis. When you first do the first dimension, agar gel electrophoresis, okay, normal agar gel electrophoresis. When you do the first dimension, they grow from unit size to twice the size. Okay, but everything in between, you get a big smear. Okay, and when you do a second dimension, they move differently. That is, the ones that are almost linear will move faster, and the ones with the conformations, adopted conformation at that particular, they will be delayed depending on their shape. So here, at this stage, this is the one that moves slowest. So this kind of molecules at the inflection point here. Okay. You can get this kind of a curve in a two-dimensional gel electrophoresis. Both are agar gel electrophoresis, but conditions are slightly different. Low percentage of agar, high percentage of agar, low voltage and high voltage, and that's about it. So that kind of conditions will exaggerate the movement based on the shape in the second dimension, both size and shape. So you get a curve like this, and in the bubble, it keeps on behaving like a high molecule. I mean, its movement is delayed and delayed and delayed. And at the end, when it crosses the end of the molecule, suddenly it falls apart. And then at this stage, it ends. And all the molecules will fall at this point, unit size. Okay. The situation is similar with this molecule also. But you get a different kind of curve because at this point, the conformations are different. Okay, at this point it moves slightly faster because it is probably assuming some kind of, it's not really exactly, but I can say some kind of a closed superhelical molecule that moves faster than the nicked molecules or the linear molecules. It is probably getting some kind of a shape that makes it moves faster. A priori, you cannot say it moves like this or this moves like this, but you have done the experiments, that's how it moves. So this is a special situation, the last one, very interesting situation, which will come later. The bubble is situated asymmetrically. So what happens? It starts as a bubble, keeps growing, and one side of it passes the end of the molecule. So now it suddenly becomes a Y-shaped molecule and behaves like a Y-shaped molecule. Okay, now you have two situations in the same gel. Okay, now you get a gel like this. Starts as a bubble, grows as a bubble, and then suddenly it becomes a Y. Y should look like this. Okay, suddenly it becomes a Y, and it will continue like this. So you get a discontinuous pattern in the gel. So these patterns are really suggestive of how the replicative intermediates look like. People have done this many times, and then this is confirmed many times. Okay. So now, what did we do with this with our experiment? What did we do with this technique in our experiment? So I will briefly explain. This is the SV40 on the left side, wild type. And this is the SV40 again, variant with the cloned insert. The wild type, if you cut with PST1, and SV40 has two PST1 sites nicely situated symmetrically, almost symmetrically from the terminus. So you have two PST1 sites here. So when you cut with PST1, you get a nice bubble, keeps increasing, grows as a bubble, finishes as a bubble, and then completes the replication. You get a nice bubble, an arc representing a bubble here. What happens in the mutant? You are expecting that on one side, the bubble grows very well, the fork travels very well, and on the other side, it is delayed. So what you get starts as a bubble and then turns into a Y-shaped molecule and finishes the replication. So now you would expect an arc representing a bubble and then a spot that is representing the Y-shaped molecule. The size of the arc and the size of the shape, size of the Y-shaped arc, that depends on how delayed is the replication. Okay. For example, if it stalls for more time, if it pauses for more time, you get more and more Ys. Okay, you'll start getting a Y right here. So it depends on where the discontinuity happened in the replication process. So so we did this experiment, and we also did some constructs like
different kinds of inserts. This time you have this SV40 wild type again and SV40 GA sequence cloned in it. This is a GA sequence with also some flanking sequences, and this is just plain GA sequence, and this is unrelated. I mean, it doesn't have any GA sequence. It is it's not random. It is taken from puck plasmid, and you have a control DNA here. So when we did this experiment with these constructs, this is what we got. Okay, these are the real curves, real experiment. So left side is the wild type, right side is the variant virus. In the wild type virus, you get a nice bubble. And in the variant virus, you get a bubble, but you also got this spur here at the bottom, as we expected. This is what we expected if there is a pause in the replication and we get a Y-shape. Pause in the replication, we should get a Y-shaped molecule at the end of its replication. So you get a short spur here, as expected. And when we do with other constructs, same thing again. This is the wild type, this is the control. Here you have the mutant, the variant virus, the altered virus. Okay, so we have seen that it is both completed molecules and replicative intermediates. Two different techniques totally to monitor those. So now there is further evidence, that is, the DNA replication can be influenced by the triple helical sequences in vitro also. Okay, single stranded DNA replication, not really replication, it is a primer extension experiment. Okay, you take single stranded DNA and do a primer extension experiment in vitro, the replication stops at the cloned unusual sequence. Okay, and that interruption can be eliminated by using 7 diazoguanines in the incorporating nucleotides. This is like a sequencing experiment. Okay, you have all the 4 nucleotides, but instead of using guanine, you are using 7 diazoguanine. Okay, and then try to sequence, and sequence goes like a charm. So it's not interrupted because that unusual sequence doesn't work with the 7 diazoguanines. Okay, and doesn't form a triple helical structure. Okay, and you can repeat the same experiment in a different way. Both are same thing, but you can use 7 diazoguanines in the template, not in the incorporating nucleotide mixture. But even then, have this 7 diazoguanine in that alternating guanosine adenosine stretch of DNA, and then again the sequence goes very well. The arrest is eliminated. And why is it? Why 7 diazoguanine is important? If you look at the Hookstein base pair one more time, see 7 diazoguanines interrupt triple helix formation. See, for this Hookstein base pairing, you need this nitrogen at the 7th position of the purine. Okay, if there is no purine in that 7 diazoguanine, that nitrogen is removed. Okay, when that nitrogen is not there, it doesn't make this bond. So you won't get triple helix 7 diazoguanines. So there is further evidence that in vitro single stranded DNA primer extension experiments also do the same thing. And how does a single stranded DNA form triple helix? It doesn't normally because it is a single stranded DNA. But when you are doing the primer extension experiment, this can happen. Okay, it is a single stranded template. You started with a primer. The primer keeps on growing.
And when it grows, the single strand suddenly sees the double-stranded DNA here and folds back. And then you have a triple helix being formed here. Once that happens, your primer extension stops here. You can't sequence anymore. Okay. So this is another proof that these sequences are influencing the replication fork movement or the synthesis of the nascent strand of the DNA in many different ways. They are causing an impediment in the replication. So the conclusion is that it influences, these sequences can interfere with the normal DNA replication, though it doesn't stop it completely and kill the cells. Now, okay, I have to switch gears here. Okay, I said that we have taken these sequences, putting in SV40 sequence, SV40 DNA, and then put them in the animal cells to see the replication. And in doing those experiments, what you do is, I said that we should look at the state of the methylation. So when you take a DNA and grow it in bacteria like E. coli, most of the strains that we use in the bacteria are DAM plus E. coli, DNA adenine methylase positive strains, most of them, unless you really want DAM minus strain for a particular experiment. When you grow them, your DNA is all methylated. Okay. Even if it is SV40 origin, eukaryotic replication origin, or anything, it gets methylated, DAM methylated. You isolate that DNA, and for your experiment, you put it in SV40, you put it in animal cells, like African green monkey kidney cells, or whatever, BSC1 cells, COS cells, whatever. You put them in those cells and allow them to replicate. What happens when these DNAs replicate in animal cells? They don't have DAM methylase. So they all get demethylated. So now you take the DNA and see if it is demethylated. If it is demethylated, it is replicating. The standard experiment, simple experiment, but that people do most of the time is digest with DPN1. If it is demethylated, DPN1 doesn't digest it. Okay, if there is resistance, that is indicative of the replication. So this slide is just to show the principle of DPN1 digestion to monitor DNA replication in eukaryotic cells, but it is not the real experiment. But you have a plasmid that is methylated, grown in bacteria, it is methylated, so it is digested by DPN1. Take that plasmid and grow it in a DAM minus strain of bacteria, E. coli. It is demethylated, so DPN doesn't work. You take this DNA, which is demethylated, and methylate in vitro using a DAM methylase. Again, it is digested very well. So the method works beautifully. You take SV40 DNA, which has grown only in animal cells and has not seen the bacterial cells, and digest it, DPN1 doesn't cut it because it is demethylated. So far, so good. DPN1 is used to monitor replication, and it works fine. But there is a problem. The problem is, for reasons that are not known, because there are too many variables in biological systems, there is always some DPN1 resistance seen in these experiments. Okay, that is not related to replication. You put anything in animal cells, take it, and then digest with DPN1. Occasionally you get DPN1 resistance, which can be confused with real replication. Okay. And okay, so when we were looking at various plasmids and sequences in a related project, we are looking at a lot of sequences, sequencing so many, and looking at sequence chromatograms, which a lot of you are familiar with. like this one. This is an example of a sequence chromatogram. We were doing a lot of these sequencings for a number of projects, including SV40 and DNA replication. At that time, when we kept looking at these chromatograms, we noticed one thing. One thing that is, you can see that the peaks are, this is a standard chromatogram, which using dye-terminator chemistry, where the peaks are not uniform as expected, with different heights, and with the sequence nicely printed at the top of these peaks. So when you keep looking at these chromatograms, you sort of start expecting the pattern of what kind of peaks you can get in what sequence, because these peak height, these are actually dideoxy terminator incorporations, which are sequence dependent. Okay, the sequence sort of influences them, but we don't care about that. We are seeing a good peak, you know, if it is a big peak, it's supposed to be that way. If it is a short peak, it is supposed to be that way. Nothing wrong, happy, we are getting those things. If it is a diprimer sequencing, for example, the peaks will be more uniform. Still, there are minor differences, but dye-terminator chemistry is the common one that is used. So you get all these peaks. When we looked at these peaks, what we saw is the DPN1 enzyme recognizes the sequence GATC. And in that sequence, the T peak is different. It is unusual again. Okay. This is what we saw. This is the DNA sequence chromatogram of the region comprising the DAM methylation site GATC. The T peak is strong. Okay. Normally, the T after A is not that strong, or in other contexts. So this is pretty strong. And suddenly we realize that, okay, it is strong in this GATC sequence. Why is it strong? We know that in the opposite strand, it is GATC, and the A is methylated. The adenine is N6 methylated in GATC sequence. Is that what is affecting this incorporation? That is easy to verify. Okay, now take this sequence and demethylate it. As simple as that. You can grow it, you can take the same sequence, grow it in DAM minus E. coli, it's demethylated, do the sequence again. Okay, now the T peak goes down. Okay, now to confirm it further, take that demethylated plasmid and then again remethylate it using DAM methylase. The peak comes back up again. Okay, and you take SV40 DNA, which is demethylated, and sequence it. This is how you get GATC, T goes down. So it's very clear that N6 adenine methylation can be easily detected by normal sequencing. Sequence chromatograms will show you which is methylated, which adenine is methylated or not. You will see the peaks in the nascent strand. That means the template strand has adenine methylation. Okay. Now you also have other kinds of methylations in DNA. 5-methylcytosine, N6-methyladenine is what we have seen just now, and also another rare one, which is N4-methylcytosine. So we thought let us look at 5-methylcytosine. Can we detect this? Did a similar experiment using the DCM methylation site, CCAGG. Okay, it is actually CCWGG because this A can be A or T. Okay, we took a DNA like that, and then let us start here. The methylated one. The cytosine is methylated. The internal cytosine is methylated. It's a 5-methylcytosine. So you have CCAGG here, and in the methylated one here, okay, the G is weak. Okay. And then if you take SV40 DNA, it also has this site. So the G peak is very strong. It is demethylated. The opposite, the cytosine in the opposite strand is demethylated in SV40 DNA. So you have a strong peak. Methylation, weak peak. And then you take this methylated DNA and then remethylate it. No, sorry. You take this you take this DCM methylated one and then grow it in DCM minus 1. So it is demethylated like SV40. So the peak comes back up again. The same thing happens when you look at the CCTGG. I have to repeat these experiments to confirm because it is a good claim. So CCTGG. So you have the same thing. Demethylated plasmid, strong peak. Methylated plasmid, weak one. Demethylated plasmid, strong peak, like SV40. So you can detect 5-methyl cytosine in this context. Okay. Then what we did is, okay, CCTGG, but then we also know that there are a lot of 5-methyl cytosines in the chromosome or DNA, which are HAPA2 methylation sites. So let us look at that one, which is CCGG. Okay, CCGG. So CCGG also very similar situation. You have the cytosine methylated. I'm getting a warning here. Okay, I don't know. Okay, CCGG is also same kind of situation. You have methylation and then in the opposite strand. So when you are sequencing CCGG, you have CCGG in the opposite strand. So if you take SV40 DNA, it is not methylated, do the sequencing reaction, you get a strong G. Okay. And then you take the same DNA, in this case, in this experiment, SV40 viral DNA, and then demethylate it. No. This is demethylated. You methylate it with HAPA2 methylase, and then the peak goes down because the cytosine in the opposite strand is methylated again. So now you have this HAPA2 methylation site, EcoR2 methylation site, and all these, BPN1 and all those. Now, there are also lots of 5-methyl cytosines which are not part of a restriction enzyme, like all the CPGs with methyl cytosine. Can we pick up those? Because they also have the sequence context there. So we wanted to check some CPG methylations. Okay. This is the DNA sequence chromatogram of a motif CGG. Okay. Again, here in CGG, you have CGG, you have CCG here, and the cytosine there is methylated again. So if it is unmethylated in this sequence, you have a strong peak. If you take that and methylate it using SSS1 methylase, which is known to methylate all CPG sites in the DNA. So you can easily methylate it again with another enzyme, though it's not part of a restriction modification system restriction site. Okay. So you can isolate that with SSS1 methylase. When you methylate it, again the GP goes down. So all these things tell us that by a simple sequence chromatogram, if you look at the peaks really well, you can pick up, N4 also can pick up, we can pick up like this. Okay, I didn't show all that, too many. So, okay, so there are mainly these are the 3 methylations you see everywhere. So all 3 methylations can be easily picked up by a simple sequencing chromatogram. Okay. But one thing we have to keep in mind is, our important thing is, in all the other methods you have to, in some other methods like you may have to treat the DNA with some chemicals and all that. Like sodium bisulfite, to pick up methylations. Some of them, not all, not all. So here, you just take the DNA and sequence it straight. No prior manipulation or treatment of the DNA. In fact, you should not do any prior manipulations, because if you take the DNA from an animal cell and then grow it in bacteria, all your original methylation state is lost. So don't amplify in bacteria. Similarly, a common practice is, when you don't have enough DNA, do PCR amplification. Okay, no PCR amplification. Once you do PCR amplification, your methylations are lost again. So directly sequence the DNA as it is isolated from its original source. Your sequencing is PCR different, no problem. That is a different story. But original DNA, don't PCR amplify. Okay, all these sequence chromatograms are coming from PCR cycle sequencing, but that doesn't amplify your template DNA. You don't use 2 primers in the proper, so that won't happen. But all you are basically looking at the nascent strand in that experiment. So don't PCR amplify your DNA, so it won't be affected. Okay, when we reported this one, a lot of people like this idea because it is so simple. It appears so easy. You don't do anything, just do a sequencing run, which takes only a few hours. Okay, so people started repeating this in a number of their experiments. Our interest is eukaryotic DNA replication. So we monitored DNA replication using this technique and we were okay. But others, there are a lot of people who use this in their experiments. And most of them are bacteriologists. Okay, people work in the bacteriology field, people work on their methylases and this kind of things all the time. So they picked it up and started using this method many times. And here are some random examples I picked up from literature in Bun Hl area, Pseudomonas, Caldi cellulosiraptor species, YHDJ gene in E. coli, and genomic DNA can be directly sequenced and all the methylations can be seen. This is another methyl transfer. This is another system where, and when they are looking at these methylations, they also came up with a connection between the methylations and the virulence or survival in the host tissues. So these are just a few examples that people are using this technique and are happy about it. So now I'm coming to the end of the talk. I don't know how much time I spent so far, but this is the summary of what all I presented. The first part is DNA replication fork movement in SV40 paused at unusual sequences, both in vivo and in vitro. Growth, plaque size, and titers of SV40 DNA are affected. Pause in completed molecules is evident by pulse labeling. Pause in replicative intermediates is evident by Brewer and Fangman analysis, which is the same as the 2-dimensional gel electrophorese that I was explaining, which was first reported by Brewer and Fangman. Pause eliminated when triplex formation is interrupted by 70 azaguanins. And adenine and cytosine methylations are detected by sequence tracing comparisons in automated DNA sequencing. It can be just intra-sequence comparison. It doesn't have to be even inter-sequence. Okay. Inter-sequence comparison also is fine depending on your experiment. The method is highly reproducible and used in many laboratories. So that's where I come to the end of my talk. So I'd like to answer any questions if there are any.
Thank you, sir.
Thank you very much
for your lucid presentation on role of homopurines and homopyrimidines sequence DNA replication. So now the session is open for discussion. If any quick questions are there, the audience can ask questions. Sir, do these sequences have any role in regulation of gene expression this SP40 virus, or what could be the biological significance of these unusual sequences present in, I mean, these homopurine and homopyrimidine sequences?
A good question. The one experiment that we are looking at is also a biological role. Okay, the DNA replication is where we are studying its biological role, what biological role the sequence can have. And there are many others who are working on these this kind of sequences, and it affects the transcription in some situations. And as I said, it is associated with many diseases where the triple helical sequence in the normal DNA, which is not uncommon, you can easily pick up the a single stranded portion of these triple helical sequences using S1 nuclease. People have known that for a very long time, and they are known in all hotspots of recombination, repair, chromosomal translocations, cancer, and all those things, and also in neurological diseases. And whenever that happens, these are sort of unstable. Okay, as long as they are intact, okay, the sequence, even if it forms a triple helical structure, as long as it is intact, the cell is okay. You are okay, but because of the inherent instability and the mutagenic potential of these sequences, something can go wrong very easily, and that's when it disrupts the functions of the normal cell, and you get. That's why it is shown up. It is showing up in a lot of biological, I mean, lot of diseases. Okay, people, you can exploit that also because you have a sequence in very crucial points, like for example, 5' of the c-MYC gene, there is a hotspot for these things. So what you can do is you can manipulate those sequences using this triplex forming technology. That means I didn't cover those things, but you can take a triplex forming oligonucleotide. Okay, under right conditions, you can add a triplex forming oligonucleotide and you make a triple helix yourself in order to control the function of that particular gene. So people have done that. So these are basically the sequences that are of interest. Using the therapeutics is still in infancy, but people have tried that to a lot of extent.
Sir, we know that the dideoxynucleotides are potential therapeutic agents to inhibit the replication of several pathogens, including viruses. Can we make use of these unusual sequences for the same purpose, or it has any therapeutic applications? Already
Yes,
said eukaryotic systems possess these unusual sequences much as that of the eukaryotic systems.
The dideoxynucleotides, by nature, are supposed to inhibit DNA replication. That's why we use them in sequencing. Okay, you can use them as therapeutic agents depending on where you are using and where you don't disrupt the normal DNA replication. You can use them as highly toxic also, but depends on the experiment and depending on the application, they can be used. These sequences, which I am saying, the triple helical sequences, their role in inhibiting replication is limited. Okay, because these are, most of them are they are naturally, most of the time we are dealing with naturally occurring HDNA. HDNA means triple helical DNA, naturally occurring one. So you cannot add one except in a very isolated system. And you are targeting something, targeting a protein or whatever. So you cannot give them as like a therapeutic agent. Okay. If you give it as a therapeutic agent, what can happen? Nothing, because it has to be sequence specific. If it is an unusual sequence like that, probably there is already a triple helix. So you are only adding a fourth strand, which will do more damage. Then there is very little that can do. Okay, but as I said, if you know that there is a sequence that has a potential to form a triple helix but is not forming, you add a triplex forming oligonucleotide. Okay, you supply a triplex forming oligonucleotide that can go there and bind it and inhibit or regulate the function of that particular protein in context.
Thank you, sir. Thank you very much. If in the options of any more questions, further questions, I thank the speaker for his excellent, lucid presentation. And also I thank the Dr. Subhashini Devi and the others for inviting me to this Golden Jubilee celebrations of Department of Biochemistry, Andhra University, and also for giving me this opportunity of chairing this session. Thank you very much. Now over to Dr. Subhashini Devi.
Thank you, Govindra sir, for chairing the session.
Thank you, madam. Thank you very much.
Thank you very much, sir. Sir, thank you, Sridhar Rao, sir.
Oh, thank you very much.
Sir, Uday Bhaskar sir, are you there? Now over.
Yes, Dr. Subhashini.
Yes, sir. We planned alumni meet online, actually 5 to 7, but I think it's 6 till 7. We'll continue the meet, sir.
Absolutely no issue. No issues.
Because the link is active till 7 o'clock.
Okay.
We'll continue the meet till 7 o'clock. I request you to coordinate the alumni meet. Meanwhile, Bhaskar Reddy sir will join, sir.
Yeah, Bhaskar Reddy has already joined, I suppose. He's already joined. Yes. Thank you, Dr. Subhashini,
Yes.
for this initiative you have taken up initially. Thank you, Sridhar. Nice of you
Thank
to
you very
present.
much.
Yeah.
Oh, thank you very much.
Sridhar is my classmate, so I take liberty in talking to him. Yeah. Now, I think I am seeing about 38 participants on board. And I think without wasting much of time in talking ourselves, I open the forum for discussion by the alumni members mostly. I just give you a small introduction of what has happened yesterday. Maybe perhaps all the persons who are now presently online must have already seen yesterday's inauguration of the Golden Jubilee celebrations, very aptly organized by the faculty of our alma mater under the stewardship of Dr. Subhashini Devi as HOD, supported by Professor Padmajyoti Hamalata, Professor Raghav, Professor Radhika, Dr. Aparanji, and all these other members. Now, yesterday, it was a wonderful thing. Many of you must have noticed the Vice Chancellor of Andhra University, Professor Prasad Reddy. He has spoken very well with regard to the opportunities for the students and what opportunities the university is providing, and what exactly the role of faculty and the role of the students in upgrading themselves with the present trends around, so on and so forth. So it was a very well articulated and material filled address, I suppose. I feel it. And it was acclaimed by many people also who were present yesterday. And the issues actually thrown for the discussion before the alumni, they have certain issues already sent to me by Professor Raghav. Before taking up, I request Dr. Subhashini as HOD or Professor Radhika, whoever is here, online. Kindly please illustrate during past 4 or 5 years, we have been little bit active on this front. So what actually you are anticipating from the alumni, and so far what efforts have yielded some result in benefiting the department from the alumni support. So if you can little bit illustrate, the members can at least, alumni can feel happy with the support that is being extended by many alumni, mostly from the academic side, from abroad, and also in India, our friends, our bi-ministry alumni friends. So Dr. Subhashini or Professor Radhika, whoever, Professor Raghav, kindly please take initiative and please come forward to tell the persons online here and what exactly you want. Because after all, people are busy in their own front. Ultimately, whoever tries to help the, students or faculty, there shall be a channel. So please speak out and what exactly you have in mind, what kind of support you are anticipating from the alumni. And once you have you people speak out, then the alumni members can always ventilate their opinions, ventilate their ideas, ventilate the nature of support they can extend in any way. It's not a compartment. It's not a watertight compartment. Kindly please, Dr. Subhashini, please take up.
Yeah, thank you, sir. Good evening, dear alumni members. For the past 3-4 years, not only for the past 3-4 years, actually the department is active during the last
Your audio is a little problem.
Madamji, you are not audible to us.
Dr. Subhashini, your audio.
Subhashini, audio is not, you are not audible. Subhashini Devi.
Mute chesara.
Subhashini, you are not audible. Anything problem?
I think a message can go to her. Raghav, Professor Raghav.
Yeah, I sent the message to her, sir.
Yeah. Meanwhile, shall we go on?
Yeah. Sir, am I audible?
Yeah, you are audible. Bhaskar Reddy, are you trying to say something?
Should I speak, sir? Should I speak? Yeah. Sir, I would like to speak.
Yeah, please.
Right. Good evening. Warm greetings to all the members of alumni. I would like to thank each and every one for your active participation and encouragement rendered in connection with conducting Golden Jubilee celebrations. In fact, without your support, it would be difficult for us to conduct events in the department. Your moral and scientific contributions in terms of lectures delivered for the past 2 days and earlier are appreciable. In fact, your presence with us is an inspiration not only to us but also to the young students in the department. I hope the same will be extended in the days to come. During the past few years, after Uday Bhaskar sir appointed as president of Alumni Association and Bhaskar Reddy sir appointed as vice president of the association, the department geared up and then tried to conduct some kind of activity in the department with the support of alumni members. I'm really thankful to all the alumni members who are in abroad, especially. Whenever we ask them to give a lecture or contribute some kind of support, they are readily doing it in an exemplary way, and we are very happy for that. In fact, as Uday Bhaskar sir said, we don't basically expect any financial commitment till today, because whatever the financial support we have, we get from the university, we are doing it. And we only request a few aspects. One important thing is awareness among students about the career opportunities available in India and abroad. Definitely, we try to inculcate scientific activity in the department with the information that is available with us. And those who are working in better laboratories and better places, definitely opportunities will be more in their place. So I hope and I expect some kind of career opportunities which are available in and around the alumni members. If they are extended to the students of biochemistry, definitely students will get benefited, and it will be they will also add to the department and continue the same to the next generations, and it will become our association will become very strengthened in terms of number, in terms of activities, so on and so forth. And apart from that, progress of the department, I think you have been witnessing time to time. With the faculty existing and with the support what we have, we're trying to conduct different activities in the department and inculcate scientific temper in the students who are joining in the department. And we are lagging behind one particular aspect, that is placements to our students, right? Most of them are pursuing higher education, and then what I observed is many of the students think joining in pharma industry and other sectors, and many of them are joining in other fields also. Apart from that, quite a few or a couple of students from each batch, I think, are unable to get any kind of placements. I think if any kind of such cooperation comes from the alumni in terms of placements, providing placements, or suggesting some kind of measures to enhance the placements in the department, I'm sure it will be an encouraging thing to the students, And as an alumni of this department, I think we can contribute to the family of Biochemistry Association in a greater extent. These are the two things that I just forwarded to President to discuss extensively, so that if any opportunity is there in your lab or in your place, in and around your working place, if you inform it to the department or to the any one of the staff members, it would be a great help to extend the support or extend the information to the students. So these are the few words that I would like to share with you. Thank you, Uday Bhaskar sir, for giving me this opportunity. And I'm sure clear in my words, I hope, and expect same support from you all to conduct many more events like these, which we have been conducting today. Thank you very much. Uday Bhaskar garu.
Thank you, Raghu Garu. Very nice of you. You have consolidated issues. Now, Bhaskar Reddy, you want to say something?
Sir, before that, I mean, if anybody if somebody wanted to introduce, they can introduce some
That's
new.
better. Yeah, correct. Whoever online, I find Dr. Sivaprasad Rao Garu, Subramaniam is there, and many other alumni are present.
One by one,
I think,
suggestions.
kindly please introduce and then open your minds and open up for the benefit of the alumni, faculty, and other people. Yeah, Bhaskar.
So good evening to one and all. So this is Dr. Bhaskar Reddy, so Vice President of the Alumni Association of Biochemistry Department. So I want to introduce myself. I did my MSc in 1977 and 79. So later I completed my PhD there. I joined in Githam. I worked there for a long time. So most of the people knows me, of course. But anyway, very nice to talk to you all. Professor Sivaprasad Garu, Uday Bhaskar, Raghu Rao. So I'm very happy to see the Golden Jubilee is going in a very nice way. But I'm missing the opportunity because I am away from that one. I could not attend, otherwise I could have been there. So sorry for that one. So nothing to say much actually. Regularly we are meeting there. So I need some comments from the outside experts. So which about their feelings regarding the Alumni Association. Thank you very much for this opportunity.
Sir, Sivaprasad Rao Masharu, Mirunnaru. I think you're online. Dr. Subramaniam Garu.
Yeah, thank you, Devaskar.
Please.
One thing one thing I've noticed is in the last for a long time is that if you give any critical comment, people take it personally, and that prevents anybody coming and doing things for the.
Can you please avoid all those things? Can you come to the subject?
Yeah, I have to, because when we are open-minded and take criticisms positively, that's how I grew up in my career. My professors, my postdoc advisors, everybody, they criticized me and I took it positively. And that's what put me up here today, what I'm doing it, right? I mean, I've tried to help I've probably taken about 8 to 10 students in 2008 to 2012. Most of them did extremely well. Many of them are in the US working for big companies. But to give that kind of an opportunity, it's not easy for anybody. For me, for example, when I did my master's, it was actually grilling. MSc, I joined as an MS student, not a PhD student in Canada. It was a grilling experience. You have to have a thesis in a master's level, and you need to have a professor who has published 100 papers, 200 papers. All these professors there are PhD in big universities, Oxford, Toronto, Berkeley, all kinds of big universities, work for Nobel laureates. Come there after postdoctoral fellowship, 3 to 5 years, they establish their own careers there as professors with all that experience, and they have published probably 100 papers by then. They are the ones who are leading the department as professors, training the students to the betterment. I'm not saying anything against the current teachers, but that is kind of a recruitment I have seen for any assistant professor that has been recruited in any university I've been to University of Colorado, University of California, Berkeley, University of Toronto, Memorial University of Canada, all these places, they recruit people with PhD, 5 years postdoctoral experience with at least minimum 25 to 30 publications and reputed journal, well-cited publications. We don't have that opportunity in Andhra University for various reasons. So with that, the problem is to give opportunities with somebody who doesn't even know how to pipette. It doesn't know what is 10 microliters or 20 microliters. I'm spending my time 1 year to train these people in my own lab is not something worth my time because I'm ready to take people who are already trained, and that helps me to move my projects faster. So something regarding to that, if university comes up with some plans, I mean, I'm ready to talk to when Vice Chancellor, if that opportunity is given to me to do that, because we have seen the world out there. So if we don't give the opportunity, I mean, our opinions and what we have seen, and that has to be taken positively. That I've done something. So what? You're arrogant. You should not do that. You have to take it positively and implement those ideas into the system. So for the young students right now who are graduating, you need to have a thesis projects, maybe in the biochemistry department or cross department like biotechnology or pharmacy, or these are all interrelated fields. I mean, I became a pharmaceutical scientist at the end. So that's how you need to give an opportunity to see what they can do, at least in the experimental part of it. 6 months at thesis, not just a dissertation, reading papers and writing something. They have to do a lab work project. And that's my opinion. If that in the students, and most people will go to bank jobs or other jobs which are not related to biochemistry. 2 years of MSc biochemistry should be productive in biochemistry field, not other areas. That's my points.
I speak?
Hello?
Yeah.
Okay. I think partly I can echo the sentiments expressed by Subramaniam. I have seen both worlds, how things work back home in India and over here. And one of the challenges that I had to face when I came to this country was I was not very independent thinker. I thought I was good at it. And you keep getting criticism from people, and you need to respond to it positively. You can't take it negatively. I still remember one of the comments which changed me. Sorry for the anecdotes, but I think I should probably say this. When I was in class 5 in a Jilla Parishad High School in north of Andhra Pradesh, a place called Sompeta, my teacher asked me one question and I couldn't answer it. And his comment was, Kakarkaya, Kekarkaya avutundi annadu. And that has actually affected me. I think that has made me think carefully where I'm going wrong. I need to change the way I'm approaching my career or my life or whatever. But that I don't think I took as a negative, and I still think that was the best comment that I ever had in my life in terms of the criticism. But the issue is, it is a cultural difference. In the West, people invite constructive criticism, even sometimes worse criticism. When you write a paper, for example, it comes up with such kind of comments, you really can't sleep overnight. It actually affects you very badly. But you need to answer them, you need to respond to them. So although it affects you initially, although it feels bitter, I think we need to respond to it. But I think I would like to stop there. I don't want to comment further on that. But I think it is a cultural difference. In India, we're not used to accepting criticism that easily, right from the top to the bottom. In the West, people invite criticism so they can improve on it, take it. There is a cultural difference. I think it needs a change in the mindset. In terms of what we can do for the department, I think that's a difficult thing to answer, largely because we have lost touch with what is going on in the biochemistry department, Andhra University, in terms of what sort of skills the students are developing, offered, whether they are ready to go into the rest of the world and face. Because, like Subramanian said, people who want to hire them don't want to teach them because they think they're already being taught. So to be able to make objective contributions to the curriculum and how it is being delivered and what sort of skills are being offered to the students. Obviously, people like me need to know what's going on there. At the moment, I don't have that knowledge to be able to make any kind of objective assessment of how things are going on over there. So I cannot comment on that, unfortunately. But what the management, as well as when I'm talking about the management, means from the university side, from the top to the bottom, need to think about is get advice from the people who are going to employ them in the future, what sort of skills they need. So identify those skills, and then you can alter your curriculum in such a way that matches with those skills. You need to align those things. And so when you make an application to somewhere, when your student makes an application to some place, he needs to be able to state in his CV, these are the skills I have, these are the skills you need, so I can confidently say, I can come and work in your lab or in your organization, whatever. Those skills need not necessarily be academic skills. They could be other skills, transferable skills. It could be the presentation skills, whatever. So I think academic is one side. There are other aspects as well, are crucial when you want to progress in life. So my answers, unfortunately, are rather vague to the questions, largely because I lost touch with the curriculum development and what skills are being offered to the students. If I can have some of those things, if I can have more information, maybe I'll be able to comment on that. But I think people like Subramanian, there are a number of people who spoke. I think one of the things I forgot to say is I must congratulate the organizers for inviting good speakers. I'm glad that I haven't given a talk because there's a good opportunity for me to listen to what our alumni are actually doing. It is very impressive. So those are the people who spoke today, for example, could be able to advise you. They're much younger and they're working in different industry as well as academia. So you can take opinions and take advice from people working in different sectors, healthcare sector or academia or wherever. and then rejiggle your curriculum. I'm sorry, it looks like it is crashing a bit.
Your voice is not coming, sir.
I can't hear you, sir. Prasad Garu. I cannot hear you well.
There is a background noise coming from somewhere. Probably it's Bhaskar Reddy.
Bhaskar Reddy, can you mute your audio? Yeah, I did. Okay, go ahead, Prasad Garu.
Sorry, I think I have come to an end. So I think I leave it to other people to speak and contribute. But I think some of you don't know me, but I was a student of Andhra University from 1973 to 75. And in 1976, I was appointed as a lecturer in Andhra University. I worked for several years, taught many students over that time, and then came to UK for doing a PhD, came back, then returned back again, and since then I'm in Leeds. So my background is mostly biochemistry. And I think when I came to this country, when I was doing my PhD, my supervisor said, you know a lot more biochemistry than most people that are doing PhD and postdocs then. So that speaks volumes. The type of curriculum that we were offering at that time was contemporary, and we could sell it to the rest of the world. What changes have taken place since then, I do not know to be able to comment on that. Okay, can I leave it to others?
Yeah, Sridhar and Sudha also should speak a little bit. I think they also have good experience to be able to.
I raised my hand actually because I also wanted to add a few words. Thanks everyone. I'm totally agreement with what you all have said. Raghu Garu, to your point about placements, I think the first thing is I want to say there are enough jobs in India. Post-COVID, this has become a very, very important area to work on. I'm working on vaccines, and I'm sure there are a lot of therapeutics, and we all know that the healthcare industry will always need people. And so I think there are a lot of jobs in India. So people should not think we have to get out of India. That's the first thing I want to say. I did my PhD in biochemistry, Andhra University. I did not go out anywhere. I was working in India till 2016. I had a pretty decent job and well settled. I just uprooted myself and came to the US for like a crazy person. But I think there are good research, good work can be done within India. Coming to your point on jobs and coming to Dr. Sivaprasad Rao Garu's comment on curriculum, I think that's why I had in my notes, the first point was the curriculum. While I'm not updated with what is the latest, I do think there is a need for the students to have an exposure to projects in their masters. I'm not sure if it is included right now, at least when the last I came, there was nothing like including a project in the master's curriculum. I think that is the time. I mean, here, if you look at any university, even within India, most universities abroad, they have a project in masters which make them go to industry or to another lab to do some research work, which is kind of not just reading from the book, get them to have a hands-on experience. And it can go to 6 months. You don't have to have it for 1 month, 3 months. I like that to be 6 months. So somewhere a master's curriculum that has a strong hands-on experience. There is a risk that students go attend every day, finally get a certificate and some dissertation. That's not what we want. We want them to have a complete interactive learning experience. So if somewhere we can build that in the curriculum, that will help. It also helps the faculty to have this exchange with maybe industry or other universities, academia, foundations, wherever, through the students. So I think that will be a good proposal that I bring on the table. Secondly was about spending time with students. I actually offered myself to Hemlata madam that I can spend time, volunteer to, spend time with students to probably mentor them. And it's a stage where they may be confused, they may be having doubts, they may be on themselves, on the system. It's very easy to blame system sometimes and everything that's around them. So I think if somebody with an experience, and if I think I can contribute on mentoring, there may be many like me who are interested to spend time with students and just give them that moral boost. I think that is very helpful. At some times in my career, I felt I needed that. I probably stumbled to learn a lot of things, and maybe that we can avoid by sharing our own experiences with students. I have myself contributed in some networking sessions with young ladies who want to get into industry and have to deal with, a lot of, I won't say social issues, but how to deal with working with at home as well as working at workplace. Some small things, but it's really very meaningful. I've done that. So I think if any mentoring on any aspects that can be included, you could have some people, ad hoc members in assigned to university, and students can reach out and say, I want to learn from you people. Another point what I wanted to say, and it's probably echoing what I heard already, is bring a change management. And when I'm talking about change management, it is about taking positively the feedback positively. I think that's important. I echo on that. I'm not necessarily saying criticism, but just being more open-minded, learning that there are different ways of doing the same thing, and maybe our university has one way, but there are other ways that can also be followed. So I think it's a change management, right from faculty to students to the facility within at Andhra University Biochemistry. I think the labs need a facelift, and I think people need to be more involved in that, and that mindset change that this is mine. This is not just what I do for a year or two, spend in the department and get out, but more if getting involved, that this is mine, and that kind of change in mindset that we'll need to bring. And I'm not having ideas right now, but maybe we all can sit together and say how to bring the change in there. I think that's the 3 points or 4 points I wanted to share from my side.
Okay, can I continue a little bit today? Oscar, or somebody else wants to speak?
Yes,
Murali and Shivakumar have excelled in academia here in India. They should also contribute to some of their experiences and how students can benefit from them. Okay.
Yeah, Subramaniam, just before you continue, just let me just emphasize on what actually the university is doing. As I understand from yesterday's speech of Vice Chancellor, there is a lot of student interested activities the university seems has taken up, like the integration with pharma companies, and also the promotion of startups. The idea, he says, the idea should be generated from the student side. So he says idea is the wealth. So like that, there are many good things they are planning. Even the principal has said the skill development centers established in the university, they are imparting good training to the students. So I request the HOD, Dr. Subhashini, or Professor Raghava, or any other faculty member to relate to the talk of Vice Chancellor with the comments made by our own alumni who have settled well abroad and also in India. and who have their own set of ideas with reference to curriculum or with reference to promotion of students' interest, etc. So how do you relate your university situation with reference to their understanding and how to go forward?
Yeah, with respect to the training, as far as in
Not clear,
but
Murali.
unfortunately,
welcome.
Okay. Am I audible? Am I
Yeah, you're
audible?
not
Yes,
visible
yes. Now you're okay. Yeah, and your face is only half.
Half.
Put your full, yeah. Put your full face up.
See, as far as theoretical knowledge is concerned, I don't find any limitations with respect to most of the students, because Andhra University is having good teachers who can teach the theory very well. But unfortunately, the amount of practical knowledge which you are going to give to individual students is not going to be easy, because the amount of funding which is available to teach different techniques individually and make them understand and make them to do is the a very difficult thing. Most of us, we got training when we went to PhD, we learned many things there itself, but we are not having that practical knowledge. That is the problem, I think, in India. In abroad, that is not the thing. Even in Germany, they do 1 year coursework during post-graduation, and they are very well versed with various methodologies, be it in enzymology, be it in molecular biology, be it in endocrinology, be it in carbohydrate chemistry, or be it in metabolic aspects. See, the setup in abroad is entirely different. Sometimes I feel that it is unfair to compare our Indian conditions with foreign conditions, because we should not always think about Stanford and Harvard. That is fine. As far as standards are concerned, we should not think that Nobel Prize. What I feel is that at a ground level, we have to think which are the best laboratories that are available in India as far as biochemistry is concerned with respect to the practical knowledge. Is it Pondicherry, Jippar is there, Pune is there, and Madurai Kamaraj University is there? What exactly they are teaching in the practical things? Because if somebody can visit and where exactly Andhra University is lacking as far as practical knowledge is concerned, if you are going to teach the people the good amount of practicals, and think how many people are able to do SGS page in the laboratory when they do their MSc, all these things definitely count. I feel most of us, I think it is, I will accept that most of us, we have learned practical things only when we are doing PhD, not during MSc. That is a ground reality. We have to accept it. We should not compare with Germany or Japan or America, where the things are different, and the amount of money they get for all these practical things, rather conducting practicals or dissertation, is entirely a different proposition. This is my opinion, and maybe you have to convince the vice chancellors to give extra funding for teaching these practical aspects for the students. That's what I feel.
Yeah, Murali, what you said is fine, correct also, because you get surprised to know what is the university's financial support to the department per annum. I don't know, Subhash Garu may say, but it is, what I came to understand is, it is just 50,000 per year. What kind of chemicals you can buy? What kind of apparatus you can buy in that? So this is the state of affairs in the department. So it is not in the faculty's hands. It's not in the faculty hands, because they have to go and then plead the vice chancellor or registrar or principal to support their cause. But at the most, the funding may go up. I heard previous years, maybe around 1,50,000 or so. That has come down. Drastically, it has come down. The faculty is somehow managing a show there. But as you said, there is immense importance to the practical knowledge of any student. So this is the background with which what I am asking the alumni with, with their wide knowledge and exposure and experience, kindly suggest the faculty what exactly, because they may be shying themselves. 1 minute, please. They are not able to come out what exactly is the problem, what exactly they want. So if there is such frequent interaction from the faculty and the alumni who have settled well in their biochemistry profession, absolutely there can be a way out integrating with the university's approach, because that is also essential. Sitting here, you can't talk of something else. Somewhere, some wonders are going fine, but they are all for learning purpose. But for implementing here in the Andhra University, during the studentship, what exactly the faculty has to do, a guidance has to go. Start kicking at curriculum is okay, but practical knowledge is absolutely essential. As a basic MSc student, I'm stating, but it is becoming difficult for the faculty to organize. They have the limitations, not many limitations. So how to come out of it? Subhashini Garu, Raghava Garu, Radhika, all these people, you have to speak out. Kindly please tell these and our own friends sitting in a very advanced situations. So please take advantage of their suggestions, interpolating with your situation, carrying forward the interest of the university also. So how do you want to make it up? Please go ahead.
Yeah, Dr. Uday Bhaskar, can I mention
Please.
few more sentences? Since I was associated with CFTRA for the past several years, every year there are 200 MSc or MTech students. They come and do dissertation for 6 months and learn the things, and they will submit as a dissertation. And many of the people, of course, they learn good things, and they invariably go abroad for PhD fellowships also. What I feel is that if the facilities are lacking in my Andhra University, how about having a dissertation at least for 3 months elsewhere?
No, that's what I'm saying. Yesterday, the Vice Chancellor was saying Aurobindo Labs and some other pharma industries, they are taking these people for project work for 6 months. It seems they are doing. So you have to take it forward as you suggested. If not local industry, any student, last time Subramaniam wanted some people to be sent, but he wanted a fixed period, maybe 3 months minimum. He said they have to spend. But problem is their financial support is not there. He can provide you the logistics, working and other things. He can't just, any sponsoring alumni cannot provide scholarship or otherwise the sustenance issues. So that's kind of, there are issues. So that we have to amicably resolve. There is no need of shying here in the alumni group. This is a group, internal group. Whatever requirements you have, whatever difficulties you have, Subhashini Garu, because now you are the HOD, kindly please ventilate, because they will understand. They will understand and they will give you a way out also. Professor Sivaprasad also says, so yeah, he has no knowledge of what is going on in Andhra University about the curriculum. At that time, some curriculum had been sent. With the curriculum would have been changed with the support of few more alumni, like Subramaniam, Murali, all these people were commenting in those days. So some kind of insight into the technicalities and the academics and some advancement in our approach, definitely we can do something, because there are lot many, well-educated, well-settled alumni here in the front of biochemistry. Please take advantage of it. Subhashini garu, cheppandi. Murali garu, meere cheppalani kuda cheppandi.
Sir, this is Dr. Rao from NIHFW New Delhi. Okay.
Yeah,
I think I am little late because I could not had my lunch, so I came back after my lecture. So I was just having, and I got a call from Raghava, and then I joined. So I don't know the head, but I could understand the last two comments that how actually the practical point of the things can be improved at the Department of Biochemistry. That's what I understand. Is that so? If that is the case, as actually Dr. Murali Krishna was saying, that need not be sometimes, let me tell you, from in our institute also, there are many universities which are like a private and the public also. They do come here for the even bachelor degree, master degree also. They come for 1 month also training for the internship. Summer internships are there. And obviously the internship or the things, we should not hamper their curriculum also, because because they have a curriculum. So they should not get affected by the curriculum, but still, if that 1 month or 2 months And the charges, this is actually the support that institutes, those who are really advanced, they can have an hands-on experience, and they charge about, for example, we charge, I am in the committee also, we charge about 1,000 rupees per month or 500 rupees per month. Yes, best when they come here, you are asking that logistics and the support for the students. See, with those kind of things, see, wherever they have a guest house, like, minimal charges, which is not that hotels we are not talking of. Per day, 200 or I don't know, 400 rupees, whatever the norms of that institute as per the governmental, staff, when they go. This way, those arrangements can be done, and couple of members can visit to my laboratory, my department here in IHW, and we do have. But coming to the, if I understand correctly, because I joined little late, maybe did I understand correctly? Maybe Murali Krishna or maybe and Ragora or somebody who are already, maybe Dr. Subhashini can speak on.
Just add to what Uday Bhaskar Rao Garu has just mentioned about Aurobindo and what was mentioned yesterday. I was part of the industry in India for last, I don't know, I just came out 5 years back, but I was there for 15 years at least. And I have trained several students from the Biotechnology India, BIRAC used to send students. So most of the corporates, they have a corporate social responsibility undertaken to train students, and we had to take at least 5 students every year. And the industry gets some benefits by way of R&D or whatever. But there is also the CSR that allows us to take students. They used to get money sponsorship from the BIRAC, but otherwise also we can take students, let's say from Andhra University or other places, to train them. That means they need to take care of if they are relocating to Hyderabad, let's say from Vizag, probably they have to take care of their housing and lodging, but the lab work, the rich experience is coming to them, free of cost. So I have done many students, right from IITs to a lot of universities, never done Andhra University student, but what I'm saying is there are opportunities. Maybe you need to tie up with certain industries. Maybe you need to make some kind of an agreement every year. We'll send 3 students, 5 students, a couple of universities, a couple of industries. Hyderabad is a hub. Vizag, you have the pharma city. I think you can work with that. Maybe it's an idea to look for, because no one is expecting you take the money or get funding to run the labs at the AU biochemistry, but you can send students to well-established labs.
Yeah, I echo what Sudha said. In fact, in my experience, because I've been in Karnataka more than in Andhra and Telangana, I actually benefited from the Karnataka state government. One of the policies they have is that BICEP program. That stands for some educational training program, but the state government of Karnataka, anybody who graduated from masters, I don't think it's limited to Karnataka, but anybody from the all around India can apply for the BICEP programs. And I've seen people come from Rajasthan, Uttar Pradesh, any part of the India, and get trained in my lab for about at least minimum 6 months or more. And that I benefited too, because I didn't have to pay them personally. Because if I'm putting my own money, I have to be more careful, because
Dr. Subramaniam, I think there are only 2 minutes left. As Dr. Subhashini said, by 7 the link will go. So Dr. Subhashini, you may please interact with reference to the comments. Subhashini garu.
Yeah, good evening.
Ragora garu. Subhashini gariki edo audio ravatledha?
Maybe thinking. No, link does not get interrupted. We can still extend it. There is a way it should be done.
That's what he said. I don't know.
Sir, Dr. Sunil want to speak something. Could you give permission to him?
Yes, good evening to all my respected seniors. I have seen and I listened to all the seniors. First of all, I congratulate and thank you to the faculty members who has taken the part and asking our views and asking our suggestions as being a alumni people. So I'm 93 to 95 batch biochemistry student and did my PhD from the biochemistry department and joined in 2001 Uttar Pradesh Bundelkhand University. My experience with the department, all the teachers are wonderful because I qualified all the competitive exams like SLECT, CSIR and GATE and with gold medal. And my experience with biochemistry department is wonderful. Teachers are wonderful. Yes, with you, and I am in the Central University, Baba Saheb Bhimrao Ambedkar University, Lucknow. So all the teachers I know, Pinky, every year our academic curriculums were changing. We changed our curriculum to the industry needs. So I hope our biochemistry faculty over there, I don't know what they are doing. But what they are looking for us is how we can contribute being alumni members. Because I'm sitting in academia, I'm not in the industry where I can see some prospects to the pass outs. Sir, really, sir, actually, one more thing is 6 months training also we started here, but we faced some difficulties with the students. We have some experiences in the Central University also. So we stopped and we started the by mode, means we have to do internal only the practical course. Yes, we are all agree with this point that during MSc we do little bit of practicals. But yes, we do practicals. In MSc also we have training. Yes, gel we are running here and we are even going for the event sequencing also we are doing. I hope biochemistry department can able to isolate the DNA and run the gel and go for the outsourcing also for this one. So what I feel is, sir, how we can contribute to the department, how we can strengthen. My suggestions are by organizing these type of seminars, you can invite the specialized persons to the department. Whenever we come to the Vishakhapatnam, we do come and we do give the lectures voluntarily. 1 point from my side I can offer. And second thing also, sir, you can send the students because I cannot train because my lab is working on the human cell culture. We are working. We are procuring the cells from the ATCC because being a central university, we had some funds. So like that, we can offer to the department. Any student who would like to come and do some experience. Not many people. I can house 1 or 2 students per annum for the human cell culture. So like that, I can offer, sir. Third thing is, I think it is very much good to you, sir. You are asking us the suggestions. So we are ready to contribute to how much we can do to the department, sir. Because we are not detached with you, sir. We are attached. So what the alumni people we are telling to the department faculty members is, sir, we are attached to you. We will be with you because of you, sir. I'm taking I got this job because you people had taught us so that we qualified the exams. So I think this time also these students also yes, there is a crunch in the employment everywhere. Even in our university also we are facing the same problem. We are also organizing the alumni meets. We are taking the help of the alumni in getting the placements. Even I feel, sir, when I go to the ICGED, my seniors had taken me, sir, because I belong to the biochemistry department. So that feeling still is there. So for this, that much only, sir, because being alumni, we should support our department. Means we take. I don't want to say anything, but I will support whatever the thing possible from my side to the department. I will use the department. Thank you, sir. Thank you for giving me the opportunity. Sir, namaskar, sir. Bhaskar Reddy, sir. Namaskar, sir. Sir, everyone, sir. Raghu, sir, Madam Garu, everyone, sir. All my seniors, sir. Namaskar, sir. Okay, sir. Thank you. Sir,
Why don't you say something about the last meeting we had?
Sir?
Radhika, we had a meeting, the committee meeting about in July, I think. We tried to suggest some things, and I don't know what Radhika should give some of her opinions on that.
Your voice is not clear. Can you please repeat, sir, once again? You are not audible clear.
Yeah, Professor Radhika actually organized a committee meeting. Myself, Sukumar, and other faculty members were involved in it. trying to give some idea of the curriculum changes and other opinions that we provided to the faculty, including Subhashini madam and Hamalata madam and Radhika madam. I think Ragora was there, I think. Can't remember, but yeah. So, Radhika Garu, any progress on that?
We have revised the curriculum and we have submitted that for the approval. And as per the faculty chairman and as per the principal of Andhra University College of Science and Technology suggested us to revise the syllabus of at least 20% of new things and retaining 80% of the old syllabus. So with that respect, we have modified the syllabus and changed the syllabus wherever there is necessity to incorporate the new things and retain the old syllabus as it is in the practical as well as the theory papers.
Okay.
By taking the advice of Professor Sivakumar Garu and Subramanian Garu and also the Suresh Kumar Garu from SKD University, all of them were our board members in this department, the Board of Studies. So I have taken the suggestions from them for every moment on moment, and then we have made the curriculum as per the requirement. May not be up to the mark, but we have made in the given time. The changes are definitely helpful for the students.
Uday Bhaskar, is it possible to arrange a meeting with VC and registrar? Biochemistry?
In what context?
I mean, we could all brainstorm in front of them and they'll see what the problems are. They may proactively put some measures into improving the department.
Yeah, if you can structure it, what exactly we want to discuss, some synopsis.
Mostly the practical training, right? That's what we need. So
See.
I would like to see the faculty apply for grants. There are lots of grants are available. They can apply for the grants.
Yeah,
They
your
can-
idea is good. Your idea is good, but that's why I'm saying our faculty, let them react.
Yeah.
What exactly is there in their university right now with reference to what yesterday's Vice Chancellor's address and principals, reiterating the facilities available, etc. And with regard to that, in addition to that, what exactly we can do, or we in the sense, you people who are well settled in that profession, biochemistry professionalism, you have. So that expertise has to be made use of by the faculty members. So how best they want to make use of your expertise so that they are the better judges. And to that extent, because university also has certain reservations, because Professor Radhika is saying, whatever suggestions you have made, again, there are comments by the principal to reduce it. The new things have to be contained with 20% and 80% of old syllabus also should be there, etc. So actually, if I don't know whether Board of Studies is the supreme body or principal is the supreme body. I have no idea. But principal can give a comment, but ultimately the Board of Studies is the competent body.
Yeah,
to suggest
definitely. Board of Studies is the body for the syllabus framing and other things. Board of Studies is the supreme body. No one can this one.
Yeah, that's what I'm saying. See, for everything, principal, vice chancellor, registrar, they are the authorities. I don't think anything can be progressed. మీరు కొంచెం దయచేసి మీరు చెప్పండి what exactly you have in mind, what exactly you have that requirement. So then only all these suggestions will be crystallized. Subramanian said we have to meet registrar, vice chancellor and all these things. They have limited time with their own activities. They will counter you saying that already these facilities are there. To what extent you have made use? If that question comes without knowing that if we go to vice chancellor and registrar, maybe we have to cut a sorry face there. So that's why you people, Subhashini garu gaani, other faculty members, you please discuss within yourselves. Before that, you please give your, you please react to whatever our own members have commented. Please.
Yeah, Good evening, respected alumni of the Biochemistry Department. As far as the syllabus is concerned, theoretically,
go ahead.
Is my voice clear?
Yeah.
Please go ahead.
Theoretically, MSc Biochemistry syllabus is very good. When we compare with the other life sciences, we have incorporated all the required syllabus for CSIR and for other competitive examinations. And every year, a handful of students actually, whoever are interested in research, they are applying and they are qualifying for CSIR examinations. So theoretically, I think we made changes, as Radhika madam said. More than 20% of the syllabus was changed. It's not restricted only to 20%. That was left to the department and the departmental staff. In annual BOS meeting, when Subramaniam sir was there, and Shivakumar sir from University of Hyderabad, and from other faculty from different universities, they gave some suggestions and we have incorporated those changes for the coming batch. So theoretically, there's no problem with the syllabus. As far as the practical is concerned, we are doing our best to conduct the practical. As Murali Krishna sir said, usually every student may not get a chance to handle page in a state university, not only in Andhra University. So whenever they go for any research or internship, they will learn the techniques. But as far as the department is concerned, we are trying our best, level best to train the students, even in the modern aspects. But and as far as the internship is concerned, every year minimum 9 to 10 students are getting chance to work with different institutes. For about 3 months, we are giving them a period, 2 months in summer and 1.5 months in winter. There is a breaking period. That too, considering the academic calendar. If we discuss that with the university authorities, we can make it a continuous 3 months. That is one thing.
Dr. Subhashini Garu,
And Academy of Sciences, they are selecting students for internship every year. They are going there. But a minimum number of students are really interested in research. Majority of the students are going for a competitive examinations. Whoever are interested in research, they are giving an additional training also. And they are getting through the competitive examination. And the drawback here is not only with biochemistry, with the entire life sciences students, the placements. So only a few students are getting placed. So that is the drawback with the life sciences students. And those whoever are interested in research, they are completing their PhD. But whenever they want a postdoctoral fellow, we need the help of alumni for guiding them to postdoctoral research. And also we need the help of my alumni for training in their particular labs. So these are the things we need from alumni.
Dr. Subhashini Garu, can I just ask one question on what you mentioned?
Yes.
You said there is internship. Is that voluntary or is that a part of the curriculum? I didn't.
Yeah, it's a part of the curriculum.
So every
At
student
the end of,
gets needs to complete that to get a master's.
Yeah.
Okay, good to know that.
At the end of the 4th semester, they have to submit project, and that is for 200 marks, madam. So after first year, summer training, 2 months, and from December 15th to February 1st, they'll be given a period to continue their project work. So they can do documentation and submit at the end of the 4th semester. It's an academic process.
Thank you. I'm glad that is already initiated.
Subhashini Garu, can I speak?
Yes.
As far as postdoctoral assignments are concerned, one is the institute name per se. Generally, if I in the past from institutes like IASC and TIFR, even if they were not having very rather productive publications, they are able to get the PDF. That was the case in the past. Now, many of the laboratories, unless and until you publish minimum 3 good papers, it is very difficult to get a postdoctoral fellowship. Because it is international competitive grants, they will see the things threadbare and select the best students. That is the way they go about selecting the postdoctoral fellowship. So it is not easy to get a postdoctoral fellowship. So it is now very, very tough. Even in institutes like CSR laboratories, people wait for 6 months to 1 year to get a postdoctoral fellowship. Either the guide has to have some connection and then he has to place them, and they will get the postdoctoral fellowship. But getting the postdoctoral fellowship on their own is going to be a difficult proposition. It's not that easy, madam.
Yes, sir. Yes. That too, postdoctoral fellow in life sciences is very difficult.
Yeah. Nowadays, you see, molecular biology and immunology, the areas which are the trust areas, people need the thorough hands-on experience. Then only they will take, madam.
I echo with what Murali said. It's really difficult to get postdoctoral fellowships unless you are doing cutting-edge technology research.
I too concur with the ideas. One of the problems with the postdoctoral fellowship is people work very hard, principal investigators and supervisors, to get money. Sometimes it comes into millions. At the end of the day, they need to provide the results. If their grant is not successful in terms of the publications, they won't get the renewal of the grant. So they need to make sure they appoint the right kind of people to deliver it. So for that, you need to recruit people who have got the right kind of a training. And that is where I think the Andhra University or any university within India should be able to offer that kind of a skills. The skills could be just hands-on experience, for example, handling a pipette or handling a machine. And the second skill, which is extremely important, is your ability to put the data into a sort of a presentable form. It could be a figure or whatever, and then able to critically analyze the data and present it in a form in a publishable quality. One of the things that happened during the COVID season was our labs are completely shut. Teaching labs are completely shut. We still ran the practicals, but the way they have been done is we provide them the data and ask the students to make the figures, critically analyze it, write a discussion on it, just like a research paper. And I think because of the financial constraints, one of the things that Andhra University could do for the students is give them data, ask them to analyze the data. There are loads of software that you can use to analyze the data, and then make figures which are of the publication quality as they appear in paper, and then interpret the data and put it in the context of the literature. So in the absence of practical training, and this is an important skill. I'm not sure whether I'm clear about it.
Yeah, I agree with you on Prasad Garu. The thing is that when I joined for PhD or Canada, that is that the professor says that you have to write your paper, you have to do your work, you have to get your own ideas. That's when I'll give you a PhD. That's it. Otherwise, you don't get a PhD. So
we need to train them to become independent, not dependent.
Yeah. Right.
So we are not providing them with recipes to follow. We need to provide them with a challenging task that they need to undertake. Because in real life, you get challenging tasks all the time. So you need to be able to become independent. I think that's where the training has to be.
Hello, this is Murthy. Hello, everybody. Can I say a few words?
Yeah, go ahead. Sorry, I'm
yeah, thank you. I mean, I joined a little late. I mean, I listened to quite a few, and also listened to Subhashini Garu's request. That was really, really nice. So we really know what's going on. These are my suggestions, my 2 cents. I know I've been out of the loop for a long, long time. So here is where we are having challenges. Let me explain, and maybe somebody can. So the challenges I currently have in my lab, for example, I don't have anybody to write really well. I struggle to get postdocs. I struggle to get good graduate students. Graduate students that go through Baylor, they're kind of okay, but again, it's another big challenge. I won't get into it right now. But getting a postdoc also, majority of the people, their CV is great, publications, blah blah blah. But when they join, they're lacking writing. Like Subramania exactly said, the communications skills are, especially from those trained from India and China, are pretty bad. So it becomes tough to like write our own papers, renew the grants. It's hard, and I understand we do consider whenever somebody comes from Andhra, I do respond and stuff like that. But I think my suggestion as far as biochemistry curriculum is concerned. So the reason I'm telling about, it's not telling about my problems or whatever, but what I'm trying to say is the gaps are in writing and communicating. That is, so somebody is there who has written or has written a review or has written a first author manuscript. That really helps. So what my suggestion in terms of the projects, they are talking about internships being compulsory. That's really nice. But I also would like to add that sometimes this is not possible to go outside and learn. It's also possible in-house training, which doesn't cost anything. There are online courses, bioinformatics, for example, you don't need any money for that. You can do online courses, learn some of these and how to write. That itself is a huge plus for the learned anywhere. I'm telling you. Nowadays, everything we have kits. It's not like the old biochemistry where you had to learn this. Now everything is available. So it's just you're
unable to hear.
not. What is important is, can you understand the concepts? Can you write something well, and can you present well? Communications are the key, in my opinion. So if the biochemistry department can incorporate training in manuscript or bioinformatics.
Sir, in between you are not audible. So the content is not communicated to us. What you are talking, sir?
It looks like he's driving, losing the connection.
Yeah.
Connectivity problem.
Yeah.
See, sir, here what I understood is that till this Dr. Murthy back to the conversation. So if that is the case, actually the writing a manuscript and looking the thing is the concern, because I don't agree some people what they say also, because I can give an example with my personal experience also. The better is that Indians are much better than Chinese, I understand that. But when it comes to if you wanted to really write the data, I think one Shiva Prasad Rao sir said is that give the data. Instead of giving data cannot be given unless until it has to be there. So I think one has to be written a publication or the review or this anything. So first, this is kind of a research methodology component can be given to the students in one of the, that few classes, and how to write a scientific paper or this kind of training may be given, and keeping that, yes, a hands-on experience is altogether is a different component. I do agree that. But coming to the communication and the analytical skills concerned, and the training can be given, few lectures can be given. In that, actually, I give those trainings in our institute to the all MDs here. So I mean, then we can orient that so on topics. They can take like a comprehensive review after understanding that. They can pen it down, and the faculty and the people who are responsible to that, they can see and they can publish that also. That is also one way of the communication and writing skills. That is exclusive. It is not included about the hands-on experience. Hands-on experience altogether, they have to virtually see that. And I understand from the HOD Subhashini madam that it's a good number of students are getting the way we used to get in high time also. Good number of people are getting CSIR and the competitive exams. I understand the theoretical component is the Department of Biochemistry is sound enough. That's what I understand that. And recently I happened to access the curriculum. When the invitation came to me, I went on to the AU website and see the department. I have in fact downloaded the curriculum, and it looks pretty good. I mean that writing that, and the sense is that if students are getting qualified into the competitive exams, particularly CSIR and other things, it is not that easy. Maybe for us, Andhra University taken a granted because we have been getting it people, so that's why we are maybe not counting, but that we have to take it as a pride. That which means that theoretically the department in a way it is sound enough. That's what I understand. And coming back to the communications, to the writing of publications, writing of manuscripts, and the analytical point of view, yes, exercise can be put into that. But hands-on experience, I do understand that you have the some of the that's what Madam is saying, that some departments are going for 9 students, are going for other places. This way, one can even explore the possibilities of the well-equipped as is. I mean, this one Sunil Babu also said that yes, we are there anytime we can come, and in terms of the knowledge skills, that what we can be giving to the department back to the in terms of the knowledge wise, which is always we are there. And if you are putting any of the research methodology and how actually the component of the scientific, how to write a professionally scientific paper is concerned, any of the course has to be given, which we are giving here, like a training courses in our institute for the outside all the, I mean, all the state people, because that is like a regular activity in our institute. So where I also used to give many of the sessions, so which always is there from our side in that angle. That's what I can say.
Yeah, I'll add a few more things. It's not just getting a practical experience. It's ability to think and design your own experiments. That's what it comes to when you're doing a PhD or master's thesis, that you are thinking on your own, you're designing your own experiments, you're trying to interpret the data that comes out of it, and you know how to write it, and you know how to present it. That's what is lacking, just the practical knowledge. Yeah, I can also pipette 10 microliters or 100 microliters. I can run a ELISA gel or this and that, but that's not what it is. You should be able to think on your own, to design your own experiment. That kind of practical training I'm talking about, not the practical training just to mix and match some test tubes. Okay.
As far as comparing with the Chinese, I would like Professor Murthy, I would like to differ with you. Indians, by far, I think they are far better than Chinese with respect to English writing. Actually, I have reviewed several papers for Chinese people. Chinese are very poor in writing compared to Indians. And in India also, we have different things. We have people who are studying in their own mother tongue up to PUC. And after BSc, they start studying in English. And how can you expect them to write? I can quote one example. My student who was hailing from the village, now he himself is a full-fledged professor. He has published 100 papers. He was not knowing how to put up the things in the initial stages. But they have learned everything. He went abroad and spent 6 years. He is an authority in cancer biology. You see, we cannot belittle our own Indians. What I feel is it is a planning how they get it and how they get exposure.
Murthy Krishna garu, Bun Hl, can I say something? Sorry, let me interrupt. Can you hear me? Can you hear me?
Murthy garu, నేను చెప్తున్నానండి, అంటే జస్ట్ ఐ వాంటెడ్ టు టెల్ యూ
one thing I don't like, the word belittling.
Please, no cross.
Nobody is, sir.
One thing, nobody is belittling anybody. Identifying the issues, trying to take it further. Nobody is belittling anybody.
Let him say, sir. Let him say. Okay, let him say. Please say, sir.
Let me finish. And I tell you, what all I have guided 14 Ps, all of them are settled well and they are in academic positions. When they come and they have written papers very badly, and it is the duty of the guide to correct them, and they will become experts in the due course of time. What I think that I feel that Indians are far better compared to Chinese, and Japanese and Germans are very poor, but their intellectual things and designing experiment may be much better, as VVS is telling. You see, the training is the most important thing. That's what I feel. And you have become such a famous professor, Dr. Murthy. I wanted to ask you, in initial stages, everybody has found the difficulty in writing this.
Right. So can I please say something? I'm not trying to belittle. I'm absolutely not. I think you got my call. I've been wrong. I'm not belittling anybody. What I was told when I first joined Baylor College of Medicine, my ex-mentor who passed away now, Randra, told me this himself. He said, Chinese are the worst in writing here, and completely Indians are better, they're learning, but he only wishes they can also be trained a little bit more in writing. Their theory is very good. That's what understanding is good, but in communicating was an issue, and it's still an issue in my opinion. I had people who I trained, they're okay. I mean, they're better than Chinese, yes. But still, I'm also talking about Europeans who are also very bad, from Russia and European countries. Their English is pretty bad also. But again, here in US, on undergrad level, also they train the students to write. All I'm trying to say is, why don't we be even better? That's all. No criticism. I am very proud of India. I'm very proud of the university. Absolutely. But my only thing is, why don't we have a little competitive edge in the writing skill? That's all I'm trying to say. I'm not trying to improve. That's all.
Fine, Murali Krishna. Enough. I think the discussion is going very nice. Kindly, please, I draw your attention to what Subhashini said. She said in her, appraisal, out of 35 to 40 students admitted every year, she didn't mention the total number of students in an MSc class. Only 8 to 10 are showing interest in doing this internship elsewhere because they want to proceed their studies further. That is the doctoral study after MSc. But again, I bring your notice to the context of discussion mentioned by Professor Raghav, that placements. That means 20 to 25% are continuing in the biochemistry field. Rest of them, that is about 70 to 75%, they are limiting their studies to MSc biochemistry and then focusing on writing competitive exams to get different employment. So I just have a question to the university faculty, that have you done any study? Because after all, whatever you study, it should result in some employment. It cannot end up with unemployment or underemployment. Okay. So if such is the case, whether the faculty has taken up any study on last 10 years after the self-financing scheme has come in, about 30 to 40 students are getting admitted every year. Out of them, how many are employed or how many remained unemployed? Because there was a discussion on this in the yesterday's inaugural function also. The principal said they have, such employment production center or something like that. But the urge for employment is much more nowadays in the biochemistry faculty. I don't know. Equally, the competition is also far better than other life science subjects. If such is the case, then can we do something? What are the methods to increase the employment with MSc candidates? What are the opportunities around? Can our friends suggest something to the faculty so that they can take up the task?
With respect to this one, getting employment for all the passed out candidates is going to be a difficult proposition, because
No. I'm not mentioning that.
Yeah.
Let
Okay.
us at least start from today, if not from past.
And I know what I want to tell you is that as far as I'm telling you once again, if you talk about the top
Opportunities. I am only asking opportunities.
Opportunities.
Yeah,
That
highlight on the opportunities available. It's not that we provide any employment.
Uday Bhaskar, not to brag, but the thing is,
Let Murali Krishna please first complete, then you please take up.
I get at least 30 to 40 resumes per week asking for a job. I can no way give it. LinkedIn is flooded with looking for jobs. I'm turning off my LinkedIn because all the MSc graduates in biotechnology, biochemistry, or anything related areas, I get at least 40 resumes on LinkedIn asking for a job. So there's no way you can fill all these positions.
Dr. Uday Bhaskar garu, one thing is that I'm just talking about maybe very standard institutes like IISc or IITs. Hardly anybody will be unemployed because the seal is there. As far as the state universities are concerned, once again, there will be preferences with respect to the universities. There are many external factors which count for selecting the candidates. As you know, I don't need to emphasize those things. Maybe I feel that at the end of the day, when you interview a candidate, the amount of subject knowledge what he is having, how he is going to impress the interview committee members is the key factor. That type of things can be trained at the departmental level itself, and thorough questioning of the candidate with respect to the subject at every 2 months or 3 months, and how they are going to present their data. If they are asked to present a new topic, let them take a journal and review the paper, and how they are going to interpret the results. These type of things, I think, definitely will train them much better with respect to selection of their candidates in a competitive things. That's what I feel.
Yeah, okay, that is one of the suggestions. Yes.
Murthy again. I just want to tell one. Hello. This is Murthy again. Can you hear me? Yeah. So I'm just suggesting this again. Why don't we include like bioinformatics as one of the courses in biochemistry, because that really be helpful, whether it's PhD or for pursuing postdoc later, because that's really the need of the day. Big data and stuff. Just a suggestion. Does anybody have any point that really helps somehow incorporate that in the course? That will really make them very competitive. This is my suggestion.
That's already in the curriculum, Murthy.
Oh, it's there. Okay.
Bioinformatics paper,
Yeah.
and statistics and bioinformatics separately, and we have a genomics and proteomics paper also. We have entered in this.
I didn't know that. No problem.
The syllabus made a separate paper for genomics and proteomics.
Okay.
We just used to have a biostatistics and bioinformatics. So we are continuing that paper along with, we are introduced in this year the proteomics and genomics paper separately.
That is
excellent. That's fantastic. Thank you. That's all.
Sudan here. I want to come.
One minute, sir. Uday Bhaskar, sir, just you said regarding the employment. I think getting all the members employment is going to be a very tough job. And if they, after the completion of biochemistry, if they are taking up a competitive examination based on the acumen or the knowledge what they got, it is most welcome, because all the finding a jobs, I think it is going to be a tough job, and because they got some knowledge and the basics and they take up the competitive exams is well and good. Actually, that's what I strongly feel that.
No. What I'm mentioning is, kindly please understand, the issue is what kind of avenues, opportunities, those things can be made available to the department from outside. That's it, because we can't polish the candidate's knowledge. We are not here to do that. The candidate has to struggle himself to compete with others. Absolutely, it is essential. That's how we all came out. But I also stopped my MSc. We did the research for some time. I wrote a UPSC exam, and now I retired as head of the state. I am from Indian Forest Service. So that is a different aptitude difference.
Right, well said. Yeah, I agree.
Sir,
can I just
maybe the A administrator will close this?
Oh, okay. So I thank you all the alumni for participating, and I also congratulate the team of biochemistry, faculty, research scholars, students, everybody who have worked very hard in making 2 days events very successful, even 3 inauguration events also very highly, with a variety of events they have organized. It's a really, it's a commendable method of organization and the team effort of the biochemistry faculty altogether. So thanks to all alumni who have come today. So thank you. Thanks a lot.
Yeah, Uday Bhaskar, one last comment here. Let's avoid the word belittling because that's not the way we all work. We have to brainstorm. Okay. If you don't like something, don't say belittling. Okay, that's
No. She already announced. She already announced. HOD has already announced.
Yeah, thank you, Subhashini garu. It is very
Sir,
well organized.
actually, why I announced this, AU administrator has to stay back if we continue like this.
Yeah.
He said he gave up to 7:45. So
It's already 7:40.
But everybody can give their suggestions because email is available. They can write it to you, and then you can consolidate the things and have the minutes of that meeting and send it to the individuals. Once again, they can see these things. Okay, in that way, there should not be any problem.
Yeah, that's a good suggestion, sir. Those who are having ideas, suggestions, as Murli Krishna sir said, you can write to the department and we'll definitely implement it if it is feasible. The department, right?
Yeah, one need not be in committees, but they can always give suggestions. That's what I feel. And IRAC give the lot of funding for the startups also. Even for MSc students are getting some of the people. So that type of things also.
Different academic backgrounds come to the department, sir.
Yeah.
They join the department, their ideas and their approaches will change, as you are all aware of that one.
Yeah.
Right?
Like everybody wants to settle down in their lives early. That there's nothing wrong in that. Unless they have aptitude for research, they should not come for research. That's what I feel.
They want to settle in life as early as possible.
Absolutely.
So
If you compare
I would like to change the word settle to
simple
succeed.
graduation, they are settling.
It's not settling, it's succeeding.
Sir, ASPR గారు ఏమిటో చెప్తున్నారు చెప్పండి సార్.
Yeah, you need to succeed in life rather than settle in life. Settle means you take it for granted everything is fine.
Yes.
Well said.
What I mean,
Yeah, well said.
you need to think about how you succeed.
Relative, sir. Okay.
Yeah.
Right.
I think can I just make a few comments based on what everybody said?
One thing is sure, sir, we have a very good students who are very much interested in doing research, right? If we meet some, something to them, definitely they will do good research and they will settle very well like other alumni members who are occupying good positions now.
I'm not disputing that.
Encourage such students, those who wanted to do research in biochemistry area or whatever it may be, right? So we wanted to encourage them. For that, we need any kind of support from the alumni so that the department, point of view, it will be an excellent support.
Could see a lot of support from
You have
everybody
that
who was here on this platform.
Yeah.
A lot of people have come forward to help you.
Sir, Shivaprasad గారు ఫైనల్ గా ఏదో చెప్తున్నారు చెప్పండి.
I can see a lot of people have come forward to help the department. I think they're nice to see. I think they've always been there.
Yes.
One of the things that biochemistry needs to do is they need to make the department and the students distinctive from the rest.
Yes, sir. Yes,
so
sir.
that is what they can sell when they're
Exactly.
applying for a job or a placement or whatever. And
Yes.
one of the things, like Murthy said, one of the problems that we have is our ability for critical appraisal, ability to write. It's not writing English. You're not
Yes.
writing poetry or essays. You're thinking and making a critical analysis of a problem. So we need to train people to ask a specific research question or develop a hypothesis, design an experiment, think about the methods they can apply, how they are going to analyze it, and how they are going to critically discuss it.
Yeah.
And those are skills, they are not easy to combine. The experiment, like one of you said, we got kits. You can do it. You can very easily learn. But these skills, they take years and years and years of training. I know Murali Krishna can vouch for me, and I think everybody on this platform can vouch for me. That takes a lot of practice. Even now I struggle writing paper. After how many years now? I've been working since 1976. So many drafts go through. And also, don't take everything that is in the textbook is right. Try and question that. There is always an alternative explanation. I think what we need to do is we need to train students to critically think and ask questions. They should not be afraid of asking questions. You may not find the answer, not today, but in a few years' time. I think that would be the core. I think that's what Subramaniam has alluded to it, if you can comment on that. But that's how we train our students. Try and train anyway.
Yeah, sure. Sure,
absolutely, sir. Absolutely. This is Dr. Rao. Absolutely, Sivaprasad, sir, what you said, because the critical thinking has to be there and there should be freedom of thought, and those kind of exercises will truly make them to take the, absolutely, the books, what we are referring is has been done by like us by somebody else before. So we don't need to actually, we always have to ponder over it and think over it. It need not be correct and we have to correct it. Therefore, actually, I took up the research job and there are no opportunities for that. I took up the research in my career as well. And the same way we did give a training to most of the Japanese universities where I worked also. This is the way actually. That is a good suggestion and we should do so. Thank you.
One only last final word. Chinese are publishing several papers. They take the help of